Hello viewers, welcome to Great Online Training. I'm Naidu. In this video, I'll talk about pharmacovigilance. First few minutes, we'll talk about what is pharmacovigilance, role of pharmacovigilance in healthcare industry, duties of pharmacovigilance professionals, careers in pharmacovigilance. In this video, we have covered a lot of topics which is related to pharmacovigilance. You want to get a basic idea on pharmacovigilance, you can just continue watching this video, and still you are interested to learn the course with the Great Online Training, please visit www.greatonlinetraining.com for live courses with us. If you have any questions, please write it in the comment section below so that we'll reply back to you in one business day. If you have any questions related to your career and job opportunities and placements, anything related to pharmacovigilance, you can write it in the comment section. We'll get back to you as soon as possible. Okay, so now let's get started. First of all, we will talk about what is pharmacovigilance. Pharmacovigilance, also call it as a PV. This is the short name, generally we call it. PV playing main role in healthcare system through maintaining the medicine safety data by assessment, monitoring, interaction among all drugs and their side effects in human. With the medicine, there are benefits and some other having side effects. When percentage of benefits is more than risks, then only medicine will be approved for marketing, or it will be retained in the market for a longer period of time. To balance and protect the patients with the use of medicine or vaccination, pharma industries following a process which is called pharmacovigilance. The PV team analyze side effects and develop safety methods to prevent them or minimize them sometimes. The other duties of pharmacovigilance is to develop and update labeling documents of the medicine. Now we will talk about what a pharmacovigilance person can do a job in real time. Pharmacovigilance professional need to collect the data from the patient who took the medicine and record there are any side effects. After collection of data, need to perform analysis of events. Based on the collected data, they have to compute benefit risk and give a suggestions to regulatory actions. Next, they need to communicate safe use of drugs. In healthcare system, the need of PV is growing day by day because we need to check side effects of different drugs. So, there is a need of PV in every country. Nowadays, so many new drugs are coming into the market at a faster rate and increase the rate of drug consumption. Because of this role of a PV becoming more important to manage a large amount of data reports to analyze side effects and benefits of particular drug. Automation is introduced in the process of PV to improve and increasing a number of reports. The main use of automation is to make a PV professional focus on accurate definition and proper use of drug and control on that use. So now we understood what is the job role of a PV person. Let's take a look on careers in PV. PV is not only career option for pharmacists, but also other science background professionals like physicians, nurses, dentists, and life science graduates can apply for PV jobs. Now, we will look into different teams involved in PV. We'll talk about them one by one. First one is case processing team. In PV, a fresher will start a career as a case processor or drug safety manager. Drug safety manager will do maintaining a database of side effects, whether it is serious adverse events or non-serious adverse event. Is it a known adverse event or it is an unknown adverse event? Everything will be managed by the case manager. Case manager can move forward to operations side if they are interested, or they can move to a technical side based on their interest. Here is a list of options for operation side job roles. Senior Drug Safety Associate, Quality Reviewer, Team Lead, and Manager. Technical side job roles: Signal Management, Aggregate Reporting Scientist, a Risk Management Specialist, Senior Vigilance Manager. Second team is under Medical Reviewer. As we already discussed about case processing team, now the second one is we are talking about Medical Reviewer. This position is for a physician who graduated in MBBS or MD. Medical reviewers, mostly MBBS persons are MD, so they have to specialize on it. You can start your career as a medical reviewer who gives ICSR reports for seriousness, casualty, and labeling. With experience, you can become a senior medical reviewer who gives ICSR and aggregate reports. With the experience, you can become a medical review manager, medical safety lead, and also chief medical officer. The third one is quality team. This team is responsible for quality management in PV. As a quality manager, you need to maintain the documents related to quality management system and in-time completion of tasks. Next, quality associate, that is responsible for QMS management. Participate in audits, SOP reviews, preparing audit plan, risk assessments. These are the different job roles for quality team. Fourth one is IT support team. To maintain the safety database, we need IT expertise in PV team. Different roles in IT supports: system analyst, system expert, IT lead, manager, backup restore and cloud expert, PV IT head. These are the different roles what we have it in pharmacovigilance in IT sector side. For make a bright future in a pharmacovigilance, we suggest take our course so that you can apply for any pharmacovigilance jobs in the market. We have been giving a training from many years so that you will have a plenty of knowledge. We will share with you, and we will share the materials, content, and the software, everything what is required for the training we'll try to provide you. I suggest go for our website www.greatonlinetraining.com for more details. By this, now you are aware of what is pharmacovigilance, a role of pharmacovigilance in healthcare industry, different duties in pharmacovigilance, careers in pharmacovigilance, job roles of PV, and we'll learn about all. In the next sessions, we are going to give you the content on different topics like one by one. This session is mainly recorded while giving a demo sessions to our regular paid students. So the first one is demo session. On the demo day, we try to introduce what is this pharmacovigilance introduction, and we'll try to see some sample cases, how a job role of a pharmacovigilance person will do in real time. The second day, we are going to talk about introduction of history and evaluation of pharmacovigilance. The next chapter, PV in clinical trials and as well as post-marketing surveillance. And the next topic, overview of pharmacovigilance sample case processing in Argus. The next topic, we will talk about different definitions of what we generally use in pharmacovigilance. The next topic is spontaneous reports, how we can able to create it. Literature and legal and regulatory reports, how we can generate it, we'll see in this topic. The next one is clinical trial reports, how we can write it. On the next topic, mainly we'll talk about post-marketing surveillance. The next one, case validity and expedited reporting. Okay, by this, we are going to cover all these topics in this video. Almost 9+ hours of video you can able to get it in this, so that it is mostly enough for the people who want to know the career, how to start as a pharmacovigilance person. You're really interested to access the database, you're really interested to see the real-time case processing and how a person will work once they get a job as a pharmacovigilance person. So if you want to know more details, please enroll to our website www.greatonlinetraining.com, and please call back on the displayed numbers. Is there any technical queries or is there any career guidance? So you can talk to the trainer and you can talk to our specialist so that you will get more details. Please enroll yourself for live training if you're interested to continue with us. I hope you like this particular session as of now. Please continue watching the remaining topics also so that you will get a complete picture. This video is recorded while giving a demo to our regular students. Thank you for watching.
Okay guys, so I'll start with the introduction topic. So meanwhile others can join later. So I'll start with my introduction. So my name is Ramya and I'm working as a pharmacovigilance trainer and I have experience of over 10 years in this pharmacovigilance domain. So I have been working, I have started my career as a case processor, and then later I have become a SME and then the quality reviewer. And then I moved on to the operational side because that's what the interest I had. And apart from the operations side, so since I manage the first team who receive the cases, so I also give the training to the new joiners in my organizations. Um, and also I'm working as a pharmacovigilance trainer for almost 3 years, and I'm a certified Medra coder. So there is some exam which is conducted by the Medra. So if you're interested, you can take the session which is conducted by the Medra team itself. Before I go into what we are going to look in the session, first we need to understand or have a basic knowledge in the pharmacovigilance. So as per this WHO, this pharmacovigilance is the science and activities that are related to the detection, assessment, understanding, and the prevention of the adverse effects or any other drug related problems. So this is the definition of pharmacovigilance. If you divide the definition and understand it, now then you will understand the multiple teams that are involved in this pharmacovigilance process. So the first team is the detection team who will identify or who will collect the adverse event information. Okay. And once the information is collected, it will go for the assessment. That is what the ICSR case processing team do, where they will receive the information from the detection team or the case intake team, and then they will process the case by when there are multiple checks are there in the case processing. And once it is done, there are some aggregate reporting team or the signal management team, and then we have the risk management team. So these are all the teams who will analyze or understand why this event is being happening, and then based on their assessment, we will provide the risk minimization activities, that is the prevention of the adverse effects. So this is the definition, and in each of this part of the definition we have separate teams are involved. Okay, so these are the different products that are covered by the pharmacovigilance. So the pharmacovigilance does not only deal with the medicine, it also deals with the herbal, traditional and supplement medications, biologic, blood products and vaccines. These three come under one category called vaccine pharmacovigilance. And then we have the medical devices, that is what we call it as materiovigilance. And then we have the cosmetics, that is what we call it as cosmetovigilance. and then veterinary and the tobacco vigilance. So we have these two as well. And the processing conventions and the guidelines are different for the medicines, vaccines, and the medical devices, and even for the cosmetics. So in our session, you are going to understand the concepts from all these products. Not only cover the products, I will also explain you about the multiple other product guidelines as well, along with the medicines. Majorly, if you are from India, you will be majorly working with the medicines or maybe vaccines. But you are from any other country, then maybe you may deal with these medical devices, cosmetics, and the veterinary and tobacco vigilance products as well. But in India, majorly we work with the medicines, vaccines, and few companies do handle the medical devices as well. But the vaccines and medicines will be our core areas. So the more in our course, majorly we cover these medicines and vaccines. I'll just give the introduction to the other products pharmacovigilance as well. Coming to our course agenda, so I will just explain you by starting from the, as you see in the definition, I will just explain in that same way. In the first week, I will just give you the sample, the what is pharmacovigilance is, how it is evolved, the history, um, and the different terminologies that are there in the pharmacovigilance. And then next, we will cover the different type of the reports. So adverse event information, we receive it from the multiple sources, like the spontaneous reports, clinical trial, or the literatures, or maybe the licensing partner or the regulatory or the legal cases. Like this, we have different type of the cases are there. I will explain each and every report type along with the example and how we receive the cases from these sources. And once we receive, what we have to do. That is what we are going to discuss in our week 1. And then in our second and third week, the 2 weeks are completely for the ICSR processing. So as a fresher, if you are joining in any pharmacovigilance company, you will deal with this, or you will be assigned with the in the ICSR case processing team only. In that again we have the multiple teams like the triage team, case intake team, data entry team, the QC team. So these are the responsibilities for the other HCPs. And if you are a physician, you will be a part of the medical review team who will review all these cases. So in our week 2 and week 3, we are going to cover the validity assessment. Whatever the reports that we are receiving, all the reports won't be valid. There are some criteria, some checks we have to do. That is what we are going to discuss. And then the most important thing in the pharmacovigilance is the event selection and coding. So I will explain that with the multiple examples by using Medra. So Medra dictionary is what we are going to use. I will give access to the Medra dictionary as well. And we are going to understand this Medra concepts with the multiple examples or the live case scenarios. And then the serious assessment, we have the labeling, causality assessment. So these three are also major for our expedited reporting. And then writing narrative. So writing narrative is one of the important factor that we have in the pharmacovigilance process. So that also I will explain differently for each type of the reports, how to write the narrative or what is the narrative template for all these reports. That is what we are going to discuss in the narrative writing session. So with that, once we complete our ICSR topics, we will move on to the core topics or the pharmacovigilance, that are aggregate review, the signal analysis, and the benefit and risk evaluation and the mitigation. So these three things will run parallel to the ICSR processing, but this aggregate reporting, signal management, and the REMS is for the experienced one. So once you have like 1 or 2 years of experience in the ICSR case processing, then you can decide on to like which team you're going to work. And so the fourth week, we are going to work on the aggregate reports. And then the fifth week, we are going to work on the signal management and also on the benefit and risk evaluation. And the last, we are going to discuss the different guidelines, what we have and which information, or like maybe you just need to have some basic understanding on the different guidelines that we have in the pharmacovigilance. That is what we are going to discuss in maybe in 1 or 2 sessions at the end of our course. So this is our course agenda. And And like I said, once you the first 3 sessions of the course are completely free. So you can attend the first 3 courses, and then you can decide whether you want to continue with the course or not. If you want to continue, you just need to confirm it back to your training coordinator. And once you're done with your initial payment, you will be given access to our Argus, that is a software we are going to provide you. So that will be we will install that software in your device, so you can also practice the cases along with me. So I'll share the multiple case examples to you on how to work these cases, so you can also practice on your own. So before I go into the question session, I will just show you how actually this case processing works. So what do you do in the company as a once you join as a case processor? What is the things or what are the steps that you do in the process? That is what I'm just going to show you for a sample. So if you have any role that is like if you get any role as a drug safety associate or the drug safety scientist or the case processor, what are the things that you do? That is what I'm going to explain you now. So first we will create the cases. For example, I'm just creating the cases randomly, but usually we get the sample AE report, and from then we will create the cases. Okay, so for the case creation, we need to enter like when the case is received and from which country the case is received, and the different report types that we have, and the products we have to code it by using the product dictionary, like what is suspect medication in the case, or maybe what is the event for that particular medication. Okay, so and then I'm going to create the case. So my case is getting created. So then a case is created. So I'm going to open the case. And so this is what the ICSR case is. So here we create the case for a single patient. So this is how our Argus look. Once we create the case, we have the multiple tabs here. So in the general tab, I will just enter who is the reporter or from where we have received the information. And then I will, in the patient tab, I will enter who is the patient, like the patient age or the gender. So we have the multiple groups here, and then gender we can enter, and if the patient has any history or any medical conditions that we can enter here, any lab data we can enter in this section. And then we have the products tab. So here is where we will add all the suspect medications in our case. And then we have the events tab. This is where we will add our events. And then this is where we have our Medra dictionary is embedded. So this is our Medra dictionary. So you can just enter. So the event selection and coding is the, like I said, it is the core for our pharmacovigilance. So we need to select and code the events properly. So like I have shown, the cases that we receive will not be as simple like this. We will it will be like very tricky situations is what we get in a daily scenarios. So but this is how we do the coding by using our Medra, and then we perform our labeling and causality in this tab. So this is where we do our labeling assessment and the causality assessment. That means we are telling whether it is a new event or already known event, or if the event is related to the medication or not. And then this is where we write our narrative, and then we submit it. So the next 3 tabs are not majorly like there is nothing that we can enter. These are just for the references purpose. Okay, and then once we do the case, we just need to save the case, and then we need to lock it, so that this is the lock option. So once you lock the case, the report will be submitted. So this is a kind of overview, but there is like in detail steps are there, like how to code the products, or which events to be considered as a medical history, or which to be considered as the events, or like how to write the narrative, how to perform the seriousness assessment, or how to understand whether the event is new event or the known event, and then how do we assess whether the event is related to the medication or not. There are multiple guidelines are there, like these guidelines are from the COMS and also from the ICH guidelines we have, and the GVP module guidelines we have. Like this, we have the multiple type of the guidelines. That is what we are going to discuss in our session. Our session is entirely based on how to apply the theoretical concepts in the guidelines into the practical view. So that you will have a more understanding from the guidelines. So whenever if you face any interview, if they ask you what is that thing and how it impacts the pharmacovigilance process, then you can easily answer them after this session. I hope you got some clarity on how and what we do in our daily life as a case processor. So, does anyone have any questions?
No, ma'am. Actually, as far as I'm
Mhm.
concerned, we are just a fresher, so we do not have any kind of question because we have not performed such cases.
Okay, so
No.
not only for the case processing, I mean like for entire course, like in the course, do you have any questions in the course, like what we are going to cover or what is your expectations? And like I said, I will provide you the training guide. So there is one guide which I have prepared, which will help you as a freshers. You no need to go and refer the multiple guidance document. So the guide will help you. So that we will provide along with that. Like I said, we provide you the software access, and also the daily recordings of the session is what we are going to provide you. Pay for the softwares as well. So yeah, apart from this, does anyone have any questions? And also, yeah, I will take the mock interviews as well, and we'll help you in the resume preparations, and we will post you whenever we know about any updates in the jobs from the references or maybe from any company. If you got to know, then we will share you the profiles on how to apply, and then how to prepare for the interview, what is their expectation, and then how do you answer the questions.
Sorry for the interruption, ma'am. Actually, what is the
Mhm.
amount which are going to charge?
It's 25,000 INR.
25,000 INR. Okay,
Mm.
ma'am. So what about that? We had paid like 3,000 rupees around. What about that?
I didn't get the 3,000 rupees.
Like where like we paid 3,000 rupees to the global pharma, like where I click the link, I
Mhm.
just go through the website, so I just paid there.
Global pharmacovigilance.
Yeah,
No,
maybe
you
it was like that.
no, I didn't get your question. So you are looking for one-on-one training, or are you looking for the recorded sessions?
I don't know what for that was, but I think I will have to connect with the customer care so that I can get the idea.
That's a global
Actually,
pharmacovigilance, but it's a
like
great online training, right?
ma'am, I just watched a video on YouTube. The video was of things by Amit. He was telling that this is the cheapest course. Like they are the charging cheapest. So I just go through that link. So
Okay.
I just paid. Uh, I paid the money. Around it was 2,900 rupees. Around 29. Yeah, 3,000 rupees around. I paid there. So I thought that this is the amount which is going to be charged for this particular course. I thought it.
No, no, that's you are referring to the different link. You said global pharmacovigilance, but it's a great online training. Maybe you got confused.
Okay, so this is. So I'm gonna connect with them. So please you continue with that. Sorry.
Okay, sure. Yeah, yeah, no
Okay.
issues. Okay, so yeah, does any others have any other questions? You can type in the chat window as well if you have any questions. And if you're not able to decide now, and the tomorrow also at the same time, you can join. Like I said, the first 3 sessions are like completely free. You can join these sessions and then you can understand what we are going to discuss in our courses. Okay. Okay, so no questions. Yeah. Yes, yeah, anyone can be eligible for the pharmacovigilance. It's not like they only look for the 2021 pass outs. They do look for the earlier years as well. So if you have some training, yes, we do provide the certification after the course completion because it's a mandatory. We have to provide, and you will also get all the interview questions that you might face as well.
Ma'am, you provide placement also after completion of course.
Like I said, the placements to provide, we need to have the market. So once we get to know about if we have any placement information, so we create the group. In that, I will share you if you have any placement, I'll let you know. And if we get to know from any of our friends or the colleagues through the references, that will also we will post in the group. So it's completely about the market. So placements, we cannot guarantee it, but because it's completely depending on the market.
Ma'am, I have one question, ma'am. Ma'am,
Yes.
after completing Bachelor of Pharmacy, like after getting graduated,
Mm-hmm.
so if we go for the job in pharmacovigilance, and can we get the job as a drug safety as like analyst? Can we get the job like?
Yes, yeah, I may be from a graduate, so I don't have any master degree. So, yeah, I have started my career as drug safety associate. Yes, you can. They just need
Okay,
the
okay.
graduation
Ma'am,
in the pharmacy.
like you didn't have any kind of experience when you joined that, right?
No, no, I was completely fresher. So, yeah, when I applied, but it was like 10 years before. But currently, the market is like, even the market is growing. The teams are enlarging. So, majorly the companies are looking for the freshers only. They're not. So that they can hire them and they can train them. So it's like for the freshers, there are a lot of opportunities are going on because if you see currently the Paraxel had some huge openings, and then the bioclinica team is also expanding because they have got some new products, and the Cognizant they're also expanding their team because they got some of the Novartis project. So like this, whenever there is anything happens, so we will have the opening. So I mean, there has to be some market so that we have the multiple openings for the freshers.
Okay. Ma'am, like in order to perform very well in pharmacovigilance, so what should be done right now as I am in third year? So what should be done by me? Like I can perform very well in pharmacovigilance. So what should be done right now? Like being in the third year,
You
what
just
should?
yeah, you just need to know. You need to be strong with the theoretical concepts of the pharmacovigilance. So you need to know about the GVP modules, the FDA guidelines, the COMS guidelines, and the ICH guidelines. So these are the core for any team. I mean, all the companies they develop their criteria or the conditions from these guidelines only. So if you're strong with these guidelines, then yes, you can do well in the pharmacovigilance.
Okay, these are like, really important in order to perform very well, right?
Correct.
Okay, okay. Like ma'am, like these are
Ma'am,
mad.
Yes,
Yeah.
Kiran. Yeah, go ahead. Kiran, go ahead. Or-
yes, ma'am. Ma'am, is there any difference that going to pharmacovigilance after graduation or post graduation? What is more beneficial
there,
after?
there won't be much, if I say. But if you have the postgraduate degree, if you are like in the it's completely depending on your interest. Like if you're interested to move in the domain knowledge, I mean, in the core areas of the pharmacovigilance, like if you want to move into the RMP team, the risk management team, or like in the scientific writing team, then in that, if you are interested there, then I suggest you can go for the post graduation. But if you are more interested with the quality check or the QA team, we have the quality assurance team as well, the audit team or the audit and inspection team. Or if you're interested with the kind of operational, that is the team lead or the team manager, or the like managing the teams, or like some therapeutic area leads. If you're interested in this kind of domain, it's like BPharma is like okay. Maybe in the distance you can do some you can have some MBA degree later as well, once you gain some experience in the ICSR, in the start of the pharmacovigilance process.
Ma'am, one more question, ma'am. Ma'am, what should be the expectation for the salary of the fresher? Like the fresher, what does a fresher should expect as per the salary? Uh,
Yeah, it will start from 3.5 to 3 to 3.5, depending on your graduate. I mean, if you are a B Pharma, then maybe you can expect a 3 LPA, but if you are M Pharma, maybe 3.5 to the 4 is what the starting salary.
ma'am, ma'am, one more question, ma'am. Actually,
Yeah.
there is a person in our village. He did maybe he spent his 2 years in QC, quality controlling in a production area, and after that
Mhm.
in the industry he spent his 2 years around, yeah. And after that, like he applied and he got selected in Sun Pharma, and right now he's working. Like he says that I'm getting 50 plus. So is it like he's a maybe what is the second post in pharmacovigilance after drug safety associate, man?
So in the drug safety associate, we have like case processing, and then we have the quality check team, and then we will have the, like I said, we have the QA team, and then we have some aggregate reporting teams as well. So yeah, aggregate reporting team is where maybe we get some higher salaries. So, but the starting won't be that much, as far as I know from my experience.
Okay. So the more we learn, the more we get, like that the process. Like the more experience you gain, the salary will be high, right?
Yeah, the
Maybe.
growth will be like pretty high. When I had joined my career like 10 years before, I was like around we I started like 16 to 18K per month, but now it's like almost my growth was like almost 7 to 8 times. So you can understand the growth. So maybe you just need to be there in the process, so that you will have the more experience, and then your salary will also increase the same. This is the one of the best field we have, where we cannot look into the salary once we join. So it will grow automatically depending on the experience.
Like there are less companies in India like who are offering the jobs in pharmacovigilance. Like there are, but the
There are many
production and
companies. No, there are many companies out there.
but
There are
we
so many
see,
companies.
but mostly they hire like a experienced person, not a fresher one.
No, for the freshers, they will have some mass drives. So yeah, there will be some freshers as well. There will be so many openings for the freshers.
Ma'am, is there any company which provides like pharmacovigilance in Delhi?
Delhi, there are many, right? So we have AppSir, and then we have TCS, and we have so many companies are there. I mean, right now I'm not remembering the names, but yeah, we have so many companies are there in Delhi. But the south of India is majorly, we have the core PV teams from the Mumbai, Pune, Bangalore, and Hyderabad and Chennai. So these are the major where the major openings will be. In Delhi also there will be, but compared to these areas, that will be a little less.
Ma'am, what about Mumbai? Mumbai, the,
Yeah, Mumbai is also, yeah, Mumbai is kind of hub. So there are like currently the TCS, there are like multiple openings are ongoing with the TCS.
okay, ma'am. That's what I was asking, ma'am.
Okay, yeah. Yeah. Does anyone have any questions?
Ma'am,
Yes, yeah, go ahead.
ma'am, like I was just asking about the interview. Like two rounds are held in interview. Like first is HR round and second one is technical round. So what are the questions which can be expected that those are being to be asked by HR in the HR round? So what are the questions?
Mm.
Like what could be the question for that? Like a couple of questions if you tell, like which are asked frequently. Those questions.
Maybe that you can refer to our YouTube videos. I have made so many YouTube videos for the questions that they will ask. So maybe you can refer. And this is a part of our session, maybe at the end of the course, maybe because it takes a lot of time. I do not want to discuss those questions now. But if you want some reference, you can refer to our the Great Online Training YouTube videos are there with the multiple questions that you can be asked.
Okay.
Okay, so yeah. Others, does anyone have any questions? Okay, so if no further questions, then I'll conclude the session. Like I said, tomorrow also you can join to have some more understanding with the pharmacovigilance, and then you can decide on it whether you're going to continue the session or not.
Ma'am, one more question, ma'am.
Yeah,
Ma'am,
yeah.
can't we
One
install
one
this?
minute, one minute, Rehan. So just we had some question in the chat window. So does CDM included? No, it's CDM is completely different topic. So that is which deals with the clinical trials, but the pharmacovigilance deal with the post marketing or the safety of the medicines, not the clinical trials. So CDM is not a part of this course. Okay, yeah, Rehan, go ahead. Rehan, okay,
Rehan, ma'am.
yeah.
Uh,
What's
ma'am,
your question?
I was asking, like, I forgot. Let me.
Okay, no, no problem. You can write down, and tomorrow also you can join and have the ask the questions if you have any.
Okay, ma'am. Yeah, ma'am. Remember.
Hmm.
Uh,
Yeah, go ahead.
yeah, I was asking about the software. Like, we use the software Argus Safety, right? I don't know how to pronounce it. Argus
Yeah,
Safety,
it's
maybe.
Argus. Yeah, it's Argus
So,
safety only.
can it be installed in mobile phone, its smartphone?
No, it's only on the device, laptop.
That's what I was asking. Okay.
Okay, so we have seen the definition in yesterday's class. So again, I'll explain because you need to understand the definition first to understand the pharmacovigilance concepts. So, as per the WHO, the PV is the science and activities that are related to the detection, assessment, understanding, and prevention of adverse effects or any other drug related problems. That means it is a science where first we detect our adverse drug reactions and then we will assess them whether they are new event or already known event, or the events are related or not related, or it is serious or non-serious event. Like that we will assess the event, and then next we will understand why these events are being happening with the medication, the main cause for the event, and the prevention to prevent that particular risk. What are the actions that we can take? So that is what the pharmacovigilance process is. And like I said, the pharmacovigilance does not only cover the medicines, it also cover the herbal and traditional and supplements, and also the biologic, blood product, and vaccine. These three come under vaccine pharmacovigilance. On medical devices, which we call it as meteorovigilance. And then cosmetics is the cosmetovigilance. And then we have the veterinary product vigilance and the tobacco vigilance. So like this, we have the multiple products that are there in the pharmacovigilance. So if you are like applying for any job role, so please look into it what they're specifying, whether they're specifying for the medicine or the vaccines, so that you can get prepared for the interviews according to it. Coming to the aims of pharmacovigilance, so we have majorly, I mean, there are many aims, but if we categorize them, we have like 4 major aims are there for the pharmacovigilance. So the first one is the detection of any safety issues or the any increase in the frequency of the known problem. That means through the pharmacovigilance process, we are doing the assessment whether it is a new event or like it is known event, and with that we will understand what is the safety problem or the risk associated with the medication. And also to improve the patient care and safety in relation to the use of the medicines and all medical and paramedical interventions. So once we get to know about the risk involved with the medication, we can let the patients or the prescribers aware of the adverse event or the risk associated with the medication, so that we can it will help the pharmacovigilance process helps in improving the patient care while using the medications. And the next one is to promote the understanding, education, clinical training in the pharmacovigilance and its effective communication to the healthcare professionals and to the public. So through the pharmacovigilance, we are educating the prescribers and I mean the physicians and also other HCPs and the end users, the consumers, about reporting of the adverse event, how to report them, and when they have to report the adverse event, so that we will get the safety profile of the medication and we can take the necessary measures to prevent certain risk. And the next one is identification of risk factors and then quantifying them. So if you see any labeling document of any product, you might have seen like this event can happen in 1 in 1,000 patient, or it can happen in 1 in 10,000 people, or maybe 1 in 1 lakh or 1 in 1 million. So this type of quantification we do in the pharmacovigilance process. When we are doing the risk management assessment, that team is responsible to quantify the risk. So that is also a aim of the pharmacovigilance. So all the processes like detection, assessment, understanding is like okay, but how to the pharmacovigilance process, how we are going to prevent the adverse events? This is also one of the most common interview question that you might face in any interviews when you apply for the pharmacovigilance fresher role. Like they may ask you what is your understanding on the prevention of the adverse events through the pharmacovigilance process. So like I mentioned, we have a team of risk management team who will take care of these prevention activities. How they will prevent? For example, there is some medication, and with that medication, there is a risk associated, that is liver toxicity or hepatotoxicity. To prevent that, what the labeling document of the product or the what guidance can be given to the patient is to continuously monitor the liver enzymes. If there is any fluctuation or any increase in the liver enzymes, then the patient has to stop taking the medication, so that we can treat the patient according to the symptoms, and the serious condition of hepatotoxicity can be prevented. So this is how, based on the risk involved with the medication, the risk management team will provide the risk minimization activity, so that other patients or the consumers cannot experience or do not experience the same event. That is how, through the pharmacovigilance process, we are going to prevent the adverse events by giving appropriate safety measures. Or maybe for a few products, if there is some risk of anaphylactic reaction, or maybe with the vaccines, you might have already, everyone might have taken this COVID vaccines, where you were like monitored for half an hour after taking the medication. So this half an hour is to check whether you are having any allergic or the anaphylactic reaction, because where the centers where you are being given with the vaccines, they will have some antidotes for the anaphylactic reactions, so that we can prevent some serious condition in those patients. That is how the risk minimization activities are like different for each and every medication. And you might have also heard like few medication has to be taken with the prescription only, and the few medication have to be administered only by the doctor who are certified to give the medication. Or maybe if anyone has taken few medications in the hospital, they have to take only in the hospital settings, and they have to be monitored for at least 6 hours, or maybe at least they have to monitor for 24 hours to see if any adverse event can happen. Like this, there may be multiple risk minimization measures. So these you might have already heard, but these risk minimization measures are what are provided by the pharmacovigilance team. So that is also one of the major aim we have, and that is how we are going to prevent the adverse events. So here I have mentioned only a few examples, and we have like multiple scenarios will be there, and for each and every event we will have the different type of risk minimization measures. So these are the aims of pharmacovigilance. And coming to the scope of pharmacovigilance, so you might be thinking like through the pharmacovigilance process, we only collect the adverse event or any event that happened to the patient, but that's not true. So through the pharmacovigilance process, we also collect the any medication errors. Medication error can be any drug dispensing error by the pharmacist, or any drug prescribing error by the physician, or maybe any drug administration errors by the nurse, or maybe by the patient by themselves, or any misuse of the medication. So these all also we will collect as a through the pharmacovigilance process. And also we collect the reports of counterfeit or substandard medicines. That means counterfeit medications are the false medication or the dummy medication. Either they contain the active ingredient or they may not contain the active ingredient, but they are developed by, so we might have heard about the drug mafia groups. They are the one who develop these counterfeit medications under one company name, and there will be some guidance will be provided to the consumers on how to check the counterfeit medication information. So these reports also we do collect, and each and every regulatory authority, even the WHO, will release the newsletters continuously if they identify any counterfeit or the substandard medications, so that everyone will be alerted about that particular dummy medication or the false medication. And the next one is lack of efficacy. So if any drug is not working, then that information also we will collect, because it will directly impact the risk and benefit balance profile of the medication. So for any medication to be in the market, the benefits of the medication must outweigh the risk of the medication. Then only the medication will be released into the market, or it can be retained in the market. So that is the reason we collect the lack of efficacy. If the drug is not working for the therapeutic indication, it's like completely not used to keep that particular product in the market. So that is why we do collect the LOE events also in the pharmacovigilance process. And also any drug misuse or drug abuse, drug dependencies, or drug withdrawal symptoms. So these all conditions also we collect in our process, and any drug interactions or drug food interaction, or any food interactions, or any interaction with the alcohol or the nicotine use, or with any other conditions. These all information also we collect through the pharmacovigilance process. So this is the scope of pharmacovigilance. Apart from the adverse drug events, we also collect these different type of events as well to maintain the safety profile of the medication. So which are the key organizations that play a major collaborative role for the PV on a global level? So majorly we have the WHO and ICH, the International Conference on the Harmonization for the medicines used in the humans. And then we have the CIOMS, that is the Council for International Organization of the Medical Sciences. So these three are the major organizations where all the country regulatory authorities are collaborated, or maybe they are the part of ICH, or maybe they are the part of under WHO pharmacovigilance program, where these three organizations are the one who provide us the guidelines. The major guidelines are from these organizations. If you see in ICH, we have some efficacy guidelines, and in the CIOMS, we have the different chapters which cover all the pharmacovigilance concepts. And then apart from this, we also have the country level regulatory agencies, like in US, we have US FDA, UK, we have MHRA, Japan, PMDA is there, Canada, Health Canada, Australia, Therapeutic Good Administration, China, FDA or NMPA, and EMA and the CDSCO for India. So like this, we have the country level regulatory agencies as well. So these three organizations will just give the guidelines that all the companies have to follow whenever they are handling any, they are giving any, approving any pharmaceutical products in their markets. But the country level regulatory agencies are the one who majorly ensure the safety, quality, and efficacy of the medicines in their country or the region. and they provide the guidelines or the legislation, and they conduct the inspections to the pharma companies as well. So though these organizations provide the guidelines, all the regulatory other agencies, they follow these guidelines. And if they have any country specific regulations, they do provide those country specific regulations as well, which the pharma companies have to follow. In all the industries that we have, the pharma industry is the one which is more regulated one, and each and every process in the pharma industry is overlooked by the regulatory agencies, and anything that happens in the pharma, it must be approved by the regulatory authority, then only the product can be released into the market. And if there is any issue, the product can be removed from the market as well by the regulatory authorities approval, or maybe by reviewing the product status, the agencies might remove that product from the market. Okay, so these are the key organizations which you need to remember, because this is also one of the most common interview question which you might face, like, oh, what are the country level regulatory agencies, or what are the entire global level agencies which handle this pharmacovigilance process or which provide the guidelines. So that is about the basic introduction. So coming to the history and evolution of the pharmacovigilance. So this pharmacovigilance is not evolved like over the night. So it has been there from from almost 150 years. So before it was there in in the 1850s, only this pharmacovigilance was there, where we are collecting the information, but it is not mandatory. But at the start of the 20th century, so there was some Biologic Control Act, where it was initiated by the US, where whatever the vaccines that are being produced must meet the quality standards, because at that time many children were died because of the contamination of some vaccine. And later FDA has extended this act to the food and drugs as well in the 1906, where all the drugs that are manufactured or the food that is being prepared has to meet these quality standards. And then later in 1937, almost like 100 patients were died because of some acute renal failure, which happened with the use of sulfanilamide drug. So this issue happened because of some solvent issue. The solvent that is used in this preparation is some diethyl glycol, which has some toxic effect. Because of this, many people were died. So that is when the FDA has initiated this Food, Drug and Cosmetic Act in the 1938. And later we have so many reports are being reported, and then there are some series of aplastic anemia cases were reported with some of the medication called chloramphenicol after the drug has been placed on the market. So that is when it was finally thought there need to be some guidance on the post-marketing safety reports. So it was started in early 1950s, but there was no guidelines or published. It was not made mandatory for the post-marketing surveillance. But in 1949, the SIOMS organization was started. So the SIOMS is the one which provide current all the pharmacovigilance process guidelines are given by the SIOMS team. And then later, the major turn of the pharmacovigilance is with the thalidomide tragedy, which we consider it as major public health disaster. So with the thalidomide, what happened is, so this thalidomide was first released into the market in 1957 as an over-the-counter remedy. That means the prescription was not required to take this medication, because the manufacturers has mentioned the product as completely safe for everyone, including for the pregnant women and also for the children as well, because they could not find a dose which is high enough to kill a rat. That is the reason now they have released it as OTC medication, and by 1960 this product was marketed in almost 46 countries. But later, in the start of, in that time, one of the Australian physician has identified this drug is also helping with the morning sickness in the pregnant woman. So he started prescribing the medication through the off-label use. And later, in the 1960s, all the children who has born to the mothers who has taken this thalidomide during the early pregnancy have developed this congenital anomaly called phocomelia, which is shortening or the absence of the limbs. So this is a newsletter from that time, though the drug makers have applied, but still there were almost 6,000 to 8,000 babies were born with the condition of phocomelia. So that is when actual post-marketing pharmacovigilance has initiated. Okay, so if we come back here, so in that during that time only, USA has initiated the Kauffer and Harris Amendment, which is currently we call it as MedWatch program. And then in 1964, the UK Yellow Card scheme was initiated, which is again the spontaneous reporting scheme. And then later in 1968, the WHO has started their program for international drug monitoring. So in that, almost 10 countries have taken a part of the program of pharmacovigilance, and later currently almost 125 countries are being a part of WHO pharmacovigilance program. And since then, all the guidelines have been developing slowly, like the Seriouness guidelines were started publishing, and then ISOP team has initiated some pharmacovigilance online courses to the organizations or to the pharma companies. And then EMA was established, and they have created some UDRA vigilance team, and we have different GVP modules are there, which we are currently, before we used to call it as volume 9A, but later in the 2012 it is replaced as the GVP modules, the good pharmacovigilance guidelines. In that we have almost 16 different modules are there for each and every process of the pharmacovigilance. And then even FDA has some IND rules or the post-marketing safety reporting guidelines document they have published. And also all the regulatory agencies from the different countries, they have also developed their own country requirement of pharmacovigilance process. So again, these are all they have taken the pointers from the ICH guidelines, and they have prepared these documents. So like this, the pharmacovigilance have been evolving. So recently also there are so many changes in the guidelines as well. So this is the history of the pharmacovigilance. I hope you got some understanding in like how the pharmacovigilance has developed, and then how we are still progressing with the multiple guidelines. Okay, so now we will see like are the drugs are safer today. You have seen like there are so many guidelines we have, like how to create the cases or how to check the validity, and we have so many risk minimization measures, and we have so many controls in the clinical trials and even in the post-marketing studies. But do you think the drugs are safer today? The answer is no, because there are so many system failures have happened. If you see, the major system fails with the Avandia, where almost, since the clinical trials are not conducted properly here, almost thousands of the people have died after taking this Avandia medication, where the patients have some cardiac issues after taking this medication taking this diabetic medication. And later with the Chantix, which is a Pfizer product, during the clinical trials, they have not considered the suicidal ideation or any suicidal symptoms as related to this medication. They have done some mistakes when they are assessing the cases, but after the product is being placed on the market, almost hundreds of the patients were died with the suicide. So that is when they have considered it as a risk associated with the Chantix, and they have used some restrictions on prescribing it. But if you see, almost there are many people have died after taking the medication. With the Vioxx, so Vioxx is considered as one of the major disaster that happened in the recent time, that is at the start of 2000, is when this Vioxx incident happened, where the company has buried the information about the cardiovascular risk associated with this Vioxx. So with any COX-2 inhibitor medication, there is a risk of cardiovascular arrest, so which was happened with the Vioxx as well. But the manufacturer did not report the events as related to the medication while submitting the information. Again, they have made the events as unrelated, which has caused almost, again, thousands of the people have died after taking the medication after being placed on the market. Though later this product was removed in the market, still the damage has done. And with the Baycol, so it's a statin medication. So like with any statin product, we have risk of muscle tissue breakdown. The same issue we had with the Baycol as well. So but here Bayer has voluntarily recalled the medication because of almost 40 deaths were reported with this product. And with the Tisabri, so Tisabri is a multiple sclerosis medication. So as you're aware, maybe there is no treatment for the multiple sclerosis. But with the Tisabri, there is one condition we call it as PML, which is encephalopathy condition. This happened at the time of Vioxx. So that is the reason the regulators did not want to take any risk with this medication. So they did not approve the medication. But still, in the clinical trials, many people have died because of this PML condition. So though we have so many regulations, still there is some associated risk is still happening, or there are many people are dying because of the adverse event. And if you see the statistics from the published articles of the journals, almost ADRs are causing 5,700 deaths per year in the UK. And in USA, from the multiple studies, they have identified 6.7% of hospitalizations are due to adverse drug reactions. And in that, 0.32% of cases are like having outcome of death. That means almost 106,000 individuals are dying because of the adverse drug reactions. And the percentages are same with the different countries as well from the multiple studies. And between 1960 to the 2013, there are almost 462 safety related withdrawals have happened. In that, market life is less than 2 years for 31% of the medications, and market life is less than 5 years for the 50% of the medication. So you can understand the risk associated with the medication. That is why through the pharmacovigilance process, we need to be very vigilant whenever we are assessing the case, and then whenever we are categorizing the risk and giving the preventive measures. That is the importance of pharmacovigilance process we have. And if you see the recent recalls, so these are the few products which are recalled from the market in the last couple of years. So in the 2020, there is this drug called Belvic, which is a weight loss drug. It was removed from the market because the risk of cancer is outweighing the benefits. And EMA has recalled one of the medication called Picato because of the skin cancer risk, which is actually used for some keratosis. But there is some risk associated with this medication. So they have recalled this medication in the 2020. So like this, so there are so many products are still being withdrawn from the market. So you must have heard about the multiple conditions of or the multiple events that are being reporting. And if you follow the regulatory agencies, they do keep us update on the recent recalls that they have done. If they have recalled any batch, they do inform us the batch numbers, lot numbers, everything as well, along with the product details. So that's how important it is our job. So we need to make sure we know we have the strong theoretical knowledge so that we can help us in creating the cases, and then we can help us in the assessment of the information. So that is the introduction that I have planned for the today. So now we can have the question and answer session. So if anyone have any questions, we can have the discussion now.
Ma'am, what does anaphylactic means? You just
It's
taught
an
about the term
allergy. Yeah, it's a severe allergic reaction.
allergic reaction.
Severe
Uh,
allergy where the patients are at risk of choking, or even it's a very kind of serious allergic reaction where the patients may die if they have anaphylactic reaction.
so, ma'am, how do we check this? Using the softwares and all?
What is that? What do you want to check? I mean, we get
Like,
the events in the adverse event report form that you will understand in the future sessions. Okay, how do we get the reports and how do we check everything? There is like in detail guidance we have. That again, like I said, I'll take you through in the next sessions.
okay, okay. Thank you.
Okay. Okay, so yeah, does anyone have any other questions? Can type in the chat window as well, because since we have some time left, we can still discuss few questions. Okay, so like I said, I mean, we have discussed about the basic introduction concepts in our yesterday's session. So where we had discussed what is pharmacovigilance, how it has evolved, and some kind of history, like how the different regulations have been initiated by the multiple organizations. That is what we have discussed in the yesterday's session. So today we will understand how the pharmacovigilance works in the pre-marketed products and how the pharmacovigilance works in the post-marketed products. For that, first we need to understand how the drugs have been developed. So now if we see in the drug discovery and development, we have the multiple process. So we have the multiple process there. So first we have the drug discovery. In the drug discovery, again, there are some multiple stages are there, where the first researchers will do the initial assessment on like for which disease they are going to develop the drug. They will research about that particular disease, what is the mechanism of action, and what are the different enzymes or the receptors or the ion channels involved in that particular disease. These are all the things that they will understand in the basic research. And then later, there will be target identification and validation phase. That means here the researchers will identify which target, that means in our body, either it can be any ion or the receptor or the enzyme, where our drug will go and affect on that particular target. That is the target identification. And then once it is identified, that target will be validated by using multiple animal models. So they will give they will I mean, they particularly inject that maybe that kind of disease into that particular animal subject, and then they will look into it whether it is a correct target for the medication or not. And then the next phase is the lead generation and optimization phase. In the lead generation and optimization, here the researchers will identify the lead compound. Lead compound means the active pharmaceutical ingredient in any pharmaceutical formulation. So that will be identified first. So for that, we have the companies will do the multiple screening analysis. It is not a single step. They may screen thousands of the compounds to identify the better one. So how do they know like which drug is going to work? So once they do the basic research on the disease, they will get to know which drug can affect on that particular target. And then later, they will search whether there are any literatures published, like maybe this class of medications can be effective of that particular disease. Or maybe each and every company will have some compound library where they develop their own compounds and keep it in their libraries. That also they can use for the lead identification. So once they have done their identified the lead, they will optimize it. Optimization stage means whatever the pharmaceutical ingredient that they have identified, they make it as suitable for the human use. That means it should not have more toxic effect and it should cause less side effects. So these are the factors they will consider in the lead optimization. For that again, they will do so many animal studies and some in vitro trials in vivo both. So they will do some enzyme analysis in the laboratory or maybe in the animals. They will give the particular drug and then they will see how whether it is causing any side effects or not. So this is the basic drug discovery phase. So in the drug discovery phase, as a pharmacovigilance associates, we do not have any role, but we do to aware of the basic information about our product. So it is best to have some understanding on how the drug is developed. Like if you are going to work for any company, we are going to work on their products. So for our better assessment of the causality or maybe for our checking whether this event is possible with the medication or not, for that we need to know the basic information about the medication. So that is why I'm just giving an introduction how the drugs are discovered. And the next one is the preclinical development. So the preclinical development is mostly non-clinical data or the non-clinical trial which we conduct in the animal models. And the basic information we conduct in the preclinical development are the ADME, the pharmacokinetics, and the potential benefits from the particular medicine and the mechanism of action, how the drug is going to work, and the best dosage and the best route will be identified in the preclinical trial, and the side effects, the most common side effects, and whether it can affect any gender, race, or any ethnicity groups, and interaction with any other treatments, and effectiveness compared to the similar drugs. So this basic information we will learn from the preclinical development. Again, this is done by the research and development team or the clinical team, but the information, whatever we get it from the preclinical development team, we use it in our aggregate reports preparation. So we have like three phases, multiple phases in the pharmacovigilance, where the first one is the case intake and case processing, where we handle the adverse event report and then we submit them. And the next phase, we have the aggregate reporting and the signal management. In the aggregate reporting, we handle the products, like what is happening with the product, what is the safety profile of the product. So that is why whenever we are preparing these aggregate reports, we need to write the information about the non-clinical trial safety information in our aggregate reports. So here is where the pharmacovigilance is involved in the non-clinical trials, that is preclinical development, where we report all the information about about the adverse event experiences to the regulatory authority by updating them in our aggregate reports. And the next one, so once the non-clinical trials are done, usually the authorities, so the companies will submit some requests to the authorities to start their trials in the humans. So once they get the approval, that is when the clinical trials will start. And again, the clinical trials, we have multiple phases like the phase 1, phase 2, and phase 3. So if you see, the phase 1 is majorly to determine the pharmacological actions and tolerability. So this will be done for some months, and a very small number of patients will be take a role part in this phase 1, maybe like 10 to 30 or maximum 100 patients are the one we enroll in the phase 1 trials. And the population we enroll in the phase 1 are the healthy population. So, or maybe some patients with the targeted disease, like for example, cancer or the tuberculosis or something. And then the type of studies are unblinded, since we are enrolling the healthy population. So it has to be unblinded only. We cannot blind the information, and it has to be with the study medication only. And the information that we learn from the phase 1 trial is the pharmacodynamic profile in the human subjects, because this is when we are starting our actual trials in the human. So we need to understand whether there is any difference in the animal studies and with the humans. That factor we have to understand here, and the pharmacokinetics as well, and the tolerated dose. So in the tolerated dose, we will identify what is the minimum dose required, or maybe what is the maximum dose that the healthy volunteers can take without having any adverse events. That is the dose we will identify from the phase 1 trials. So now, where is pharmacovigilance involved in the phase 1? So the clinical trials are conducted by the clinical trial team, and we don't have any role in the clinical trials. But whatever the safety information that we are getting from the clinical trials, we have to submit to the respective agencies and also to the ethics committees and some IRB boards. That is where the pharmacovigilance is involved in submitting the reports to the multiple authorities, and also assessing the information whether the reported event can be because of the medication or not. That is also one of the major thing that we do in the pharmacovigilance process. And the next one is the phase 2. So once the phase 1 trials are done and the results are published, it will go for the phase 2, where we majorly evaluate the safety and efficacy, and it is done for some months and the years. And the sample size here we select is a large patients we select, and the population with the target disease, For example, if you are doing any medication for the cardiovascular, then we will enroll the patients with that particular disease. And the sample size maybe in like hundreds, maybe 300 to 1000. That is what we enroll in the phase 2 trials. And here we have some blinded trials and some active control. So we have the different type of the trials. That is again, I will discuss in detail when I discuss about the clinical trial reports. And the information that we learn here is about some drug-drug interactions and the drug-disease interactions, efficacy at the various doses, and the patient safety. So here, usually how the phase 2 are done is, again, they will divide the phase 2 trials into phase 2A and phase 2B. In phase 2A, they majorly focus on the efficacy factor, and in the phase 2B, the clinical trial team majorly focus on the safety information. So whenever they are checking the efficacy, they will divide the patients into multiple groups where one group of patients will receive, for example, their like minimal dose information they have to identify. So certain group of patients will receive 10 mg and another group of patients will receive 20 mg and then 30 mg, 40 mg, 50 mg. Like this, since we already know what is the maximum dose from the phase 1, till then they will divide the group of patients in such a way, and then they will see how effective the efficacy of the medication is at, like they have to see like at which dose that medicine is having high efficacy. That is what they will understand from the phase 2. So, and also safety, any short term adverse events can be identified from these phase 2 trials. And then we have the phase 3 trials. So the phase 3s are the larger trials where we majorly evaluate the effectiveness and the risk and benefit ratio. And after the phase 3, since the product will go for the marketing authorization, and all the products are marketed based on their efficacy and risk and benefit balance. So that information is what we identify from the phase 3 trials. So these are done for some years, and these are the larger where thousands of the people will be enrolled into the phase 3 trials with the diverse population that means phase 3 can be done in the multiple countries, and then these are mostly randomized in the control trials. And here what we will identify is the dosage intervals. That means whether it has to be given once a day or once in a month or like every weekly or once in 2 weeks. So these dosage intervals are identified from the phase 3. And risk and benefit information and efficacy and the safety, this is the major information we learn from the phase 3. So from all these phases, whatever the safety information we have or whatever the efficacy information we are getting, that information will reach to the regulatory authorities via pharmacovigilance process only. So we have multiple teams who handle some people will handle the case processing of SAE reports, few people will handle the risk and benefit evaluation, and then few will handle the risk minimization measures, and then few will handle the signals or the risk that is being identified from the clinical trial, how to manage the risk, and then how to assess the causality. So that is how we have different teams in our pharmacovigilance process who take care of the information that we are getting from the clinical trials. And at the end of phase 3, and once we have the risk and benefit profile, and if the benefits are outweighing the risk, that is when the products will be released into the market. And once it is released, the phase 4 trials has to be done. So that again I'll discuss in detail in the next slides. So where we monitor the long-term side effects in the phase 4, which we call it as post-marketed trials. And we need to have some here we have some diverse population with our target disease, some new age groups, and at-risk population also we enroll in the phase 4 trials. And the information that we learn here is the safety comparisons and like epidemiological data, efficacy and the safety within the large diverse population, and the pharmacoeconomics. So these are the factors also we considered in our phase 4. So now you might have understood the difference between the clinical trials and the pharmacovigilance. Clinical trial team, they will design the trials, they will enroll the patients into the trial, and then they will just give the medication and they follow the protocol of the clinical trial. And if there are any adverse events are being reported in the clinical trial participants, they will report that to the pharmacovigilance team. Okay, any adverse event information. So from then our pharmacovigilance team will take care of whether that event is, we will perform our assessment, we will select the events, and we will check whether it is identified risk or it is a potential risk. And then we update whether this event is because of study medication or not. That is, we perform the causality, and then we write down the narratives, and then we submit the information to the regulatory authority. So that is the pharmacovigilance role, and this enrolling the patients and conducting the trial is the clinical operations team responsibilities. So this is how we have the multiple phases of the clinical trials. And then after when the regulatory authorities are approving the medication, they will check the benefit and risk profile, the drug toxicity, drug efficacy, the pH, and the bioavailability. So these are all the factors the authorities will review, and then they will approve the medication. And once it is approved, we will have some post-market surveillance or post-market monitoring of the products. So this is about the drug discovery and the drug development and the clinical operations that we have. And like I said, any reports of adverse event has to be reported to the regulatory authorities within a specified time frame, and it is done by the pharmacovigilance team. And also for the clinical trials, we need to prepare some annual reports where the pharmacovigilance team is responsible to prepare these reports, and then we submit the information by updating the risk and benefit profile of the medication to the regulatory authorities. So this is the role of pharmacovigilance in the pre-marketed products or in the drug development. So does anyone have any questions still here? No questions? Okay, then I'll move on to the pharmacovigilance in the post-marketed products. Okay, so how the pharmacovigilance will work in the post-marketed, but the majority of the pharmacovigilance process is for the post-marketed products only, because from the pre-marketed products we have like a structured way of collection of adverse events from the clinical trials, but that is not the scenario with the post-marketed products. So now we will understand why the pharmacovigilance is needed after the clinical trials. Because from the clinical trials, we have already identified so much of information. We have all the safety data and efficacy data. So why we still need it after the trials? Because there are three things. The first one is that because the clinical trials are too small. Like I said, we will only enroll some 3,000 to the 500 to the 3,000 or maximum 10,000 for the products, for the medicines and for the vaccines. Maybe like 40 to the 60,000 are the ones we enroll into the trials. So if you have observed any events, like they mentioned it is a very rare event, or it is a common event, or it is very rare or maybe rare, or like uncommon event, like this they will give some categorization. So for to get that categorization, so it is done like very rare means 1 in 10,000. This event is happening in 1 in 10,000, or maybe 1 in 1 lakh patient, or maybe 1 in 1 million. Since the trials include only like some thousands of the patients, maybe one event can happen at the maybe at the 20,000 patient can have some event which was not observed before. So this information we cannot observe from the clinical trials. So the rare adverse event can only we can identify from the post-market experience. And the trials are too short, and if there are any long-term side effects which we cannot observe them from the trials, and the settings are too unrealistic, so where we have to follow some standard protocol how to enroll in the patients, like they have to be in certain age range, they should not have they should not take any other medications, or they should not have any other medical histories while taking the medication. Like this, we have so much limitations in the clinical trials. So, but once the drug is released into the market, anyone can take the medication. So that is why we will get the information from the underlying conditions or any drug interactions or the concomitant information. We can only get it from the post-marketed products. So that is why the pharmacovigilance is needed even after the clinical trials. So now if you see how the companies have to plan their pharmacovigilance for their post-marketed drugs. So ICH E2E guidelines clearly talk about how the companies has to plan their pharmacovigilance program once they are placing their products into the market. So now we will see what are the things that the companies have to consider whenever they're releasing their products into the market, especially from the safety perspective. So this is some kind of guidance which is provided by the ICH team, where it includes all the activities or the maybe how the companies have to be prepared for their early post marketing period of any new drug. And majorly the main focus will be on the safety specification and also on the pharmacovigilance plan, like how they are going to collect the adverse event information for their marketed products. So this document is divided into some multiple sections where we include the safety specification. That means in this section, the companies will include all the risks that have been identified from the clinical trials. So from the clinical trials, whatever the adverse events that have been identified, that they will document under this section. And for those events, what is the pharmacovigilance plan? Like how they're going to collect the adverse event reports, whether they have any standard way. I mean, each and every company have their own call centers or maybe their certain emails who collect the adverse event information. So like this, how they're going to collect the information, and by when they're whether they're going to submit aggregate reports or the ICSR reports, or are they still doing any post-marketed studies. That we have different kind of studies, which again I'll discuss in detail when we discuss about the post-marketing reports and the different pharmacovigilance methods. So this is what this document includes. So now if you see, this planning is required throughout the product life cycle. So this pharmacovigilance planning is not only for the initial stage, but throughout the life cycle. And it is majorly prepared based on the risk science-based approach, and here effective collaboration between the regulators and the industries. And it is across all the ICH regions. That means it is required to be prepared by all the pharma companies. So in the safety specification, so what do we include in this document? So first, we include all the risks that have been identified from the clinical trials, and we also document what is the missing information. So if you observe any clinical trials, that they will not enroll any pediatric population, elderly population, and at-risk population, or the pregnant women or the breastfeeding mothers. They will not enroll into any trials. But once the drug is released into the market, this population also can take the medication. So for those population, how we are going to collect the adverse event reports, that is what they will include in this document. Okay, so the safety specification of the pharmacovigilance planning, it includes all the non-clinical data, clinical data, and the clinical trial adverse event information, the population that are not studied, what are the interactions that have been identified, epidemiology, that is from the different geographical regions, what is the adverse event profile, and the class effects, whether this class of drug can cause any certain type of events. That is what we mentioned, and at the end again we will just write a summary of all the events that have been identified from the clinical trial and the non-clinical trials. And so then the pharmacovigilance planning. So here again we will just write the summary, and then we will provide what are the different methods that the company is going to follow. Like we have the routine pharmacovigilance method and additional pharmacovigilance method. Routine pharmacovigilance method is submitting of some ICSR reports and some PSR reports preparation or the aggregate reports preparation. So how we are going to do the basic details. And for additional, whether we are still conducting any trials for that particular safety issue. So for example, after taking some medication, the patient is going to have some decrease in the heart rate, so the patient has to be monitored for certain time. So in this kind of example where the patient can experience bradycardia after taking any medication, the safety plan can be like, the drug has to be given in the hospital settings, and the patient has to be monitored for 24 hours until his heart rate is stabilized, or to check if there is any disturbance in the heart rate. That can be the action plan for the safety issue. Like this, any safety issue that has been identified in the clinical trials, we have to provide the action plan for the safety issues. So that is what we mentioned in this document. Okay, and these are the different pharmacovigilance methods we have. So we have some passive surveillance, active surveillance, and some observational studies, and some targeted clinical investigations. So what are these methods and the different types under this surveillance that we will discuss in detail in our later sessions. But these are the different pharmacovigilance methods that the company will follow for their post-marketed products. So now let's look into one case study. So how they have identified some event and what is the pharmacovigilance plan they have developed for that particular product. So this case study is about the drug called tenofovir, and with the experience of renal toxicity. So now if you see in the mid of 2000, there are like some cases of proximal renal tubular dysfunction and other renal toxicity in few children with the HIV taking this tenofovir was being reported. So there are like, we are getting so many reports of renal toxicity after taking the tenofovir, especially in the children population. So based on that, the EMA has asked the company to conduct some post-authorization study to assess whether this renal toxicity is because of the medication or not. So what the company does is, what the company did was, so they have enrolled the children population and adult population both, and they have clearly checked the patients did not have any underlying cause of this renal problem, and then they have given the medication, and later they have identified many patients whoever was taking this medication have this renal toxicity was experienced in those clinical trial participants. So after doing the so many studies and conducting some systematic review, they have clearly highlighted this event is because of the medication, and so they have given some restriction like this drug should not be given to the children. So like this, for each and every risk that is being identified, if the risk is serious, then there will be so many studies will be conducted, and then they will decide like who has to take the medication and who should not take the medication. So that information we are giving through the pharmacovigilance process, and that's how important our PV process is, and the knowledge that we have in the PV, we have to apply them correctly, then only we can assess the information accurately. Okay, so yes, that is it I have. And does anyone have any questions in today's any topic?
Not right now from me.
Okay, we have some time, so we can discuss not only in this topic. If anyone have any further questions, you can just ask me.
I was having a question about the fees, ma'am. So actually, you are charging very high amounts. Actually, there are so many people who wanted to, like, who willing to join, but due to the charges, they are not able to join. I'm sorry. Actually, this question was hammering in my mind, so I was just like, I thought that I should not share, but I thought like I this should share this.
I mean, yeah,
Uh,
that's fine, because if you look into the course curriculum, and if you compare with some other organizations as well, if you see, they will not give you like end-to-end process training with this much of less charge. So, but maybe whatever the other organizations work, maybe, or maybe they give about the normal GVP guidelines, what are the guidelines, that is what they will let you know. the basic knowledge of the about the TV, but this is the advanced course. So where you will not only get the ICSR processing guidelines, and you will get complete exposure on how we do the processing in the industry and how we prepare the reports in detail session it is. So that is why, and even it is not that high end for the advanced course. Usually advanced courses will be at the more than 40K if you look for any other organizations.
Okay, ma'am, this was the question which was hammering in my mind, so I thought I should ask. Okay, thank you so much.
Yeah, okay, no issues. And also, like I said, we are also providing you the database. So it's like you are downloading the software in your device where you can practice on your own, and then you can work on the cases by yourself. So whenever you have time, and it's a lifetime access. Once it is downloaded, it will be there with you until you get the job to work on the cases.
Ma'am, hello.
Yes,
Ma'am,
go ahead.
I was asking that can this job can be done being at home? Like work from home is
Work from home. Currently, due to COVID, we had to work from home. But maybe once the situation is settled, maybe we need to go into the office, depending on the company that you're going to work.
like
Because majorly since we are dealing with the patient safety data, there will be so many data compliance issues if we work from outside. That is the reason majorly they look for the person who can work from office, or maybe like sometimes you can work from home depending on the situation. Few companies do offer work from home, but few companies they would only want the team to work from the office.
actually, ma'am, due to COVID-19, some companies like declared that you are supposed to do work from home. But before COVID-19, was it possible? I want to ask this. Like work
No,
from
no, that's
home
what
was
I
it
said.
possible?
Only like one or two companies from India are offering the work from home. But in some other countries, so like in US or UK, they do give the work from home option. But in India, we don't have that facility
Actually,
with all the
there is a
organization.
person there is a person in my own village, and he is in the pharmacovigilance. He is a drug safety associate, maybe, and he has been doing work from home for around 8 or 9 months, and he says that he is connected in Sun Pharma or so which is located in Mumbai, and he has been doing work from home. Like he will be called in his company after some time. He was telling like that. So maybe I was asking that work from home was
Not at all.
allowed before
Like I said,
COVID-19.
no, we have no. It will not be work with like that. Maybe with service-based companies, we should always go to the company because we work with the multiple teams. We means not a single person can handle all the things. We always work with
Now,
other members as well.
okay. Now, yeah, who is this Samruddhi? I would love to talk regarding. Like we all are fresher. Actually, I yesterday mentioned it as well that we all are fresher, and it's gonna be much better if we connect with each other. Like having a discussion with each other, we're gonna buy the course. That would much better. I was just mentioning this as well yesterday when I attended the class. So can I share my number so that I can connect with them? If they are interested, they can call up to me.
And like I said, so I mean, sharing the numbers I mean, it's not a professional way, and you can connect through the LinkedIn profile. I mean, that is the more better way to approach the other professionals. So you can take their LinkedIn profile IDs and then you can connect them, but you should not take their personal contact numbers even for the discussions. That's what I would suggest. And the other persons, they had some experience and they are at the different levels of the requirement that they have in the pharmacovigilance. So maybe you can take your decision on your own, and yeah, it's up to the others if they want to contact with you. Maybe you can share your LinkedIn profile to them so they can, and you can send their request to them so you can contact with them offline as well.
Okay, ma'am. Okay. So LinkedIn one would be much better. Okay, ma'am. Ma'am,
Yeah.
Divya just unmuted herself. So I think. Hello, Divya. What's actually I'm not able to pronounce. Hello.
Yeah, hi.
No, she's also not from India. She's from US,
I'm not
and
from India, I'm from US.
yeah, she's
Yeah,
having some different
actually,
expectation there.
whatsoever you are, actually, I don't like so much about that. I just want to be like be frank, be connected with, so that we can have a conversation regarding that, and we should go for it or not. Like I was saying like that. If you are
No,
interested, then Divya, we can just connect through WhatsApp, or we can connect through email as well. Because LinkedIn, actually, I have no access to LinkedIn, but
no, you can create the profile. I would not suggest giving personal numbers and emails. It's always through the LinkedIn if you want to connect to the professional way, Rehan. And I'm repeating myself again. So it's about their
okay, ma'am.
independent choice. We cannot ask them or we cannot pressurize them. So you can take your
Okay.
decision and they can take their decisions because they're at the different levels compared to you. You are just in your third year of the graduation, but they have already completed their doctorates and some other physicians are there here. So you cannot
That's why.
discuss about their profile, your profile. You cannot compare your profile with the others. So you can take
Uh,
your decision to continue or not, and they will continue if they like my session, they will like to continue because they know the difference that we have providing compared to the other organizations.
okay, okay, okay.
Yes.
I want to get some more information about the course. Ma'am, there is Samruddin who is asking
Yeah,
that he
I
wants
looked
to.
into it. So yeah, I'll respond to her. Yes, Samruddhi. So yeah, you want to get more information about the course means like in which concept you are looking for.
Just a general overview about the course, what you would be covering, etc.
Yeah,
And
sure. Yeah.
is there is
Let
there
me
like
know. Yeah, yeah,
is
go
there like
ahead.
a practical aspect to it? Like I think you mentioned that you will be giving access to the software wherein we can download and practice. Uh,
Yes.
so is that like would give us an insight about when we get into the job, what work we will be doing, etc.
Last
Yeah,
January, ma'am.
sure. So
Last January.
let me just open my the course agenda and then I'll let you know.
Also, the timing, would this be the time for the course?
We can decide on the timing maybe from the
Okay,
practical session. Like I said, the first three sessions will be like free. So once we get the
okay,
confirmed number of students, we will get again. We can discuss the timelines because if the only things are from the US, then maybe we can. Maybe if you have the students only from the US, maybe we can look for the other time depending on my schedule as well.
okay.
I mean, it's
Um,
just collaboration. Hmm.
and just one last question.
Yeah, yeah.
Does pharmacovigilance job involve travel as well, if you're aware about it?
I mean, at the start level, no, it will not be involved. Maybe once you
Okay,
have some experience, maybe
okay,
you may have to travel, depending on the job
okay,
role you have taken.
okay.
Okay, so this is, I mean, I'll just show you, I mean, this is the entire overview of the pharmacovigilance process, and our training is also developed on this process only. So first, in the first week, after we complete our introduction topics, we will start with the core areas of the different type of the reports, where I will explain you with the multiple examples of report types. So how we get the reports type, and then I will also show you some samples of the adverse event reports, and then once it is done, I will show you one sample case, how we create the cases in our database. And once it is created, we have some multiple ICSR steps here, like checking validity. And again, each and every session, we will have some live case scenarios I will present so that you will have an understanding like whether these are valid or not, serious or not, non-serious, and especially the Medra coding. So I have like 3 day session planned only for the event selection and coding because it is the core for the pharmacovigilance. So that also I will give you access to the Medra where you can work on your own, and I'll share all the case scenarios in the drive, and then labeling causality as well and the writing narrative as well, that will be explained with the multiple examples with the live case scenarios. And then aggregate also, we have the majorly PSUR and the DSUR are the major ones. So I will explain you by writing the PSUR, and I will show you the sample of this PSO or the DSO documents, how they are prepared, and with the signal management as well. So from the Argus, I will show you how the signals will be detected, and then how do we pull the different line listings, and how do we write the summary from those line listings. That's what we are going to discuss along with the theoretical points there. And even with the risk evaluation and mitigation and the risk management plan as well. So in there as well, how these documents are prepared and what are the points that the team will consider whenever they're writing the risk and benefit evaluation. That is also I'm going to explain with the sample format that we have. So I mean, it's like more or like practical case practical sessions only we have. So theoretical, you will find it here and there because you need to have the theoretical knowledge as well. But at the end of the course, what I have planned is I will just show you from where I have taken this pointers for the session. Because anyway, at the end, we need to refer to only guidance that is provided by the multiple organizations. So I will just show you where I have taken the reference and which information I have collected, so that you will have both the theoretical and the practical exposure at the same time.
Okay, sounds good. So are the pharmacovigilance practices in India and US, are they, are you following the same guidelines that are followed here, or do they differ?
Okay, so here, I mean, if you're working for any company, it is
Mhm.
completely depending on their product's marketing status, because one
Okay,
company can market the products in the multiple countries. So whenever
right,
any event happen, we have to submit the information to all the countries where we are where we are marketing that particular product. So
right.
that is why the guidelines are like kind of standard, irrespective of the different regulations or the regulatory authorities.
Okay, okay.
Okay.
Ma'am, except India and USA, how many countries are there which are providing jobs in a pharmacovigilance?
All the countries, each and every country have their pharmacovigilance team. It's a mandatory as per the WHO and ICH. Any country you can go and you can have the job in pharmacovigilance.
Okay, okay, ma'am. Ma'am, how long have you been in this field?
It's almost 10 plus years I have experience in the pharmacovigilance.
Okay, okay. So right now, are you working somewhere like, or just training to the people regarding this particular?
I am working for an organization, so and it's a kind of my offline thing. I'm doing it currently. Maybe I'll take up the training as the full-time role, depending on the situation.
Okay, okay. So, ma'am, in India or USA?
India, I'm from India.
Okay, you are from India. I was asking that are you working in India or USA?
For Indian company only, I'm working.
Uh, if you don't mind, may I know the name of the company?
I work for Novartis, Hyderabad.
Novartis. Sorry, ma'am. Actually, could you please, what is the company? Rebated or Rebated?
It's Novartis.
Okay. Thank you, ma'am.
Okay, so yeah. Okay, so does anyone have any further questions?
Yeah, ma'am, I have one question.
Yes, Divya, go ahead.
Previously, you told that once we get experience, we need to travel in this pharmacovigilance field.
I mean, it's like depending
So,
on the role that we have. It's not mandatory
okay, okay,
because in my 10 years of experience, I have never traveled anywhere. I just work from one company only. But if you are in the
okay,
senior management level, or maybe if you are a part of training team, and if your company has some other part, because sometimes, yeah, I travel to like Chennai or the Bangalore to train my
mm-hmm,
associates only like maybe one
okay,
week or some, depending on the training requirement, or else
okay,
like for more time, I have never traveled.
okay.
Maybe you may move
Ma'am,
on to the maybe Japan or the China if you have the teams
okay,
there, maybe for some
okay,
few weeks or the months.
okay. Ma'am, could you please share that yesterday screen recording for us?
Yeah, sure. Yeah, I'll ask my training coordinator to share the recordings to you.
Okay, ma'am, thank you. That's it.
Okay, so yeah, if no further questions, then I'll conclude the session for today. And yeah, tomorrow we can discuss what are the different terminologies that are there in the pharmacovigilance. Okay, so I hope you got our guide and the recordings for the first few sessions. Maybe I'll need to check and upload again if there are any recordings missing for the introduction classes. Okay, maybe by today or tomorrow I'll be uploading all the sessions till we are going to cover today. Okay, so now we will start with the terms and definitions that are there in the pharmacovigilance. So the first thing that we need to aware is the adverse event. What is adverse event? What are adverse reaction, adverse drug reaction? What is serious adverse event? What is SUSAR? So there are different type of adverse events are there. We need to know what are these terms, and then we need to understand how these terminologies will impact our reporting to the regulatory authorities. So the first one is adverse event. Adverse event is any occurrence or any worsening of an undesirable or unintended sign or the symptoms. It can be abnormal lab data as well, or any disease that is temporarily associated with the use of the medicine, whether or not related to the medicinal product or the procedure. That means anything that happened to the patient after receiving the medication is what we call it as adverse event in the pharmacovigilance. That means anything like maybe a patient after taking some Benadryl syrup, the patient was diagnosed with the cancer after 3 days, still we will call it as adverse event irrespective of causality. That means we clearly know in this example that the cancer was not caused by the Benadryl, still we will consider it as a event. So these type of events where there is no causality was established and there is some abnormality in the patient that happened after receiving the medication is what we call it as adverse event. Okay, so these are the examples of adverse drug reaction. Okay, so the next one is adverse reaction. So adverse reaction is any response to the product which is noxious and unintended and which occurs at the doses normally used in the man for the prophylaxis, diagnosis or therapy of the disease or for the restoration, correction or the modification of the physiological function. That means adverse drug reaction means when we establish the causality between the medication and the event, then that adverse event will become adverse drug reaction or the adverse reaction. So here the causality has to be clearly mentioned in the report, and then only we will call it as adverse drug reaction. So these are the few examples of adverse drug reactions. So these are the examples of adverse events. So where any worsening of conditions that are present at the onset of the study, that means the patient is having hypertension even before taking the medication, and later it got worsened, or any condition that was worsened, we can consider it as adverse event. Usually, to consider any event as adverse event or adverse drug reaction, the sign or the symptom must start after taking the medication, and the patient should not have any symptoms before taking the medication. But in case of worsening, then we can consider it as a adverse event, or else we will not consider them as adverse event, that the event that the patient have even before taking the medication. And any concurrent illness or any drug reactions like drug food interactions or the food interactions, drug interactions, and also any abnormal lab values or any significant abnormalities upon the lab data or any x-ray or any examinations, any surgical complications for the transplantation, and any event reported by the participant which meets the AE criteria, that anything that happened to the patient after receiving the medication. So these all will be qualified as adverse event. Now let's see what can be the adverse drug reactions. So now we know with few medications based on their pharmacological property, some events can happen which we call it as side effects. But before we consider them as side effects, they have to qualify as adverse drug reaction in our through our pharmacovigilance process. So, for example, with any diabetic medication based on their pharmacological property, the blood sugar may be decreased. I mean, the patient can have hypoglycemia as a side effect. So these will be qualified as side effect, but these are not the adverse drug reaction. Adverse drug reaction means, for example, after taking paracetamol overdose, the patient had this renal toxicity or hepatotoxicity. So this is, we call it as adverse drug reaction. So this hepatotoxicity is because of the paracetamol, and it is not a common side effect that can happen with the medication. So these kind of reactions, we call it as adverse drug reactions, where we clearly know like this event is because of the medication. And the next one is serious adverse event. So it is event, or the SAE is a defined as any adverse event that suggests a significant contraindication to the ongoing therapy. That means it any adverse event which meets the following criteria are considered as serious adverse event, like death, life threatening, or any inpatient hospitalization or prolongation of existing hospitalization, or persistent or significant disability or incapacity, or any congenital anomaly or the birth defect, or any event that requires intervention to prevent the permanent impairment or the damage. So if any event meets any of these criteria, then we will consider it as a serious adverse event or serious adverse reaction. So to understand them in detail, I mean, we have separate session planned, like when to select the seriousness criteria, how the information will be reported to us, that you will understand in detail in the later session. But the definition is any event which meets one of the outcome, then we will consider it as a serious adverse event. And the next one is SUSAR. So the SUSAR is suspected, unexpected, serious adverse reaction. So these reactions are any serious events which are suspected to be caused by the medicinal product. That means here we are establishing the causality as related to the medication, and which are not consistent with the information about the medicinal products. That means the event is not yet being observed. So if any event is being identified to be caused by the medication, then there is some team who will check further whether the labeling document or the prescribing information of the document has to be updated or not. And if they decide, then they will update the adverse drug reaction to be added into the labeling document. So that is what we will check. So we will refer to our labeling documents, and we will understand whether the event is expected or unexpected. Expected means if the event is present in the labeling document, we call it as expected, and if the event is not present, then we will call it as unexpected. And then the serious. So if the adverse event have any of the outcome, then we call it as serious adverse reaction. So this is the definition of SUSAR, and SUSAR are the events which we usually call for the clinical trial. So in the clinical trials, if any patient or any subject experience any event, then we will categorize either it as SUSAR event or other SAR or maybe non-SUSAR events. So these SUSARs have the separate timelines for the submission. That again we will discuss in the next sessions, like what are the how these cases will affect the reporting timelines for the different products. And the next one is ICSR, which is individual case safety report, which we also call it as a safety report. So it is a document which provides most complete information related to an individual case at a certain point of time. That means this document includes all the information related to the case, like what happened to the patient after receiving the medication, what are the medical history, what are the other conditions, whether the patient had any serious criteria because of the event. So these all information we prepare in a case, and then we write the summary. So this is what the ICSR report is, the individual case safety report. And the next one is adverse event of special interest. So this we call it as AESA, and adverse event of special interest is, it can be for the clinical trial products and also for the post-marketed products. It is any noteworthy event for the particular product or the class of the drug that a sponsor may wish to monitor carefully. That means, for example, if any product is causing like some AV block or the myocardial infarction, then within 24 hours of taking the medication, then we take some measures like if the patient are developing any bradycardia or the patient is having any kind of AV block of first degree or something. So these kind of symptoms, we keep a tab on it. And then if the patient are experiencing those symptoms, then maybe we will ask the patients to not to take the treatment further, to stop taking certain medications. So that type of events which are precursors for the serious event or the severe events are what we call it as adverse event of special interest. And these all symptoms will be identified by the research and development team. When in the from the preclinical trials, they will identify all the serious risk and what are the precursors for those risks, and they will the company will all those precursor events, and they will notify to all the investigators who are taking a part of that particular clinical trials, and then they will ask if any of these events also experienced by the patients, then they have to report it to the company. Because usually from the clinical trial cases, non-serious cases are not required for the submission, but only adverse event or special interest cases are required to be submitted, because these are the kind of precursors for any severe condition. So that we will have some understanding like how many patients have the possibility of experiencing some serious event. And the next one is de-challenge and re-challenge. So this de-challenge and re-challenge information is really important whenever we are doing our causality assessment. That is, we are checking the relatedness between the product and the event. So now we will understand what is this de-challenge and re-challenge is. So now the patient has taken some medication, and after taking, the patient had some, maybe the patient had severe headache after taking some medication. So now the patient has discontinued taking this medication to see whether he is able to recover from the event or not. So this kind of process where the patient was withdrawn taking the medication due to event is what we call it as de-challenge. And after withdrawing, if the patient is able to recover from the event, that means his severe headache was recovered, then the de-challenge will be positive. And if the patient is not able to recover from the event, then we call it as negative de-challenge. Okay, and coming to the re-challenge. So now after positive de-challenge, that means after withdrawing the medication, the patient was able to recover from the event. So now the patient wants to restart taking the medication. After this process of restarting the medication after positive de-challenge is what we call it as re-challenge. And if the patient's event reoccurs, that means in this example, if the severe headache reoccurs, then we call it as positive re-challenge. And if the headache did not reoccur, then we call it as negative re-challenge. So this kind of information will give strong it is kind of a strong background information that we can have for our causality assessment. Any case with the positive de-challenge and positive re-challenge will be considered as causally associated to the suspect medication. So that's how insignificant and important this de-challenge and re-challenge is. Okay, does anyone have any questions still here?
No.
Okay, so the next are this DSUR, PSUR, and the signals. So these are the different type of aggregate reports, and then we have signal is separate again. So now let's see what is this DSUR or the PSUR report is. So DSUR is developmental safety update report, where we prepare a summary of information about a study medication in a periodic way. For example, now in some countries, some clinical trials were initiated. So after 1 year, the companies have to prepare a summary of what happened in that particular 1 year, like how many patients were enrolled, what is the safety profile, what is the efficacy profile, what is the risk and benefit balance, what are the adverse events, the serious adverse events, non-serious events, everything we have to prepare and write in this document. And for the PSUR means it is for the marketed products, DSUR is for the clinical trial products. In the same way, PSUR also we prepare in a timely manner. Each and every regulatory agency has a different timelines for the PSUR, but usually we follow the standard time given by the ICH team. So again, I'll explain in detail when I discuss about the aggregate reports in detail, like how we prepare with some examples. And the next one is signal. Signal is information that arises from the one or multiple sources that suggest a new potentially causal association or a new aspect of known association between an intervention and an event or set of related events. It can be adverse or beneficial, which is judged to be sufficient likelihood to justify the further action to verify. That means anything. So in our ICSR processing, when we are doing a single report, we will assess the seriousness of the event. We will assess the causality. That means we check the related or unrelated, and then we check the labeling, whether it is a new event or already known event as per the labeling documents. After we do our initial assessment, there is the signal team who will see if there is any new event has been reported with the medication. How they will identify new? Because we have assessed the causality as related and labeling as new event, so that they will know it's a new event, and that is what signal is. And if it is a known event, if there is a frequency of for example, there is events of myocardial infarction which was reported 1 in 1000 cases before, but now the frequency was increased to 10 in 1000 or the 20 in 1000. So this also will be qualified as a signal because there is an increase in frequency of already known event. So, and if we have the information, then again the signal team, what they do is they will check whether it has to be verified. So once after the initial assessment of ICSR, it is not like we have done all the information required for the pharmacovigilance process. It is not. It is just a start of the PV process. After that, the signal team will look into each and every event in detailed manner. It's like they will contact the patients as well directly to understand why this event happened and to get the more information from the patient. That level of assessment they do in the signal process. So that is what the signal is. So where whatever the information we have received, it has to have some information where we can justify it, whether it is a positive signal or whether it is a negative signal. Okay, so these are the most common terminologies that we use in the pharmacovigilance, and rest all again we will discuss in detail. And also in the guide I have shared, there are so many guidelines are there as well. So there are so many other definitions as well, which we can understand it by reading them. And also, like I said, other definitions anyway, I'm going to explain in detail in the whenever we have the sessions on that particular topics. Okay, so now again I will explain the overview of pharmacovigilance so that you will have a knowledge on like how we are going through it from now on, because it's like kind of core PV process we are going to start. Till here it is just a kind of introduction topics. So if you remember the definition of pharmacovigilance, it is the detection and understanding, assessment, understanding, and the prevention of adverse events. That means we have some 4 phases in the pharmacovigilance. First one is detection of adverse events. For that, we have multiple types of the reports, like we have spontaneous or voluntary reports, and then we have clinical trial and the post-marketing reports. We have regulatory, licensing partner, literature, and the legal. So these can be the multiple sources where we get the information about the adverse event. So how we get the information in this process, or the different type of documents we get, that again I'll explain in detail from the next sessions. We will start discussing what are the spontaneous, and then how do we get these reports, and then how these documents looks in detail from the next sessions. So the next phase is the assessment of the information. Who will assess? The drug safety team, that is the ICSR case processing team, the individual case safety processing team. So whatever the reports we are receiving, we'll talk about a single patient, and even the phase 2 also will talk about a single patient, like who is the patient, what happened to the patient after taking certain medication. So here again we have multiple steps there, like how do we assess? First, we will check the case validity. That means all the information that we are receiving from the patient or the reporter are not valid. We have some validity checks are there. That validity assessment we do. And then later, once we confirm it as valid, then only we create a case in our database, and once the case is created, we enter all the information. Usually in Argus database, we have almost 290 plus data elements are there. In that we have some mandatory data elements, which again I will discuss in detail in the next sessions, like which information is mandatory, which are not mandatory. And the next and important process in the ICSR is the event selection and coding with the Medra. So this is the core for all the next steps that we have in our processing. That is because if we select an event incorrectly, then we may miss to select the perform the seriousness incorrectly, labeling incorrectly, causality incorrectly, and entire case may be submitted incorrectly to the regulatory authority, or it might have missed, we might miss to submit the information to the regulatory authority. So this is kind of in detail session we have later. Anyway, event selection and coding, like how this how we have to select the events, and once we select, we have different Medra conventions we have to follow to code the events, which again we will discuss in detail in the next session. And the next one are the product coding. So we code the drugs by using some dictionaries, and then we perform seriousness, causality, labeling assessment, and then you write the summary in the form of narrative, where we have some standard template we have to follow to write the narrative. And then once it is done, the medical reviewers in the ICSR team, they will look into it whether it was done correctly, and then they will provide their assessment or their comment on the case, whether they are considering it as a valid event or they are considering it as a related or a not related event. And then we, after this medical input, we submit the case to the regulatory authority at the phase 2 itself. So in the phase 2, and that means in this step, we submit all the individual patient reports to the regulatory authorities. And then the phase 3 and phase 4 talks about the product, not the patient. Okay, so product means now we will understand whatever the information we are receiving in the phase 1 and the phase 2 in a combined way we will see, and in a cumulative manner we will decide, and then we will understand whether the how many events are being reported with the product, and then in that how many are new events, are there any increase in the frequency of known events, or whether there is any risk and benefit profile change for the medication, and if the drug was withdrawn in any market. So like this we have multiple information, we will understand it, and then in that we have multiple teams again. We have aggregate reporting team, signal management team, and the risk and benefit assessment team. So the aggregate reporting team prepare the periodic reports about the product, and the signal management team handles any new information that we are being identified in the ICSR, that they will take care. That means they will review in detail whether it is really related to the medication or not. And then we have the risk and benefit assessment team, who will check because of the signal whether there is any change into the risk and benefit balance of the product. So they do some calculations there, and they do this prepare the risk and benefit assessment document. And then after this we submit these periodic reports or these summary reports to the regulatory authorities by providing what are the actions that we are going to take because of the adverse events. So the company will provide the information, but the final action will be taken care by the regulatory authorities. So few examples are like, they will ask the companies to update the prescribing information, or they will ask the companies to educate the physicians whoever is giving the medication, or like giving counseling to the patients before using the medication. Like this, any information that they can add, or maybe in severe conditions the product may be withdrawn from the market as well. So like this we have multiple different actions that also we are going to discuss in our later sessions. But this is the entire overview of the pharmacovigilance process. So now coming to the roles that you will be taking part when you join any company. So if you are a HCP and not from the not a physician, or you don't have any doctor degree, then you will take a role in the case processing as a fresher. And then once you grow in your career, within 1 year you can move into the quality review team, or maybe you can switch to the aggregate team as well. But to be a part of aggregate team, you need to have some experience in the case processing, or like 1 or 2 years of experience. It's a kind of aggregate data preparing is a kind of scientific writing or the medical writing we have. So that's what the aggregate team does. And if you are a physician, then you will be taking a role of medical review team in the case processor, or maybe even in the aggregate team. We have the two persons. One is the safety, the aggregate review scientist, and aggregate review physician. So there also you will be reviewing these aggregate reports prepared by the scientist, and you have specific role there as well. And also, anyone can take the role in the signal analysis. So in the signal analysis, the starting signal detection, signal validation, signal prioritization, and signal assessment will be done by the HCPs who do not have the medical degree. And with the medical degree, you will take the major or the main area of the signal management, that is the recommendation of action and the exchange of information. So these are the role for the medical reviewers, who has to assess the information that is prepared by the scientist, and they have to confirm what are the actions that they are going to take. That means physician is the one who will recommend what the status of the product can be, whether any labeling changes required or any training or education is required. Final action is anyway it is in the regulatory authority responsibility, but we have to provide the multiple actions that we are considering. And then we have the benefit and risk analysis team, that is RMP team. It is, again, we have two roles there. So one for the physician and one for the scientist. So scientist role can be applied by anyone who has the HCP degree, but with the medical degree you can be into the more, I mean, it's like kind of overview is the what they do with the medical degree. So they will just confirm whatever we have done it correct or not, and what is the final comment that we are going to provide from the medical perspective. And again, majorly from the aggregate data, signal analysis, and the benefit and risk analysis, it's a kind of documentation work more, where we have to prepare multiple documents, and then we have to write the summary of information from the database, whatever we are preparing in the ICSR reports. Okay, so these are the different roles that we have in the pharmacovigilance. So that means we have in the ICSR, we have the case processor, quality reviewer, medical reviewer, and then we have another submission team, E2B submission team, which is like kind of non-core PV process, but again, it come in the pharmacovigilance only, but can be done by anyone. And we have the aggregate review. So in the aggregate reporting, again, we have the scientist, and then we have the physician. Signal analysis also, we have the scientist and the physician role. And the benefit and risk analysis also, we have both, like who are the scientist and the physician. So there are multiple roles that we have in our PV. And apart from this, we also have the QA, who will be doing these audits, or they are responsible to prepare the SOPs. And then you will have some training team or the trainers, subject matter experts as well, because we need to be completely aware of this process to become this SMEs or the quality reviewers. What are the guidelines that are provided by the COMs or the GVP modules or the FDA guidelines or the ICH? Because all the companies do follow only these guidance only for their processing or any other conventions related part. Okay, so that is the overview of pharmacovigilance process and the multiple teams that we have here. Okay, so yeah, and also, yeah, one more thing I have missed, the most important part here. So we have two teams in the pharmacovigilance. I mean, not the different processes that I'm talking about. I'm talking about two level of pharmacovigilance process. One is the local pharmacovigilance team, and another one is global pharmacovigilance team. So the local pharmacovigilance team means, so now, for example, if I'm working for the Pfizer, then Pfizer is marketing their drugs in the multiple countries. So, and as a part of ICH guidelines, or the as a requirement from the WHO, wherever I am marketing my medication, I need to have a pharmacovigilance team there. Okay, so that team is responsible to collect the information about the adverse events that are experienced in their country. That we call it as case intake team or the local pharmacovigilance team, because sometimes we get the adverse events in the non-English format, so they're responsible to translate it to the English. And then each and every country has some local guidelines or the data privacy rules, which we cannot remember for all the countries. That is why that will also be taken care by the case receipt team. And if we have to do any follow-ups, we cannot go and directly contact the reporter because of again the data privacy issues. It has to be done only by the person who is there in that particular country. So that is the reason each and every company will have the case intake team from the countries wherever they are marketing the medication. So they will collect the information, the different type of the reports, they will collect it, and they will do their initial check, and they will inform us like when this case have to be submitted, if there are any different timelines, not the regular timelines that we follow, and if there is any priority is required, or if any follow-up is required, they will only generate it, or they will ask the medical reviewer to ask for the follow-up so that they can request the reporter again. That initial assessment they will do, and then it will come for the global pharmacovigilance team. That is where our ICSR case processing starts. The data entry or the quality check or everything, the in detail process will start in the global pharmacovigilance team, and from the global pharmacovigilance only we submit the reports to the regulatory authorities. Okay, so yeah, this is about the overview of pharmacovigilance process, and that is how all the industries work. I hope you got some understanding with the pharmacovigilance process. So does anyone have any questions?
Mrs. Ramataya,
You
sorry.
can call me Ramya. Yeah, you can call me Ramya. That's fine. Yeah.
Yes, I just want to know a little bit about the re-challenge. They say that before to do the re-challenge, you have to see the benefits, the risk and benefits of that. So that can be a bit dangerous as well, correct?
Yes, yeah, it is always whenever we are trying to reintroduce, it's completely depending on the benefit and risk. By comparing that only we can restart the medication for the re-challenge process. If we feel like the risk is like kind of minimal or very it's not if it is a minor risk or the moderate, then we can try for the re-challenge, and if the risk is severe, then we will not go for the re-challenge.
Okay, good. Thank you so much.
Okay. Okay, if any other questions? Okay, then if no further question, that is it I have planned for today. Then from tomorrow, we are going to discuss about the different types of the reports that we have in detail along with the multiple examples. So meanwhile, you can contact your training coordinator so that you can download the software, because maybe once we start with ICSR processing, in the meanwhile, we can start creating the cases and you can explore like how the database looks, what are the different data elements that we have. Yes, Divya, do you have any question?
Yeah, as with the PharmD degree, so if we keep like 4 to 5 years experience candidate means which kind of role we can expect?
Okay, so within the ICSR role, you can apply for the quality review team, or maybe you can apply even for the lead role as well. Since you have the 4 to 5 years of experience, you can apply for the lead role, because the operations is as important to the process that we have in the pharmacovigilance, because we get the multiple type of the cases per day, so has to be someone who manage like the case. So maybe you can apply for the lead role as well with the experience, or
Okay.
if you want to go more in the domain, then yes, you can move into the aggregate reports. Even there also we have the same operational model. We have like associates, managers, and then therapeutic heads. So we have the multiple levels there as well. Even signal also you can take pair. And if you are more interested in the safety medicine plans, you can go for the SMP, the RMP preparing this pharmacovigilance planning. The one that we have
Okay.
discussed in the earlier session from the ICH for post marketing, where we prepare the pharmacovigilance document for each and every product. That role also you can take up.
Okay, but for MR medical review role, we are not
Uh,
fit, right?
no, it's always for the MDs or the other doctor degree that they have.
Okay. Okay, thank you.
Okay.
And Mrs. Ramya, I have one more question.
Yes.
Regarding about about jobs. For example, I'm new in pharmacovigilance, and every time I just sometimes I check on the internet to see jobs, and like I said, I'm very new, and they all want experience. So how you can move from, could you start, for example, after the online training, how you can launch yourself going to that job?
Okay, so here you can search first with the drug safety associate roles. I mean, if you or you can search with the pharmacovigilance scientist role. So this drug safety associate role will be mostly for the freshers. So that role you can search, and also what you can do is you can just mention like you are well trained by the professionals. Like maybe you can mention you have some professional mentor who was trying you like in the all the PV process, so that you have some practical experience, so that you can keep it. And but yeah, for the job search role you can search only with the drug safety associate, then you will get all the fresher roles or the pharmacovigilance associate. So this type of the search, the job role search you have to do when you are searching for the fresher role.
Much very helpful. Thank you.
Ramya, even I wanted to know about what roles I can get in.
Yeah,
I
you
have,
can apply for, yeah, you can apply for both, because you are eligible to take a part both in the processing and even for the medical review. So you can search with the drug safety physician role or the medical reviewer role, or the or you can also search for the case processor or the drug safety associate role as well if you want to start from here, because that's a benefit for the if you since you have the medical degree, you have the benefit both there, where you can apply for the physician role and also for the processing role.
okay. Yeah.
And also you can directly apply for the scientific writing, that is aggregate report writing as well, without experience, because you have the degree, not starting with ICSR process.
So is it the same as scientific writing is same as medical writing?
I don't say it's the same, but yeah, it's a kind of similar job only that we do in aggregate reports. But mostly, it's kind of like we have to review so many cases, and then we have to write the summaries. That is why that job also looks like medical writing only, the aggregate reporting.
Okay.
So today we are going to, from now on, we are going to discuss the different type of the reports that we'll get, and we will understand how do we get the reports and how does these forms look. That is what we are going to understand from these sessions. So the first type of report is the spontaneous report. So now we will see what is the definition of from spontaneous reports. So traditionally, the reports on marketed product experiences are referred to as spontaneous reports, which we also call it as voluntary reports, unsolicited or anecdotal reports. So these are the different names that we have. That means any event that is being happening on the marketed products is what we call it as spontaneous reports. And as per the ICH E2D guidelines, the spontaneous report is an unsolicited communication by the healthcare professionals or the consumers to a company, regulatory authority, or other organization like WHO Regional Center or the Poison Control Center, that describes one or more adverse drug reactions in a patient who was given one or more medicinal products, and that does not derive from a study or any organized data collection scheme. That means these are the voluntary reports. If anyone experience any event and if they're voluntarily reporting to the company or to the regulatory authorities like FDA or EMA or any other country's regulatory authority, or any other third parties like directly reporting to the WHO or any poison control centers. So if they're submitting any reports to these organizations, then we consider them as spontaneous reports. And the report that we are receiving must have patient should be there, and the medication and the adverse event after receiving the medication must be mentioned in that report. So these reports are the spontaneous reports. So now how we will receive these spontaneous reports. Before that, we will see what are the strengths and weakness of spontaneous reporting. So the strengths are like these are the cornerstone of the PV, because for all the medicinal products we won't conduct some post marketing studies. We just need some routine pharmacovigilance activities for those products. So to get any adverse event information for the products which are there already in the market for like 20 years or 30 years, we will not do any studies for those products. Whatever the safety information or adverse event information, we can identify only through the spontaneous report. That is why we consider spontaneous reporting as the cornerstone of the PV. And they are cheap and easy, because the companies no need to spend some money to collect the adverse event reports. They no need to enroll the patients, or they no need to initiate any programs to receive the adverse event reports. And it encompasses all the clinical settings. That means we have no restrictions here, who anyone can use the medications as per the prescription. And the lifetime span detection of rare adverse drug reaction. So like I said, any event that is happening after 20 years or 30 years that the drug is being placed on the market, that events we can identify only from the spontaneous reports. And the major weakness here is the underreporting, because majorly we get the information from the consumers or the physicians, where they won't update all the details that is necessary for us, like the patient medical history, concomitant medications, action taken, de-challenge, re-challenge, or the treatment information or the outcome of the event. These all information will not be mentioned. So sometimes we get reports where they mention, after taking the medication, I experienced rash. That's it. They will not mention what is the medical history, what kind of medication that the patient is taking. So that is what the underreporting is a major concern that we have for the spontaneous reports. And the factors that influencing the reporting of any adverse events for the spontaneous report. That means how the patients will know, or which events they will be reporting. So the seriousness of the event, like if the patient experienced any serious event because of the event, then it can be kind of trigger where the physician or the consumer, they would want to report the information. And whether the reaction is labeled, I mean, if the information is already there in the labeling document about the prescription or the leaflet of the product, then maybe physicians or the consumers will think it is like not required to be reported again, since it is already identified to be caused by the product. So they in certain times they won't report such information. But usually, though the information is already there in the labeling document, we still need to report the information, because if we have any frequency in the number of such and certain events, we can take some risk minimization activities. And the next is length of the time of a drug has been on the market. So again, like if the product is there on market for 30 years or the 40 years, one might think maybe we no need to submit the reports now because it's already well established product. That factor also will influence the spontaneous reporting. And the promotion or the publicity about the medicine or the reaction. So if we if we release any newsletters or any kind of lawsuits are filed for any drug, in that situation, again, it will kind of trigger to the consumers and the physicians. They will think, okay, even I have taken the medication, and I have some adverse event happen, so I need to report this information. So these are the factors that influence the spontaneous reporting. So the spontaneous reports are like we collect all the serious and non-serious events, and we receive the events from the healthcare professionals. Like I said, if the physician or the pharmacist or the nurse or any other HCPs, like if they have any patient who experienced any event after taking the medication, they can directly report it. And the consumers, that is the patients or the relatives of the patient or the friends or the family, anyone can report. And the regulatory authorities, like sometimes, like as if you see the definition of spontaneous reports, where the patients will directly report the information to the regulatory authorities. So the regulatory authority will report the information back to the companies. So that is how we can get the spontaneous reports. And the legal, so sometimes a few consumers, if anyone has died because of any medication, they will file a lawsuit against that product. So if any lawsuit is filed, then we get the reports from the lawyers So those type of reports we majorly handle them as spontaneous reports, but under the category of the legal. And the company websites. So in the company websites, there will be like contact us information or report an adverse event kind of section will be there in each and every pharma company websites. So where we can directly go ahead and report the information, or the companies do share the email ID, fax, or the telephone numbers. So we can directly contact them if in case of any adverse event happen after taking their company medication. And majorly in the product leaflet only we can find all the information, like how to, in case of adverse event, and how to report the information that will be there on the product leaflet. On the social media site, sometimes few people will update their adverse event experience in the social media sites. That also we consider it as a adverse event report for our case processing. And the pharmacovigilance centers, like each and every country has the pharmacovigilance centers, which is handled by the government regulatory authorities. So they do collect the adverse event informations. And other pharma company, so for example, the patient has taken some multiple drugs, and in the drugs, the patient has clearly mentioned he experienced the drug event with some drug B. But the company which the patient has reported is not the manufacturer of the drug, but some other company is the manufacturer. So what does this company do is they will send the report to the manufacturer of this drug B. That means here we have received information from other company about the adverse event. So these type of events also we consider them as spontaneous reports. Okay, so these are the different type of the ways where we can get the information spontaneously from the multiple reporters. So now who will be considered as HCP and who will be considered as the consumer? So HCP are the medically qualified persons like physicians, dentists, pharmacists, nurse, coroners, or as otherwise specified by the local regulations. Because in few countries, the physiotherapist or someone will be considered as HCP, but in some other countries, they will not consider them as HCPs. So that is depending on the local regulations. And the consumer will be considered as the patient who has taken the medication. So majorly in the pharma companies, so whenever we have received any report from the HCP or the non-HCP, we have to categorize the information accordingly. If you remember the one case that I have processed the sample scenario before we start with the session, in that case, I have clearly mentioned like where where we will select whether the case is medically confirmed or not, because that will impact the reporting, regulatory reportings. A few countries would want only HCP reports to be submitted, and few countries would want all the other reports to be submitted. So majorly, like in European countries, mostly they would only want the medically confirmed cases to be submitted to their regulatory agency. They do not want any consumer cases to be submitted. That is why we need to make sure like we are selecting the correct reporter as the primary reporter. So now we will see what is this definition of medical confirmation. So medical confirmation is a situation in which HCP, preferably one directly involved in the care of the patient, confirms or agreeing that the circumstances as reported by or on behalf of the patient occurred, and that the facts as amended or updated in the confirmation process, and it constitute an adverse event case for which there is a suspicion by that healthcare professional of the drug causality. That means whatever the consumer is telling, like if the consumer mentioned after taking some medication, I have headache, and if the doctor also confirm, then the case will become medically confirmed case. And if the doctor clearly mentioned the consumer has the event of headache after taking the medication, and if the physician mentioned I am not aware that the patient had this kind of event, then that case will become non-medically confirmed. That is how we need to understand whether the case is medically confirmed or not. Like I said, this will impact the regulatory reporting. And whenever we are doing any cases of the spontaneous reports, there are some points which we need to remember. So the first one is all the any reports that we are receiving from the consumer or the non-HCPs. First, we have to get the information about their primary care physician so that we can send the follow-up request to get the case as medically confirmed and to get the causality assessment. And then whenever we are getting the information, we should make sure we are protecting the patient's privacy. That means we have to follow the data privacy rules. And when the event is reported as non-serious, okay, and it is already there in the labeling document, then as per the follow-up rules, we no need to send any follow-up request further because we can consider the case as closed because anyway that event is non-serious and it is already there in the labeling document. So this case, we no need to have the information about their physicians to get the more details. So we don't need to perform the follow-up. But if the event is serious or non-serious, which is not there in the labeling document, then we have to conduct the follow-up where we have to contact the consumer's physician to get the other details that are required for our causality assessment. If the patient is refusing, then we should attempt to we need to make the like usually we follow the 3 attempts to the consumer to get the information about the physician. And if the permission is obtained and if the physician is willing to contact us back, then it's good. We can just send the follow-up request and they can fill in the information, they can send it back. And if the physician is not willing to confirm or not willing to respond back, then we still have to do our 3 follow-up attempts and then we can consider the case as closed for the follow-up if you're not getting any response back. And though if we have the medical confirmation or the case is not medically confirmed, still we can create the case in our database, but rest submissions will differ depending on the different regulatory requirements. But for the case creation purpose, we should never consider the causality factor. Okay, though the case is confirmed or not, medically confirmed or not, related or unrelated, labeled, unlabeled, serious, non-serious, these all factors does not affect our case creation, but these factors will impact the reporting to the regulatory authorities. And these are the few points which we have to consider for the spontaneous reports. So how to report to the regulatory agencies? So we have some, each and every regulatory authority have their own schemes for the spontaneous reporting. Like for India, we have suspected adverse drug reaction reporting form and UK yellow card, Australia blue card, and the US MedWatch FDA 3500 form, which is a voluntary reporting form. Like this, each and every country's website or the regulatory authority have their own way of collecting the information. And how the pharma companies receive, I mean, as a pharmacovigilance person, if you're working for any pharma company, how do you get it? So we will receive from the sources which we have discussed earlier. So they can directly report, fill the adverse event form, and they can send the report to the company via email or the fax, or they can publish in the social media or in the company website or the company email that is provided to them. So these are the sources that is used by the reporters to report the information. And this is a sample ADR forms which will be filled by the reporters. So each and every country so will have the different forms. So we don't have like, okay, we get the information only in this form. There is no certain rule we have. We can get the reports in any kind of the form, but I'll try to show you as many reports as possible so that you can be familiar with the different type of the forms. So this is the one of the form from India. So though these forms looks different for the different countries, but if you look into the information will be like similar. So if you see, the reports will have patient information, the adverse event details, medication details, and the reported details. These 4 sections will be common irrespective of the different type of the forms if you will see. So in the patient details, we will have the patient information, the age, gender, and ethnicity, or the initials, adverse event, like what is the event in detail, what are the different medications that the patient is taking, what are the different tests that can be that the patient has underwent, what is the seriousness, what is the outcome, who is the reporter address. This is what the IDR forms include. And if you see the UK yellow card, so this form is slightly different. So, but the information is, like I said, it is same. We have the patient detail section, the suspect medication, and adverse event information, the seriousness outcome, other medications, other medical history, and the reported details. This is what the UK MHRA card also include. And then coming to the MedWatch, US MedWatch form. So here also same, the patient information, adverse event, product information, and if it is a device, then we need to have the device information and the reported details. So this report also have the same type of information, but these forms are different to look. So this is a standard AE template wherein the reporters can fill in the information and they can share it to us. So coming to a few sample scenarios from the internet source, like how did we receive from the internet source or from the social media, I have taken few examples. You can also look into it. So this is one of the website which is actually which this website contain only adverse drug experiences, where the patients kind of forum thing where the patients can directly come there and they can just write down their experiences, and the companies will review this. I mean, whenever they are doing this sampling from the multiple websites, they do take this information for their pharmacovigilance process. So this is the internet source case. You can read to it too. You can read through it. So the patient is directly reporting here. I took doxycycline 3.5 days, and I now have total insomnia. I feel like I don't sleep, but I do sleep and dream. It's just in my mind, and it does not recognize it as a sleep. This is a nightmare, and I stopped because I had pressure in head and eyes and the TMJ joint, wrist and ankle pain with paresthesia and irritated throat with trouble breathing. And these all symptoms are went away after discontinuing, and the patient's condition was improved. And the patient is being on Seroquel and had some insomnia from some other medications. And it took like 5 months till I felt like I was sleeping again, but now I feel like I will be like this forever. So, like this, the patient has mentioned about her experiences. So, this is how we get our spontaneous reports. If you get the AE report, that form also I'll show you, we get all the information in a structured manner that will be easy for us to process. But if you look into the internet sources, where if it is published like this, then we have to understand the case first. Then we have to do our initial assessment of case validity, and then we have to select the event, suspect medication, everything. That how we will do. Again, I'll explain in detail in the next sessions. So now, in this case, the patient is telling with the doxycycline, she's having insomnia and some pain and some pressure in head and eyes. So these are the events for the doxycycline. That is the case. So we have a patient here, the medication here, the reporter, and the events. So we will consider this case as valid, and then we will process this case as spontaneous report as a medium of internet source. And the next example from the social media source, where they have published in their Twitter or the Instagram, or the Twitter is the major for the AE reporting. Each and every company have some data, some software tools which can pull the information from the Twitter or the other in other social media sites about their adverse event report that are being published by the users. So coming to the social media source here, they have mentioned I did some weird stuff when I took Ambien. I cut banks for myself. I ate an entire huge cookie made for coworker's birthday, and then I drew dragonflies in the multiple colors of Sharpie all over my apartment walls. I never turned into a racist. So like this, she has mentioned some experiences that she had after taking some medication. So this also, yes, we do consider for the case processing. And the next example from the Instagram, someone posted like in my whole life I have like long hair, and then she was diagnosed with some Crohn's disease, so she's taking Humira, and after Humira she has had like balding was there, so her height almost fell out. And then she mentioned like she stopped taking Humira, and then she's taking some other vitamins and some other products so that her hair can come back. So like this, she has mentioned her entire experience. So this is how we get the social media source reports, where we need to read through them, and then we need to understand whether this is actually case in this information or not, and then we have to decide which information we have to submit to the regulatory authorities. Okay, so before I go into the questions, let me show you the example of standard AE form, like how the standard AE template looks, so that you will have some understanding on that point as well. Okay, so this is how the standard AE templates look. Okay, where you can see it, where it will mention like the patient details will be clearly mentioned, what happened to the patient, they'll clearly mention here, and the other medical histories. So everything will be there like this in a documented manner, where we can just pick up the information and then we can update into our database. Okay, and then we also have some other, ADR templates are like multiple types, and this is also one of the ADR template where here also it will be like in a standard way, the information will be there, whatever the adverse event, the medications, medical history, outcome, who is the reporter. So this is another case. This form is different. You can see the difference in the different forms. This is also one form, and this is another type of ADR form. These are all the templates I have shared in the shader, and I have given you the access as well. You can just go through this and you can explore them, like how these different forms look, so that you will have some basic understanding on the different types of the reports that we may get. Okay, so like this, you will get the multiple types of the reports for the spontaneous, and there is no standard. Sometimes we do get the discharge summary reports as well. Okay, just imagine like in hospital, whatever the discharge summaries that they provide, that also we will consider as uh, as a reports for our for our case processing. Okay, so this is about spontaneous report. So does anyone have any questions in the spontaneous reporting?
I have a few questions from you, ma'am.
Yes, you can just call me Ramya. Ma'am is not required.
Okay. The first question is like you said, spontaneous reporting. If someone puts it on social media, you gave a few examples. So if if suppose the patient has headache, or like with Humira, someone had hair loss. So if that is like a known side effect, so should we consider it still, or it will not be a valid case?
Yeah, so that's what I said. Irrespective of the labeling or the causality, we still consider it as a valid report.
Okay. Even if it is a known side effect, it's
Yes.
already discussed. Okay.
These
So
factors will only affect the reporting to the regulatory authorities, but not for the case creation.
for the case creation. Okay. And then like social media is so huge right now. This, I think somebody reported on the website where we have to report the adverse events. But like suppose if I am just writing a post on my account, okay, like suppose I've used a cream and it gave me a rash, and I just put it on social. Oh, I used this cream and it gave me this kind of rash, and I put up pictures and I will tell everybody, okay, don't use this. This caused this to me. So like. Does the pharmacovigilance team work on the social media as well and look for such reports? So do they scan them, or is there somebody else doing that for them?
Yeah, so here, and if your profile is like kind of public, so we have the private and the public profile. So if you're kind of in the public profile, then yes, the team, it is a separate team, the case intake team or the case detection team is different. The starting team, there will be like few people who will be checking on this adverse event report on the social media sites. They use, they will not go there and check like normally,
Each
like
and
case by case, they cannot check, right? So they just
It's
apply
so
some
huge,
data
right?
analytic
That's why
tools.
I had
Yeah,
this
yeah, it
question.
is. So there will be some data analytic tools will be there where once they
Got it.
enter their product name, it can automatically pull some information for the company.
Yeah.
And they
They
can
will
pick up
put some keywords.
yeah, they use some keyword like how we do some searches, and then they get the information.
Okay. So only those case, whatever, if the data
If it is
analytics team
big, that is
collects.
what we will get for us for the processing.
Okay. Okay. Like, and even like you said, in hospitals, they have like a doctor will have his notes and summaries. Not everybody will report everything to the authorities, or maybe not every time they get the time to report it. So like especially in a new product that is on the market. So post marketing also the pharmacovigilance team will be working on it, right? So do they
Yes.
contact the authorities and check every time about the product, or they
So
just consider the cases which are reported to them by
yes.
the
So whenever
physician
we
or
are getting the information from the regulatory authorities or other companies, since we
the...
cannot contact them by because regulatory authority, as per the data privacy, if they are receiving the information, they should not share it to the company. So we cannot contact the reporter directly. And if it is a regulatory
Okay,
authority report or other company, we can just consider that case as closed for the follow-up.
okay, 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 got it. Thank you.
Yeah. Okay, then yeah, if no further questions, then I'll continue with the next type of report, that is clinical trials. So clinical trial reports have different type of processing we have. So where these trials, I mean, first we need to understand which report are considered to be clinical trials, and the complete reporting requirements are different for the clinical trials compared to the other type of the reports that we will discuss in detail in the next sessions, like which reports have to be submitted or which we will not submit for the regulatory authorities or with the different type of the reports. So before that, we will understand what are these clinical trials are. So these are the studies that we do, the test on the um, humans, and here the people will voluntarily take the part in the clinical trials to test the medical intervention, including the drugs or the biologics or the any surgical procedures or any devices or any radiological procedures or any behavior treatment in the preventive care. So this is the definition of clinical trials, and we have already seen the different type of the clinical trials and the phase 1, phase 2, and phase 3 trials in our earlier sessions. And the pharmacovigilance, where does it work in the clinical trials? So the trial design, preparing the protocols, I'm preparing IB document. So these all will be taken care by the clinical trial team, like enrolling the patients and collecting the information and then updating their safety data, their database. So we have the clinical trial database will also be there, from where we get the information to the pharmacovigilance team. So whenever any adverse event happens or any serious adverse event happen or any seizure happens in the clinical trials, that is when the pharmacovigilance team will come into the picture, where we will receive the information from the trial team, and then we process the information, we assess whether it is related to the study medication or not, and then whether it is expected or unexpected, and then we submit the information to all the required teams that are there in the clinical trials. And then we have these annual reports which we have to prepare in the clinical trials, like we call it as DSUR reports. Every yearly we have to provide the summary of what is happening with the study medication, how many patients are enrolling, how many patients are withdrawing, what are the different events that are being reported with this product in the particular year. Like this we have to write the summary. So that summary, that report is also under responsibility of pharmacovigilance team. So, like this, any adverse event experience, if they want to communicate to all the parties, that is via pharmacovigilance process only from the clinical trials. So, this pharmacovigilance process and the clinical team, they work in parallelly. So, they share the information from the clinical team to the pharm PV team and the PV team to the clinical trial team. So, exchange of information will be keep on happening between these two teams. Okay, and then coming to the different type of the clinical trials we have. So we have seen the different phases in the earlier session, but now coming to the different types. In each and every phase, depending on the product requirement, we will have the trials are designed in a different ways. So now we will understand what are the different type of the trials that we have. So the first one is the pilot studies and the feasibility studies. So these pilot and feasibility studies are not actual studies. So they are done just before they start doing some big studies. They will see whether these studies are possible to conduct or not. So these pilot studies are done just before they start doing some big studies. They will see whether these studies are possible to conduct or not.
Are you there? I can't
cancer
hear.
drug and
Okay.
comparing with the any okay. Or anyone preparing the conducting a trial or a fever medication. So here for fever medication, no one will show the interest to take because everyone aware the paracetamol is working fine. They don't want to take a part in this medication. But if anyone is trying for any oncology drug, then the people will show interest to take a participation in that particular trial because they're getting a free treatment and to participate in the trial, maybe they're getting some money as well. So like this, they will think like which study is feasible or not. These type of the studies are done before we start with the phase 1 trials. And the next one, we have the pilot studies. So pilot studies are the small versions of the main study. Here we enroll or we check, I mean we do some minimal assessment on the patients, whether this drug can be injected or we may get the actual patients or not. So to understand that these pilot studies are done. So because maybe a few diseases will be there which are like rare, like encephalopathy or like tuberculosis, because right now the patients are not having these kind of events. So for these events we have to check certain criteria whether we can enroll them or not. So these type of the things are checked in the pilot studies, whether we can get actual patients or not for these trials, and if we get the actual patients, whether what inclusion and exclusion criteria that we can apply for the patients. So this kind of information is learned in these pilot studies. And the next and the key and the major trials are on the controlled trials. Controlled means here uh, the trials are designed to compare the different treatments. That means here we will have one standard treatment and one new treatment and the placebo. New treatment means that is the study medication that our company is preparing. Standard treatment means the product that is already there on the market. Placebo means dummy medication where there is no active pharmaceutical active ingredient. They are just other ingredients that are there in the formulation. So now, for example, if anyone is preparing medication for the GERD, then we can prepare we can the medication that the company is preparing will be like XYZ, and other medication that are there on the market like esomeprazole or any other rabiprazole or any medication that is pantoprazole or any other which is already there on the market, well established in the market, that can be the compared to other standard medication. Placebo means where there is no active ingredient like any other medication will not be there and it is just a dummy product. So like this, the treatment will be assigned to the 3 different groups and then we will check the efficacy by comparing all these 3 groups. So these type of trials are called it as controlled trials, where we are comparing the efficacy and safety of the treatment by using the other standards or the placebo treatments. And the next one is randomization. So randomized trials means mostly all the phase 2 trials on the phase 3 will be randomized only. That means where people are allocated at the random to the treatment groups in the trial, usually by using a computer program. That means in the controlled trials we have the new treatment, standard treatment, and the placebo. So now these 3 treatments, how the physician or the investigator or the company will decide like which patient should get the new treatment or which patients, which group of patients to get the comparative, the standard medication or the placebo. So here in the randomized trials, no one has that control and it is all done by the computer program. Once any patient is enrolled, once we enter the information in the computer program, it will decide randomly whether the patient should get the study medication or the standard or the placebo medication. That is how this randomization process works. If we give the control to the physician or the like company, then we know about the health condition of the patient. So based on that, the investigators can involve and or they can decide like whether they have to use a study medication or the standard medication. And if it is like this, then what are the results that we are getting, then we may not get the accurate ones, because all the patient population in the trial should be like equal. There should not be any discriminations like this, and we need to get accurate numbers for like if if the trial has designed like to get certain numbers, we need to have like 100 patients to take the new treatment, 50 patients to take the control treatment, 50 to take the placebo treatment. And if it is to the computer program, it will be assigned automatically. And if we give the control to the physicians, then we will not get to that numbers. Maybe all the patients will be receiving study medication, no one will be receiving placebo or the comparative medication. That is why so many of these trials are being randomized, and most of the phase 2 and phase 3 will be the randomized trials only. And the next one are the blinded trials. Blinded trials means here the patients or the the investigators or the sponsors, no one will aware like which treatment the patient is going to receive. And there are like multiple type of the trials are there here in the blinded. So how and why these blinded trials are done? Because we know like we don't know which patient is receiving treatment or the standard treatment or the placebo. And if we get the reports or if the patients know, then it will impact the adverse event reporting. And I mean like if any adverse event the study medication, then I would be interested to report. If I'm on the placebo, and if I have any adverse event, then I will feel like okay, it's okay, anyway I'm not on the study medication, no need to report. That kind of feeling the patients will get. So to avoid that and to get the standard reports, and to make the patients not too nervous or anxious, or to continue them with the study, because if they are not on the study medication, most of the patients will discontinue or withdraw from the trial, because anyway they're not receiving the treatment. So to avoid that, so most of the companies will plan for the blinded trials. So if any most of the trials, if you see, there will be like randomized, controlled, randomized, and the blinded trials. So these three will be there in most of the phase 2 and the phase 3 trials. So now coming to the blinded trials. So in the blinded, we have the multiple type of blinded trials. We have single blinded, we have double blinded, and then partial blinded, and the open label or unblinded. Single blinded means here the treating physician is aware, but only the patient is not aware which type of treatment he is receiving. Those type of trials, we call it as single blinded studies. And coming to the double blinded study, here the participants, physician, and the site staff and the study team, no one is aware of the treatment that the patient is receiving. Only the statistician or the computer, like which it has assigned the person, whoever has assigned the treatment, only they will know. Okay, and in case of partial blinded, so here in case like if there are multiple medications are there in that in the trial process, then maybe one product name will be revealed and one product name will be kept blinded. Those type of trials we call it as partial blinded. And in case of open label or unblinded, so here everyone will be aware like which type of treatment that the patient is receiving. So these are the different type of blinded trials that we have. Okay, so is this clear?
Yes.
Yes, Ramya.
Okay. So coming to the different stakeholders in the safety monitoring. So now we have seen the different type of the trials, and now who is responsible for the safety reporting and safety monitoring. I mean that is adverse event collection and reporting to the pharmacovigilance team. So first of all, investigator. The physician, whoever has enrolled the patient, is responsible to report all the serious adverse events immediately to the company. Okay, so here again they have to follow the protocol and investigator brochure, because sometimes in this protocol and investigator brochure they will specifically mention like which event has to be reported and which event should not be reported, and based on that they can report it to the sponsor, that is the company. So the sponsor, they will send it via email, or anyway they will know the contact details and how to report. The investigators, they will send the information to the company, and the company will send the information to the pharmacovigilance team. Okay, so that is how we get the information from the investigators. And they have to prepare the investigator report within the 24 hours. And recently I mean, still it is in the draft version. FDA has published one guidance to all the investigators whoever is participating in the clinical trials on which information and what is their responsibilities in the safety reporting in detailed manner. In summary, so these are the points that the document has, like the information has to be reported within 24 hours, and they have to include all the data elements, and then they need to perform the causality assessment. So it is again the company's responsibility. That means it is a responsibility of pharmacovigilance person, whoever is handling that case. They have to complete the case for the regulatory reporting. But whenever the investigator is submitting the report, they have to perform the seriousness assessment, the causality assessment. The initial assessment of the information has to be done by the investigator. And in case of any queries are there, then we can we can contact back the investigator, and we can wait till we get any query or any information is missing. And if you are unable to wait, then we can just follow. We can go ahead with the processing of the case. And in case of non-serious, usually for the clinical trial, non-serious cases will not be submitted to the regulatory authority, and the non-serious cases no need to be reported by the investigator as well. Anyway, but those information will be collected by the study team, and they will keep it in the in their clinical trial database, and they will send it to us in the form of line listing. Okay, so these line listing forms, once it is received by the pharmacovigilance team, we will create the cases separately, so that how anyway that information we will not report it to the regulatory authority, but still we have to create and keep the archive the cases in our database. And which information that the companies or the investigator have to report in their AE report. So first is the details of adverse event and the patient condition, like patient details, medical history, concomitant medications, or any risk factor like any alcohol use or nicotine use or any abuse of the medication, that informations. Therapy details like when the study was initiated, what is the dose, the duration of use, indication, temporal relationship, that is the action taken, de-challenge, re-challenge, how they have diagnosed, lab data, what is the outcome, any treatment was given, and any lab data. So these all information have to be included in that particular SAE report that we are receiving from the investigator. Okay, so I'll stop here because again the how we manage the blinded trials is again like one big topic that we have. I'll cover that in the tomorrow session. Okay, till here, does anyone have any questions in the clinical trial cases? Because these are clearly really important. And if you're working from US, majorly most of the pharma companies, they hire you for the processing of the clinical trial cases, so that you need to have more understanding on what are the different requirements that we have in the clinical trial process. Any questions? Anyone?
No, not for me.
No, Ramya, even not for me. No questions.
Yeah. Okay, then fine. Yeah, you can we can continue the remaining topic on the blinded trials in the next session. So that's it I have for today. If no further questions, then we can conclude our session for now. Okay, so in our yesterday's session, we have discussed about what can be the clinical trials, the different types of the trials, and the blinded trial information. So now, today we will discuss more about the blinded trials and some other conventions that we have in our clinical trial case processing. So, so how do we handle the safety reports from the blinded trial? Like I said, in the blinded trials, no one will ever like who will be receiving, whether the patient is receiving study medication or the standard medication or the placebo. Then for our case processing, we have to perform the labeling assessment, causality, and then we have to check the other factors. We have to write the narrative as well. In case of blinded trials, how do we update the information, or how do we submit the information to the regulatory authorities? So we have a guidance as per the GCP, and it is followed by FDA and all the other regulatory agencies, where that guidance clearly mentioned if there is any serious adverse event is reported in the trial, then it has to be assessed with the assumption that the subject is taking the test medication. That means we have to do our labeling assessment by using the reference document of the or the RSA document of the test medication or the study medication. And if the event is there in the test medication, then we have to check for the comparative medication as well. If the event is labeled in the comparative medication, then we don't need to break the blind. We can just retain the blinded information and then we can log the case. In case if the event is new for test medication, or if the event is new for the or it is new for the comparative medication, then again the expected then we have to break the blind, and then the case has to be submitted by breaking the blind. In simple, any report that is getting submitted to the any regulatory agency, the case or the information must be unblinded, and we have to send only unblinded information to the regulatory authorities. And in case if the event is happening with the placebo, then usually the placebo reports are no need to be submitted unless there are some concerns that there is some reaction to something in the placebo, maybe any allergic reaction or any contaminant that is caused by the manufacturing issue. Only in such a situation we will consider then only the requirement, then only the case needs the submission, or else we don't need to submit the information from the blinded trials if the patient is taking the placebo treatment. Okay, so in simple, this is the summary. So when we have to break the blind, if the event is unexpected for both the test drug and comparative, then we have to unblind, and then we have to report as a SUSAR to the with the test or the comparative medication, which are the medication the patient is on, and then we have to submit the information. And then if the event is expected for the test drug and it is not expected for the comparative drug, then we have to prepare a SUSAR report and submit to the regulatory authority with respect to the comparative product. And if it is unexpected for the test and expected for the comparative drug, then again we have to unblind the report and report it as SUSAR for the test medication. And if it is expected for both the test drug and comparative medication, then we no need to unblind the information, or the reporting is also not required to the regulatory authority. So this is a summary of the blinded trials, like which cases need the submission and which does not need the submission. Okay, is that clear? The blinded, like when we have to break the blind and then when we have to submit the information?
Yes.
Okay, so the next one is analysis of similar events, or SUA summary. So this is a major requirement from the US FDA we have for all the IND products. So whenever any company is preparing any IND safety report, that is, if you're crossing any ICSR report with the serious event, then we have to write this AOSC summary, which includes all the similar suspected adverse drug reaction. We have to analyze them and we have to give the information to the FDA. This analysis should include all the similar reports from all the INDs that are held by the sponsor and any other relevant information that is known to the sponsor. And here the sponsors have to evaluate the suspected adverse reaction in the context of other related reports or the adverse events, including that are occurred with the placebo and other comparative group, and also with the pre and post marketing studies as well. For example, if there is a case of myocardial infarction is reported for one of the study drug, then what we have to do is we have to go back and check in that particular trial how many of myocardial infarctions have been reported, and which event, or whether it is reported as myocardial infarction or heart attack or stroke or any other terminologies that are similar to the myocardial infarction. So we have to check all those terminologies, and then we have to check how we have assessed that cases, either as related or unrelated, and then we have to write the summary in our present case. So this is a requirement for all the serious events which we are submitting to the US FDA, especially for the clinical trial cases. So now this is the requirement, like which cases requires the AOSC. So any country of incidence which is the study is sponsored by the company, that is company sponsored interventional trial, and if the causality is suspected, that means if the event is related to the study medication, then yes, we have to submit this AOSC report, and irrespective of the causality we have to submit it. And in the if the country of incidence is USA, then if the type of clinical trial is investigator initiated. That means here the company is not responsible for the sponsorship. Investigator or the hospital is like they are sponsoring for to the patient by taking the medication from the company, and they initiate the trial. So here the company does not have any role in the clinical trial process, but for only for the safety reporting we need to follow the standard requirements. And for any investigator initiated trials for USA country of origin, we no need to prepare the AOSC reports. And for other countries, for investigator initiated, if both the investigator and the sponsor consider causality as suspected, then only we have to submit the report of AOSC, or else it is not required to be submitted. Even either of them will consider the causality as not suspected or not related, we no need to prepare this AOSC summary. So now let's look into the template for this AOSC. So we will include the case summary. So where we have to include all the reported AEs in detail, and then we have to write the summary of all the relevant clinical information. And we have to mention the previous reports, what is the case number, what is the event reported in that case, and when that report was submitted to FDA. And in that case, what was our assessment, whether we have considered the event as related or not related. And with this case experience, are there any change in the causality assessment, like with the new experiences, whether we are considering the event as related or not related. That information we have to write in this AOSC. So when I show you the Argus for the clinical trial case, then that time I will explain how to write this AOSC and then where to write this information. Okay, so this is about AOSC summary and also about the clinical trial cases. So these are the most important points we have to remember whenever we are crossing the clinical trial cases. Yeah, does anyone have any questions in the clinical trial reports? I'll just show you how we get the reports for the clinical trial cases. I mean, usually from the clinical trial cases, we get the standard SAE template. So we have the different type of SAE forms, but the information again will be like same, where it will include the case numbers, the protocol number, the study details, and when the event happened, and who is the patient, what are the events for the case, and then the category of the SAE intervention, that is treatment required or not, and the causality assessment, action taken, the challenge, re-challenge, all the medical history lab data, whether it is initial or the follow-up, and the date, this is the MRD date, and who is the principal investigator. Like this, in a structured manner, we get the reports from the clinical trials. And sometimes, yeah, they do attach the discharge summary reports if the patient is hospitalized for the adverse event. But usually, for the clinical trial cases, whatever the events that are considered as a serious event or the events by the investigator, we have to retain the same, and we should not select any events extra as per our analysis, especially for the clinical trial cases, because clinical trial team, they have their own database, and at the end of the trial they have to write a CSR reports, and they have to publish all the adverse events that are experienced in the clinical trials. And in the same way, we do prepare the DSUR reports, where it includes both the safety and efficacy profile of the medication. And at the end of the trial, I mean annually, we also submit these DSUR reports in the pharmacovigilance process. So what happens when we select extra events from the case or the discharge summary? There will be some discrepancy will be there. So that is why, to avoid that, anyway, there is one process which we call it as SA reconciliation process is there. Okay, and through that process, anyway, in between there will be some reconciliations will be done, whether we are entering all the information correctly or not. If there are any discrepancies before study closeout, only they will check, and then they will ask us to correct if the information what we have entered is incorrect. That process again I'll explain in detail at the end of the sessions about like SA reconciliation process. And then based on that we will correct it, and we try to maintain both the reports, the events that are reported in the both CSR and the DSUR are kind of equal, so that to avoid any discrepancies in the submission of regulatory reports. That is why one point that we have to remember in the clinical trial cases is whatever the events that are considered as SAEs by the investigator, Those are the ones we have to consider, and we should not select any other events extra as per our medical analysis of the case. It is different for the spontaneous cases, PMS, and literature cases, where we have to select the events and then we have to code them according to it. But for the clinical trial cases, we will be given with the case details, and then we just need to enter and select the correct coding option for that particular event. So what are these literature reports, and why these literature reports are to be, I mean, why the companies have to search the literatures? I mean, there'll be like thousands of the articles will be published on a monthly basis, and then it will be like very difficult for the companies to search. So how the companies will handle these thousands of articles, and then how do they identify whether there is any valid case reported in those articles, and then once it is identified, how the process will be. So before I go into the slides, I will just give you information on how industry works, especially for handling the literature reports. So the spontaneous reports, the post-marketing or the clinical trial reports, we know like these reports, we get it either from the patients or from the consumers. Clinical trial cases, we get the information from the investigator, but they directly report the information to the company. But for the literature articles, anyone can publish the article. So usually for our pharmacovigilance process, we refer to the medical and scientific journals, where they include the information about adverse event experiences that they report with the products. So that information they can publish in any of the journal, and then and then in our process we have some literature search team and literature processing team. The literature search team will search these thousands of the articles. So usually each and every company has their own data analytics tool, where it will automatically pull the articles which has the valid information about their company products, and then again the search team will review those cases, and they will identify whether there is any valid case in that, and if the valid case is identified, then only it will be sent to the case processing for the further analysis and assessment of the report. So now usually what will be the workflow and all. So yeah, if the like for the bigger companies like Novartis, Pfizer, or the GSK or Alcon, they have like very huge portfolio of the drugs. So and monthly on an average, like 20 to 30,000 of the articles they have to review, or maybe more than 50,000 of the articles is what they review on a monthly basis. Okay, so that is how they review, and it's like kind of automated tools are available right now in our industry. So this made our work like bit easier, where we already get the information which is filtered, and we only get lesser number of reports for our further analysis. So this is how we work for the literature cases. So now look into the guidelines, like what does the guideline states, like how about the literature reports. So the scientific and medical literatures are the significant source of information for monitoring of safety profile and the risk and benefit balance of the products, particularly in relation to the detection of any new safety signals or any emerging safety issues. So others, I mean, usually if there is any new experience about any event, that is what usually the authors will like to publish in the articles. If the event is already there in the labeling document, they will not submit the information, or they will not publish a report if the event is already known. If the event is unknown, that is when it will trigger the um, authors to publish the information. So that is why whatever the information we are getting from the literatures are the significant source for the new safety signal or the new risk associated with the medication. So here we refer only the literature report means any adverse drug reaction that is reported in the published abstracts or articles in the medical and scientific journals, and any unpublished manuscripts involving case reports, and then any important safety findings or clinical studies, including posters, letters to the editors, and associated communication from the scientific meetings. So these are the literature reports. So now let's look into what does the different regulatory authorities talk about the literature case processing. So worldwide literature monitoring is a regulatory requirement for all the companies, and it has to be performed for as long as the marketing authorization of the product is active, and whether the product is on the market or not, still we have to search for our products article. So whatever the products we are marketing, we have to perform the literature search for all those products. Here the goal is to identify individual case safety reports and any possible changes to the benefit and risk profile, particularly in relation to the detection of any new safety signals or emerging safety issues. So in the literatures, there will be two type of reports. One reports will talk about the safety of the medicine, that is any risk or any adverse event, and few types of the literature articles will publish about the efficacy information. Okay, that means they refer to multiple regimens or the multiple treatment options for one disease, and they will compare the efficacy and then they will publish that information. So this type of articles we collect it and then we use that information in our aggregate reports preparation. Okay, so that is how we use these literature articles in our pharmacovigilance process. And as you see, this is a requirement from all the regulatory authorities worldwide. So here in the recent times, the PV regulations, they have majorly focused increasingly on the medical literature monitoring, that is MLM process, which is a complex process with the scope continues to expand and the deepen. And the quality and efficacy of the literature monitoring process has significant impact on the literature search, because if our search strategy is not accurate or it is not meeting the quality standards, then there is a high risk of missing some articles which are talking about benefits of the product, or maybe we are missing an article which has new safety information. That is why the quality and efficacy of the literature monitoring process is really crucial, and we need to have very well trained professionals there. And then we need to have if you are using automated tools, then those tools must be validated well before they use in the production. So below safety information is what we collect from the literature report. So that is any new unexpected serious or non-serious ICSR reports with the causality and any pregnancy outcomes. It can be terminations as well with no adverse events, and or maybe any products that are used in pediatric population or any compassionate use program, or any lack of efficacy, any overdose abuse, misuse, any medication errors, and any non-clinical safety findings or animal study results. These all the information we collect it. So in this ICSR and pregnancy reports, and then lack of efficacy, overdose, and everything we will use for our ICSR case preparation. But the non-clinical safety results is the one we use it for our aggregate reports preparation. So the literatures are used for both ICSR and also for the aggregate reports preparation. So coming to the regulations that we have for the literature monitoring. So as per the EMA, the companies are required to monitor both the local and global medical and scientific publications in the countries wherever they have the marketing authorization, irrespective of commercial status. Currently, maybe it is not marketing, or like it is just registered but not yet marketing. Still, the company has to collect the reports about that particular product. And as per US FDA, all the reports of serious unexpected ADRs that are in the scientific literatures for the products with the same active moiety as the products marketed in the US must be submitted, irrespective of excipient different or the dosage form difference, or the strength or the route of administration or indications may vary. But still, if the company is marketing that particular generic form in the US, then they have to collect the literature, any adverse event that is reported in the literature article, and they have to submit it to the US FDA. And yeah, literature section in the benefit and evaluation report requires summary of all the safety findings. So for that, like I said, we will have to write a summary of these articles, and then we have to update them in the PBRER reports. So what does this MLM guidelines are? So recently in 2017, the EMA has published in detail guidelines on MLM process, like to avoid some confusions or to avoid some duplication of the work, what did this EMA does is they have, I mean, as per this MLM guidelines, the companies have to perform systematic literature review of widely used reference databases no less than frequently than once a week. That is, weekly we used to do the literature search unless active substances of their products are present in the list of publications monitored by the EMA. That means EMA has some list of products. They have like list of 400 active moieties where the EMA perform the literature search by their UDRA vigilance team. So if they identify any valid article, then they process the case, that they process that ICSR case in the UDRA vigilance database, and then they will share the information to the companies, whoever is manufacturing that particular active moiety. Okay, so that is what this MLM guidelines is. So in detail they have mentioned, and the date when the company has received it from the UDRA vigilance, that will be the day 0. That is when the clock will start. So now you must be wondering, anyway the company is doing the search and the EMA is doing the search, so there may be some duplicate reports. Yes, there will be duplicate reports. We have to perform the duplicate search thoroughly, and anyway for the literature cases, searching the duplicates is quite easier, where we can just enter the article name and the journal name. We will get the all the details or the cases that are published from that particular or reported from that particular article. So we can get the information easily, and then we have to check whether we have already entered the information or not. If we have already entered the information, we just need to update this EV number in the cases, and if it is not already there, then we have to create the case, and then we have to prepare the report and submit to other regulatory agencies, but not to the EMA. Okay, so this is a summary of all the EMA guidelines. So that is what it is mentioned, like all the SARs that is identified the EMA in the literature will be reported or not transmitted to the MAHs, but they will transmit it by the UDRA vigilance team, and the UDRA vigilance team will process the cases, and then they will send it to the MAH. They will download these ICSRs in our database, and then we do our assessment. And if we are agreeing to the UDRA vigilance team, then it is fine. If you are not agreeing to it, then we have to report them back. We have to contact them back with our assessment. And if it is a duplicate, we just need to enter the UDRA vigilance number in our case. Okay, so that is about the UDRA vigilance MLM guidelines. Now coming to the literature search, like how we do these searches. So usually the companies will perform the search based on the multiple reference databases in no less frequently than once a week. So what are these reference databases we are talking about? It's about PubMed or Google Scholar or MedSafe or Embase. So these are the ones which include all the articles, all the scientific or the medical journals will be published in those global databases. So that these are the ones we use for our literature search. And for the search, and MAH has to perform the search whenever they get this approval for the marketing authorization holder authorization, and then to identify any new safety signals or any new information that is being reported in the articles. So what are the search criteria that we use for the adverse event? So to search the adverse event, we not only use a terminology adverse event, along with that we also use the pregnancy, breastfeeding, overdose, misuse, drug abuse, pediatric, elderly, any occupational exposures, medication errors. These are the things also we use in our search option. And we also include off-label use and compassionate use options as well when we are doing the search. And coming to the products, what are the things that we use? So we usually search with the active name, active substance name, or the generic name, or any synonyms that we can use, or any salt forms that we have, all the things that we have to use. And we should never search with the brand name. It is always required to search with the generic name, because mostly the authors did not include the generic, they do not include the trade name in the articles. They will only publish the articles with the generic name. That is why we should always perform the search with the generic name. And if the manufacturer name is mentioned, then only that company will pick up the case. And if the manufacturer name is not mentioned, then all the companies, whoever is marketing that particular generic substance, then they have to create the case and submit the information to the regulatory authorities. Okay, so that is about the literature search criteria that we use for the medicinal products. And in case of class of drug effects, if there is a class effect and there is no specific event associated with the company medication, then yes, that case will not become valid, but we will use that information for our periodic reports preparation. Okay, so yeah, I'll just show you how we use the search criteria, the sample search criteria that we use for our literature search. So yes, so these are the terms that we usually use to search for the adverse drug reaction. So like I said, no, so it is always automated tools is what we use, where we just need to use some command. So all the cases with this criteria will automatically, all the articles with this criteria will be available to us. So for the adverse event, so these are the terms we use, like adverse drug reaction, drug overdose, or drug misuse, drug abuse, substance abuse, or pregnancy, drug efficacy, and drug withdrawal, drug tolerance, medication error, interaction, off-label use. So like this, these are the different terms we use. And coming to the products, so this is the synonyms. These are the synonyms that we have for the paracetamol. So paracetamol can be called in the different names in the different countries. So these are the different names that we have for the paracetamol, along with the different salt forms. So these lists will be maintained by the company. So you can see there are like hundreds of synonyms are there with for this paracetamol. So we use all these names for the search of any article that are published with this paracetamol. Okay, like this for not only hundreds, there are like thousands of synonyms are there, I guess, for the paracetamol. Okay, these are all for the paracetamol only. So yeah, I guess we almost have 10 pages for the paracetamol. And then we have for hydrochlorothiazide, so there are the multiple synonyms. Like this, company will maintain their own document on like which events, which are the products that they have, and their synonym names. Okay, so this is how the literature search process work. And once the articles are identified, then there will be a team who will be checking the validity of those articles, and then they will share those articles to the case processing team. Then from there, the normal case processing activities will start for the literature cases. Okay, so this is about literature cases. Does anyone have any questions here?
So Ramya, what is the timeline as soon as an adverse event is reported? What is the timeline? It begins like with day 0, right? The day it is reported. So, what is the timeline to submit to prepare all of this report and to submit it to the authorities?
Yes, yes. So I'll talk about the reporting timelines maybe in the next sessions. But yeah, so we have the different timelines for each of the report type. So depending on from whom we have received and to whom we are submitting, the timelines will vary. So again, I have in detail session planned for the reporting timelines and the submission.
Okay.
Okay. Okay, and the next, okay, any other questions? Before I move on to the next report types.
Not from me.
Okay, so the next type of reports are licensing partner reports. So licensing partner reports. So what are these licensing partners and why do we get the reports from other company? Okay, so any pharma company can get into a licensing agreement with a third party to produce or to market or market a drug formulation that the other company has developed. And each licensing partner does have the PV agreement to meet the safety requirements in the marketing country. So these two companies who have this marketing agreement, they will also have the pharmacovigilance agreement. That means agreement that is entered by the pharma companies to set for the protocols and procedures for the reporting of adverse events and also any aggregate reporting preparation in complying to the regulatory authority reports. So, for example, there are two companies like, for example, we will take Novartis and Roche. So, there is this product called Lucentis. It is used for macular degeneration. So, in US, this is marketed by the Roche, and in rest of the world, it is marketed by the Novartis. So, these two companies have some pharmacovigilance agreement, which states any events that are being reported in the US, the Roche will submit the reports to the US FDA, but not to any other country because Roche is not responsible for the marketing in other countries. So what does Roche do is when they are submitting reports to the regulatory authority, they also prepare and submit the report to the licensing partners. So that is how the Roche will share the information to the Novartis, and Novartis will pick up the information and we will process the case and we will submit to other regulatory agencies wherever we are marketing that particular product. So this is how the licensing partner cases work. So the information that we are receiving about the adverse event that we are receiving from the other company under this pharmacovigilance agreement, we process them as licensing partner reports. And the next type of reports are regulatory authority reports. So regulatory authority report means like how and why do we get the information from the regulatory authorities. So in the spontaneous reports you have seen if any report can be directly reported to the regulatory authority by the consumers or by the HCPs. In such situation, what does the regulatory authority do is they will create the case in their for example, US they have AERS database, and then European Medicine Agency they have ultravigilance database. Like this, they have their own databases where they can create the case, and once it is created, they will do their initial assessment and share the information to the company whoever is responsible for marketing of that particular product. So why do they share the information? Anyway, at the end, the information has to reach to the regulatory authority. But if the case is submitted to only one regulatory authority, but as per the regulatory requirement standards, the adverse event experience must be reported to all the regulatory authorities wherever we are marketing that particular medication. That is why, yes, we have to report all the information. Since we have to report all the information to other regulatory authorities, we have to pick the information from authority and then we have to process the information as it is received by the authority and submit to the other authorities. These type of adverse event reports where we are getting from the regulatory authorities are handled as regulatory authority reports. Okay, and we should not submit back the information to the FDA again, or maybe to the authority from whom we are receiving the information. Okay, so that is about regulatory authority reports and the licensing partner, literature, and the clinical trial and spontaneous. So does anyone have any questions in this, the multiple reports that we have discussed? We have still the post-marketing reports are there that we can discuss tomorrow. So till here, does anyone have any questions? So the next type of reports that we have are the post-marketing surveillance report. So you might have heard about there will be so many clinical trials and then there will be some post-marketing trials as well. So how do you think like these post-marketing trials are designed, and what is the requirement of these post-marketing trials and why the companies have to do this? That is what we are going to learn from today's session. So these post-marketing surveillance studies are the commitments that are required or agreed by the company that are conducted after the regulatory authorities has approved a product for marketing, and it is an important part of the science of the pharmacovigilance. So here it is a requirement from the regulatory authority, and it is not mandatory for all the products to have the post-marketing studies, and it is only required for the few products for which the company has agreed that they will be doing the post-marketing studies for certain products as per the regulatory requirements. So since most of these drugs or maybe the devices or vaccines, they are approved as per the clinical trials, which involves majorly the small number of people who have been selected for this purpose. That means we do not have a diverse population won't be there, and the few rare events we cannot identify from the clinical trials. Like this, we have certain limitations in the clinical trial. But once the drug is released into the market, there are no such limitations. That is why these post-marketing surveillance studies are required for few products where there is some risk associated and the risk is not yet identified in the clinical trials. So the goals of post-marketing surveillance studies are to detect any adverse events or the risk as they arise during the real-world usage of the device or the drug, that is when the drug is released into the market. If there are any adverse events, that is what we identify, and also we compare the efficacy of our drug with already existing products in the market or maybe with the standard products as well, and we update the clinical guidelines as certain population or the groups find more benefit than the others. That means we update like only whether if we have any if we have any new efficacy identified in the children population or in the elderly population, that information also we update for that particular product as per the post marketing surveillance, and we have to comply with the regulatory requirements. That is why we do these post marketing surveillance studies. So what are the different type of PMS reports that we have? So ICH E2E guidelines and GVP module 8, they both talk about these post-marketing surveillance reports, and we have different type of post-marketing surveillance reports are there. As per ICH E2E, we have passive surveillance, we have active surveillance, observational studies, and the targeted clinical investigation and descriptive studies. So these are the different type of pharmacovigilance methods are there where the companies can follow. Now let's see in detail what are these methods and which type of reports fall in each of the method. So the first one is passive surveillance. All the spontaneous reports fall in this passive surveillance. So that means any report that is received from a consumer or the HCP voluntarily who can share the information to the company or the regulatory authority or to the third party about the adverse event experience that they had after receiving the medication. So this is what the spontaneous reporting. Anyway, we have learned in detail about spontaneous reports in our earlier session. And the next type of reports which come under passive surveillance are stimulated reporting. That means here we have several methods are there where we encourage the healthcare professionals or the consumers to report the events about some new products or maybe for the limited time periods. So here these methods are included like online reporting of adverse event or maybe systematic stimulation of reporting some adverse event based on some pre-designed method, like maybe conducting some surveys or maybe giving some enrolling the patients in some patient oriented programs so that we will get to know or maybe informing patients or the physicians about some risk associated with the medicine so that if they show any symptoms they can report it to the company. So here the company is taking some effort to stimulate the reporting from the patients or the HCPs. These type of reports we call it as stimulated reporting, but for our case processing we process them as spontaneous reports only. And then we have this intensified reporting. So intensified reporting is kind of extension of spontaneous reporting program. Here we have major aim is to enhance ADR reporting of some specific medicines in early post-marketing phase. So this is also a type of stimulated reporting only where we have. But so this intensified reporting is required for some new drugs or any vaccines or any medicines that require some additional studies. For those products, we do this intensified reporting. For example, any antiretroviral medicines, we have to enroll the patients in some program so that we can, if they have experienced any events, we can directly get the information so that the reporters will know whom to contact if they have any adverse event. That is what intensified reporting is. And then we have some targeted reporting. So this is used to learn more about the ADR profile or some specific medication in the population. That means if any patients with HIV infection, if they are taking our company medication, then how our drug is working, what are the adverse event experienced in those population. So these type of reporting, we call it as targeted spontaneous reporting, where we target the patients with underlying condition of any immunosuppressing event or any HIV or any diabetes or any cardiovascular event. So these type of population we select, and then we see how our drug is affecting these type of population. Okay, so this is what, for example, monitoring of renal toxicities in with the use of tenofovir-based regimen in antiretroviral therapy. So like this, yeah, we see with patients with some underlying condition how they are reacting to our medication. That is what we observe in the targeted spontaneous reporting. So these are the 4 different type of passive surveillance methods where the companies can follow for their products. But for our case processing or for our pharmacovigilance process, these all 4 type of reports, we consider them as spontaneous reports only for the processing. And the next one are the active surveillance reports. Active surveillance means, in the contrast to passive surveillance, where the companies do not need to spend some money, or they no need to design some studies, or they need to have some pre-organized process in the passive surveillance. But for active surveillance, the companies need to design some study or the program, and they need to be some pre-designed or pre-organized process has to be there for the patients who are taking a part of these active surveillance studies. The design typically follows standard epidemiological study design. So that means where we use certain population, occurrence of the disease, these are information, we compare it with the exposure groups. So now let's see what are the different type of active surveillance studies are there. So we have sentinel sites. That means it involves collection of adverse event data from only one part of total population to learn something about the larger population. That means to observe the study of the trends in a disease, then we select the sentinel sites. For example, maybe any department of neurology, medicines, and neurology patients we can consider with some underlying condition of some any neurological problem. We just select few population, and then we see, and then we select some other population with no underlying condition, and then we compare the drug with these 2 population, and then we will see which population is having more efficacy or the more risk with the medication. These type of studies we call it as sentinel site studies. And then we have drug event monitoring. So here the patients will be identified from the electronic prescription data, or maybe from the medical insurance or the health insurance claims, where we can send the follow-up questionnaires depending on the adverse event experiences that the patient had. So in US, majorly everyone follow this electronic prescription. So if they have any adverse event, that information also will be there in the prescription data, or maybe it will be available in the health insurance claims. So that is where we will identify the adverse event information, and if we need further information, we can contact the physicians as per the requirement. So it's a kind of simple process we have about the drug event monitoring, where we can directly get the information about adverse events from these claims. And the most important one here is the registries. So we have some 2 different type of registries are there. Okay, we have disease specific registry and the drug specific registry, or we also have type of exposure during the specific life event, that is pregnancy exposure registry. Like this, we have different type of registries are there. So now coming to the disease registry. So in the disease registry, what does the what the information that we will be studying? So here in the disease registry, we will see with the treatment how they are going to, how the patients are able to recover, and without the treatment what is the recovery rate. That is what we will study here. And in the drug registry, we will study the difference with the difference of efficacy and safety with the similar type of medications. For example, if our company is giving manufacturing the product for drug A for some disease, and other company is making some other medication for the same disease, we call it as drug B. So now in this registry, they will divide the patients into certain groups, where one patient will receive our company medication and few other patients will receive some other medication. And then they will observe which patients are having higher efficacy and which patients are having more adverse events. and the information will be published, and also we will process the information about adverse events according to it if there are any adverse events are there in these registries. Okay, so these are the active surveillance studies. And then coming to the comparative observational studies, so here also we have some different type of observational studies are there, where we usually observe the incidences with one group and then the other group. So first one is the surveys, that is cross-sectional study, where we collect the data from certain population at a single point of time to regardless of exposure or the disease status constitute a cross-sectional study. Irrespective whether the patient have event or not, we just go to the group of patients, whoever is taking the medication, and then we conduct some survey to understand if any adverse events are being happening. And we will also check them what type of medications that the patient is taking apart from the our company medication. And the next one is case control study. So case control studies are similar to the cohort studies or the whatever we have in the registries, where we compare the studies within the two groups. But here, we will understand the case of disease that are being identified, and then we will decide the controls of the patients without the disease or event of interest, and then also we select the from the source population that give rise to the cases. That means, for example, with any medication, if there is some cardiovascular risk is being identified. So now let's say in our animal studies or in the preclinical studies, we have an event of hepatotoxicity has been identified. But in our clinical trials, this event was not identified. So this becomes the possible risk for us because the event is already identified in the animal models but not in the human patients. That means there is the risk of hepatotoxicity with the humans. So that is why we have to conduct some study to give a proof to the regulatory authority like this risk is minimal in the patients compared to the animal models. For that, the companies will design this case control study in the after post marketing, and then they will publish the results accordingly, where they will select certain group of patients with no underlying cause of any hepatic problem, and they will give this medication to them, and then they will observe how this medication is affecting that particular liver or if it is causing any liver disorders or not, that they will understand in this case control studies. Okay, so these studies are majorly useful when the goal is to investigate whether there is an association between the drug and specific rare adverse event, as well as to identify risk factors for the adverse events. That is when they will use these case control studies. So if you observe any clinical trials, mostly the clinical trials are based on these case control studies only. So whatever the risk that are being identified in the animal models, based on that, the trials will be designed, and even the post-marketing studies are also designed based on that. And then we have this cohort studies. Cohort means here we create, a group of people who share a common characteristic like exposure to the drug within the different time period. So this here we divide the population into exposure group, and the incidence is calculated and compared between the exposure groups. So here in the cohort studies are similar to the drug comparisons that we do, or how the clinical trials that we do, randomized clinical trials, where we divide the patients into comparative group, placebo group, and the study treatment. Here also we divide the groups into patients into two groups. One patient will receive our company medication, and other patients will receive other products that are there in the market, and then we will compare the efficacy and safety in these cohort studies. And the next one is targeted clinical investigation. So these are like majorly, and for our processing part, what are the events that are receiving from the active surveillance studies and from the comparative observation studies, we all handle them as post-marketing safety reports only. And the targeted clinical investigations are like when there is some risk identified from the animal models. So that is when these clinical trials are designed. So now you can understand how the trials are designed. So there will be like for one product, there will be different type of the trials will be there. So how they design their trials, it is based on the risk that is identified from the pre-animal models. So they conduct studies to understand pharmacokinetic and pharmacodynamic studies, which we call it as bioavailability and bioequivalent studies. And then genetic testing will be done, interaction studies will be there, and special population like pediatric pregnancy or elderly women that studies also will be there, and large simplified trials. I mean, that means there are pure clinical trials that can happen even after the product is being placed on the market. Still, there is a chance of conducting some clinical trials. Those studies we call it as large simplified trials. So these are some clinical investigations. Though whatever the information we are receiving from this interaction or the genetic testing or the pharmacokinetic and pharmacodynamic studies, we will not get any adverse event information from these studies. This information we will just add it in our aggregate reports, but very rarely we get any ICSR reports from these targeted clinical investigations. And the next type of studies are descriptive studies, which we also call it as epidemiological studies. So these descriptive studies have some important role in our pharmacovigilance process. Though from these studies, we do not have, or I mean, we do not actually enroll any patients, or we do not identify any adverse events through these descriptive studies. But what we understand is the data, whatever we have received till now, we will select it and then we analyze the information, like how our drug is working in the different populations. So we have different type of studies there here. Like first we will understand natural history of disease. So it is majorly focused on how this drug is affecting depending on the geographical region, like in Asian countries how this drug is showing efficacy and safety, and in other markets how it is showing efficacy and safety, and with the different age groups and different economical factors and different medical histories. So all the things that they will consider here. So pharmacoepidemiology is one of the important and also one of the interesting field that we have within the pharmacovigilance, where we analyze and study the data that whatever we have till now, and then we give the information like which patients are more having more risk associated with the medication, and which group of patients are less having less risk associated with this medication. So these we call it as drug utilization studies. They describe how a drug is marketed, prescribed, and used in the population, and how the factors influence the outcome, clinical, social, and economic outcomes, including any different population like elderly, children, the pregnant women, and some patients with hepatic and renal dysfunction. So these all information we collect it, and then we will check how our drug is affecting in all these patients. Okay, so these are the different methods we have, and like which method and when do we use these methods. So routine pharmacovigilance, that is the passive surveillance, is recommended for all the products. Whenever any product is being marketed, irrespective whether it is a new product or it is after the patent is gone, other products or other companies also will try to make the similar medication. So whether it is already the established product, still we need to have this routine pharmacovigilance process in place for all the medications. And coming to the enhanced and the focused pharmacovigilance, so it is required when there is any safety issue has been identified and which needs to be addressed. That means we are going to assess the causality here. I mean, through the individual case information, we are unable to assess. We need to check, we need to conduct some study to understand the relationship between the medication and event. That is when we do this focused pharmacovigilance studies. And there will be some active studies are also undertaken when the data is needed quickly, like some patient oriented programs, or maybe some marketing surveys or any marketing programs like this. There will be some quick studies also will be there, where when if we need the information in as soon as possible, that is when we conduct these active surveillance studies. And there will be some studies which can be demanded by the regulatory agencies depending on the as at a time of registration. They will ask they do ask to conduct some trials for few products. So now you might have got some understanding about the post authorization safety studies, so how and when they are initiated. So for all the products it is not required. That means whatever the events that are being identified in the animal the animal studies or maybe expected from that particular study medication. If all the events have been identified in the clinical trials, then we no need to initiate these post-marketing studies for those established medication if the medication is already there in the market with the different brand names. But if it is a completely new medication and the similar type of medications are not yet available in any market, that is when, irrespective of the identified risk, we need to conduct some post authorization studies, since there will be some risk associated with those medication. Or else for other medication, the normal spontaneous reporting is enough, and the normal spontaneous reporting and submission of ICSR reports to the regulatory authorities and preparing the aggregate reports, the normal process is enough for the other products. Those one we call it as routine pharmacovigilance process. And any studies that are required to assess the event and the product causality, then that we call it as the additional pharmacovigilance activities. And this is required, additional pharmacovigilance activities are required for all the new products, vaccines and the biologics and the devices. So, and if there is any event is being identified which is not for even for the well established product, where through the routine pharmacovigilance process, if any new event is being identified, then to assess that event, the companies will start new post-marketing study as per the risk minimization plan activity, so that they will understand the association between the event and the medicinal product. So this is how the post-marketing studies are designed by the companies. Okay, so does anyone have any question still here? Okay, so now let's see what does the GVP module 8 talk about this post-authorization safety studies. So as per EMA GVP module 8, so these past studies are any studies that are relating to an authorized medicinal product which are conducted with the aim of identifying, characterizing or quantifying a safety hazard and confirming the safety profile of the medicinal product, or of measuring the effectiveness of risk management measures. So these are the post authorization safety studies. So they have to identify, characterize and quantify the risk. That means if you see any labeling documents, you will understand like there will be some events which are characterized as rare, common, very common. Like this, you will see certain classification. That quantification is what we do from our post authorization safety studies. And also we confirm that safety profile of the product, and we also confirm measure the effectiveness. That means whenever any drug is being placed on the market, there is this RMP document that we prepare, which is risk minimization plan document, which include all the risks that have been identified in the clinical trials and the animal studies. And for all those risks, we provide some action plan to minimize or to prevent that risk. So during this post-authorization safety studies, we understand how effective these risk management measures are. How we will identify, we have different type of the studies, like we have drug utilization study, it is which majorly talks with the data. So those studies are the one we use to measure the effectiveness of these risk management measures. Anyway, I will explain in detail about how this risk management plan is prepared and how what information we include in the document in the later sessions. And there are different type of past studies are there. So here there are two types. One is post authorization efficacy studies, and in that again we have interventional and non-interventional. And then we have post authorization safety studies. In that also again we have interventional and non-interventional. So if interventional means they are the clinical trials, non-interventional means they are the post marketing trials. So when we consider any study as non-interventional, then the study meets few criteria. That is when we consider it as non-interventional study. So if any study has the medicinal product which is prescribed in the normal manner, that is as per the normal prescription guidelines, and there is no strategy, therapeutic strategy is designed for the patient. It is they just follow the prescription, and they assign the physicians will take the decision to include the patient in the study. And There are no additional diagnostic or the lab data are required for the patients to enroll or to take the medicines, and epidemiological methods are used for the analysis of the collected data. That is when we consider that particular studies are non-interventional studies. So the non-interventional studies and the post-marketing studies are kind of synonyms, but EMA uses the term of non-interventional studies and other regulatories use the term of post-authorization safety studies. Okay, so what does FDA talk about this post-authorization safety studies? So FDA also want the companies to have these post-authorization safety studies for several reasons. So the companies have to mention what are the studies that they are preparing as a part of their initial marketing authorization and during the post-authorization regulatory procedure, and also if there is any emergency safety concern is being identified, that is also when the companies have to have some post authorization safety study to be planned. And these studies are maybe by the different MAH in response to the safety issues or vary when the rare events are identified, because when there are any rare events are identified with some medication called like a, with atorvastatin there is some rare event is being identified, and the statins are also manufactured by different companies. So what companies can do is they can collaborate and they can see how rare this event is, because few events happen in 1 million or 1 in 10 million. Even one occurrence of such events will be considered as a serious event associated with that particular class of medication. So like these studies also can be planned. And the different methods, so it is same as per the GVP module and FDA. They have considered as like again same as per ICH, like we have active surveillance, observational clinical trials, and this drug utilization studies. Okay, so this is about the post-marketing study. So does anyone have any questions in this post-marketing studies? Okay, so that is it I have planned for today. Maybe tomorrow I'll take some extra time to explain other situations that we have in the PMS. Okay, so we'll continue from our previous session. So till last week, we have covered all the different type of the cases on how we receive the cases. So now, once we receive the information, the first step that we do in our process is like prioritization. That means we need to know which case has to be prioritized and which case will require the submission. Anyway, before processing the case or by just looking at the case, we may not be able to identify like which case requires a submission, but now it's better to understand the timelines before we start with the actual concepts and the requirements for the regulatory reports. Let me share my slides. Okay. Okay, so the submission of the single patient reports through the post-marketing report or the clinical trial or the spontaneous or the literature or the irrespective or different type of the case, we get some we get the report about a single patient or maybe some number of patients where we create separate cases. And in that cases, we no need to submit all the reports to the regulatory authority, and we only need to submit few cases. So which cases are requiring submission, that what it defines the expedited reporting. So which means an individual case safety report of one or more adverse events that must be submitted within a specific amount of time due to the severity of events is expedited reporting. That means anything that happened to the patient after receiving the medication, which we are entering in our ICSR report, must be submitted to regulatory authorities within certain timelines. So that process is what we call it as expedited reporting. So worldwide regulatory authority, they require expedited reporting of these ICSRs received by the drug companies, which meets specific criteria. That, like I said, we will not submit all the cases, and there are some criteria that are required for the submission. In that, we have validity, seriousness, expectedness, and the causality. So these four factors will impact our submission to the regulatory authorities. And the purpose of this expedited reporting is to make regulators, investigators, and other appropriate people aware of new important information on the serious reaction. So, how we will identify whether it is new or already known event, that again we will discuss in detail in our labeling assessment. So, this is a requirement of submission of ICSR reports to the regulatory authorities. So, this kind of reporting is like kind of standard, and the guidelines are taken from the ICH, but the timelines are same for FDA, EMA, and most of the regulatory authorities, except like few companies, few authorities like China FDA or the Saudi Arabian FDA. They have separate timelines, which again I'll discuss at the end of the session. So now let's see the standards for expedited reporting of clinical trial cases. So for the clinical trial cases, which cases are required for the submission? So we have to submit all the SUSAR reports, that is serious unexpected ADRs. That means all the SUSAR cases are required for the submission, irrespective of seriousness. SUSARs are serious, irrespective of the different type of seriousness. We have to submit all the SUSARs. And for an expected event, that means if the event is already known, then for those cases, if there is any increase in the rate of occurrence, which is judged to be clinically important, that is when we have to submit the expected events. That means the events which are already there in the IB document will not go for the submission, but we just capture them in our ICSR database so that we can use the information for our aggregate report preparation. And any event or any significant hazard to the patient population, like lack of efficacy with a medicinal product using in treated life-threatening disease. That means if anyone is using a medication, for example, if the patient is using atropine, which is actually used in the life-threatening condition, and if the drug is not working, that means that lack of efficacy is considered as reportable, or else the normal lack of efficacy for the medications which we use for non-life-threatening conditions will not be considered as expedited reporting cases. And any major safety findings that is being identified in the animal models like or any teratogenicity or mutagenicity, any of such events has to be reported to the regulatory authorities from the clinical trial cases. So these are the cases we submit. So now let's see how we will submit and when we have to submit these reports. So coming to the SUSAR with the seriousness of death and life-threatening must be submitted within 7 calendar days to the health authorities. And the submission is irrespective of country of incidence. For example, the clinical trials are ongoing in the US, in the Europe, and even in India. Then now event happened in India for the trial where this is ongoing in some other countries as well. So irrespective of the country of incidence, we not only submit to the Indian CDSCO, we also submit to the US FDA and EMA. So the information or this SUSAR report must reach all the regulatory authorities within the 7 calendar days it is. And apart from the other SUSARs, like apart from this death and life-threatening, the remaining hospitalization, congenital anomaly or other serious SUSARs must be submitted within 15 calendar days. And also any serious expected reports also must be submitted within 15 calendar days depending on the requirement. So these are the timelines we have for the clinical trial cases. And for the clinical trial cases, non-serious cases will not go for submission, but we will database them so that we can use that information for our aggregate report preparation. And if any event happened after the patient completed study, usually we will not collect the information, but if the patient is reported to the investigator, then investigator has to report it to the company. And those reports also we handle under either 7 calendar day or 15 calendar day timelines. That is the timelines we have for the clinical trial cases. So coming to the post-marketing products, like for the post-marketing products, which cases are required to be submitted? So first, all the serious unexpected adverse reactions. That means the serious and the events that are not there in any of the labeling document, and adverse drug reaction. That means where the causality is there between the medication and event. These events are required to be submitted, and the standards are same irrespective of country of incidence. All these serious, unexpected, serious adverse reactions should be submitted to all the regulatory authorities wherever we are marketing that particular medication. Okay, and cases of other adverse reaction which are serious and unexpected are required to be expedited. I will go for expedited reporting. And if there is an expected reaction, that means if the event is there in the labeling document, usually it does not go for expedited reporting, but again it depending on the different regulatory authority. So few authorities will need serious expected events, few authorities does not need the serious expected events. And then we have non-serious adverse reaction. Usually the non-serious events will not go for submission until and unless it is clearly mentioned by the regulatory authorities. So, and also apart from the single case reports, any safety information from other observations which could change the risk and benefit profile, that information also has to be informed to the regulatory authorities, but not by ICSR, but maybe in the maybe in any risk by updating the RMP document. Maybe there are some other ways where we can submit the information. That is how we send the information to the authorities. And any lack of efficacy will not go for submission until and unless it is for the life-threatening, if it is a medication which is used in the life-threatening disease. And any medication errors without any adverse reaction, we will just database them, but it will not go for submission, but we use that information for our aggregate report analysis. And coming to the timelines, so irrespective of seriousness, all the serious cases will be submitted to regulatory authorities within 15 calendar days. And the non-serious cases, it's usually not required as for the submission as per ICH guidelines, but the EMA needs non-serious cases to be submitted within 90 calendar days. And if you see the Saudi Arabian FDA, they need the non-serious cases to be submitted within 15 calendar days. And there are some some regulatory authority where they do want the non-serious cases of the domestic cases to be submitted within 15 calendar days. Domestic cases means, for example, if the country of incidence is US, then these reports have to be submitted to the FDA. Like this, country of incidence and the country-specific regulatory authority need the domestic cases as well. So now how we will remember all the, I mean, these timelines are kind of standard, but how do we remember the local timelines? Like we have like hundreds of regulatory authorities are there, then how do we remember? That's why I said we have one team which is local pharmacovigilance unit who will take care of these local submissions. So they will be well aware of the regulations from their own country. So whenever any case requires for submission, then they will email us to the global team, and where we just need to process the case on priority so that they can submit the reports. And if since you are from US, so like I said, for US, non-serious cases will not go for submission. Only the serious cases are required to be submitted. Okay, so these are a few scenarios. So does anyone have any questions in this both clinical trial case and the PMS cases, like which cases are going for submission?
No.
Okay, so now let's see how we submit the special situation reports, and then first we will understand what are the special situations, and then in the special situation, which cases are required to be submitted. So the special situations are, these are like kind of, these are not exactly the events or the adverse event, but because of these conditions, the patient might have some serious problem, or maybe they may experience some adverse event. So these kind of situations, which are actually not an adverse event, but they can be any medical condition, or they can be non-medical conditions as well. So I have listed down few examples like which require for the submission. So first, let's see about the pregnancy cases. So irrespective of the outcome of pregnancy, And if any female patient is taking the medication, and if the patient becomes pregnant, or any medication which is apart from any prenatal or the antenatal medications, if the patient is taking any medication during the pregnancy, they have to report the information to the manufacturer so that they can have a record. And if we know the outcome of pregnancy, like we know it's a miscarriage or abortion or any spontaneous abortions as well, along with the normal deliveries, even the outcome is normal, this information have to be reported. So why we have to collect this information? Because it gives us the information about the missing details. So in the clinical trials, we won't enroll these pregnancy patients or the breastfeeding mothers. So we will not have the safety of the medications in this population. So when we report the information, then only the company can update their efficacy profile. Like they can mention like this product was taken by the patient, the pregnant patients as well, but they have not experienced any event. So that means this product is safe for the both mother and baby. Like that, they can update their label, so it can be used in the wider population. That is the reason we have to collect the pregnancy cases as well. And for this pregnancy cases, which cases will go for submission? And all the pregnancy information that we are collecting will not go for submission until and unless there is any mutagenicity or the teratogenic event that happened to the baby, or any event that is experienced by the mother while taking the medication, or any abortion or the miscarriages. These all information is what will go for submission to the regulatory authority, or else only pregnancy without outcome unknown or with the normal outcome will not go for the submission. So the reports from the baby, we include all the congenital anomalies or any developmental delays. These all information has to be submitted to the regulatory authorities. That is fetal death or spontaneous abortion, or any suspected adverse reactions in the neonate which are classified as serious will go for submission. And then coming to the breastfeeding, so it is again same. If any event happened to the baby because of the medication which was transferred via breast milk, then that reports have to be submitted, or else we can still database them so that we can use this information in our aggregate report preparation. And that is the same situation with elderly or the pediatric population as well. So recently the pediatric PV is becoming they're providing more, we are giving more importance to this pediatric population pharmacovigilance. So any event that happened in this population, we have to collect it, and then we have to report it, and then we have to follow up with the reporters as well until and unless we get the complete information about the cases. And then coming to the medication errors. So medication error include all this drug overdose, abuse, off-label use, drug misuse, or any other occupational exposure, drug prescribing errors, dispensing errors, administration errors. Like this, all these terminologies come under this medication errors. So this refers to any unintentional error in the prescribing, dispensing, or administering of the medicinal product while in the control of HCP or with the consumer. So these all the reports, with or without adverse event, we have to create the case in our database. But for submission, if there is any serious adverse event associated with this condition, then only those reports will go for submission, or else these reports will not go for submission, but we just keep in our database for the PSUR report preparation. Okay, so that is about the medication error, and that is same with the lack of therapeutic effect as well. Usually, the lack of therapeutic effect cases does not go for submission until and unless the lack of therapeutic efficacy is reported for the medicines which are used in critical conditions, or for the treatment of life-threatening disease, or the vaccines lack of efficacy, or contraceptives. So only in these situations, only for these products, if lack of therapeutic efficacy is reported, they will go for submission, or else we no need to submit the lack of therapeutic efficacy cases to the regulatory authorities, and it will go for the 15-day timelines. And Whenever any lack of therapeutic effect is reported, there has to be some medical judgment. So where we have to decide whether it will go for submission or not. For example, if the patient is using antibiotic for a life-threatening situation, but this antibiotic is not for this indication, then if it is not working, then it will not go for submission because this antibiotic should not be used in this situation. So that is how we have to look into the scenario, whether the patient has taken the medication according to the labeling document or not. If it is outside label, then it will not go for submission. And all the other reports will be submitted within 15 calendar days, and the same with the lack of with the vaccines as well. Any vaccine lack of efficacy, I mean, if you are taking vaccine for any condition and if we still experience that infection, then we call it as lack of therapeutic efficacy of vaccines, and that cases must go for submission because it will impact the efficacy profile of the particular vaccine. Okay, so that is about the pharmacovigilance in the special situations, like which cases will go for submission in these reports. Yeah, Rajini, the first step that we have in our ICSR processing. Okay, so now we have the report in place. Now what you do first? So by just looking at the adverse event report, what we will do? There are multiple teams are there, and we have multiple steps as well in our ICSR report. So first we need to check our case validity. The first thing that we do whenever we receive the case is checking the validity, whether this case required to be database or not. So it is not for submission validity, it is for creating the case in our database. Okay, and then once we create the case, it will go for the data entry and then other duplicate check, and then we code the drugs by using multiple dictionaries, and then we code the adverse drug reactions by using Medra dictionary, and then we perform the seriousness, causality, and the labeling assessment. We write the narrative, and then if any information is missing, we go for the case follow-up. Okay, so these are the process we have. So before I go into in detail, like what are the conventions we have, I'll just create a case and then I'll show how we process the case in our database and how the workflow will be, so that you will have more understanding on the terminologies or what are the steps that I'm going to mention in the next sessions. Meanwhile, we will read our document first. I'll open the Okay, so let's read the document. Meanwhile, we'll understand the case. So this is a spontaneous report which I have taken the information which is published in the website, and the reporter is some physician, and the case is about tramadol related psychosis in a patient with a bipolar disorder. Okay, so that is the title we have, and this report is about a 59-year-old female who had underlying bipolar disorder and received the lithium treatment with a stable effective status. And then 1 month before hospitalization, she had been taking this tramadol and acetaminophen for the joint pain. Then she developed obvious prosecutory delusion. However, her clinical picture did not meet the criteria of any mood episode. And after treatment of risperidone in addition to the lithium, she was discharged without any psychotic symptoms. And she remained euthemic without any psychotic symptoms on monotherapy of lithium, 3 tablets once daily. Okay, so now this is the case we have. And like I said, the first step that we do is the checking case validity. So here from the checking validity means we have some 4 criteria. I mean, the case has to have reporter. So we have the physician, and the physician name is also available. And then we have the patient. So that is 59-year-old female patient. The patient information must be there. And then we need to have the suspect medication, that is tramadol and acetaminophen, and the event. the patient had this tramadol related psychosis, that is what they have mentioned, that is our event. So these are the 4 criteria that we follow. So now our case has all these criteria. The next thing that we do is we'll create the case. So before we create, we will understand what actually the reporter is trying to say. Okay, so what usually I do is, everyone has a different ways of processing the cases. Okay, so what I do is first only, since I know the database, I will divide my information also into the different sections that we will go. I use different colors. Let me just show you how I usually review the cases once I get, and then how I enter the information. So this information about the female patient who had this bipolar disorder will be my underlying condition, or it will go into my patient tab. So we have different tabs. So I'll just consider it as my this information. I use blue color. It will go into my patient tab. And then the patient received some treatment with a stable, she's receiving some treatment. Okay, this is also some underlying condition because it is not related to my event or the medication. So my suspect medication is tramadol and this acetaminophen. This will go into my products tab. So I'll just highlight with this green, and then this is some dilution happened, so it's my event. I'll highlight with some color. And minute new episode after treatment, the patient has received the treatment for the event, and then she is having some other conditions, rhythmic, and patient is being on the lithium as well. That will all go into my underlying condition. Okay, so now this is the information I have. So I have the diagnosis as well. I will just highlight into some color. So now I have decided my information, like what information I'm going to enter into my different fields. Now let me show you what informations we have in our Argus. Okay, so we create the cases by using, so you can see the multiple options here. So from the case actions, you can see open new and new case from image, or here also we have some shortcut where we can directly create the new case. You can just click and this new case page will pop up. So from this, we create our case, and here you can see some multiple fields. It includes general information, reporter information, patient information, and if it is literature, we have the literature reference. So, and you can see some flags as well. Flags means this information must be entered to create the case. So what I'll do is I'll just use the initial receipt date. Again, I'll explain in detail how I have considered these dates. Country, whatever the country where the event has happened, and the report type will be the spontaneous. And since it is a training version, we have not included much products, so I'll just product what are the drugs that are there in this dictionary. So this is the company dictionary. So when we join any company, what are the products we are marketing, that all the details will be there in this product browser. So for now, I'm just taking some random medication because my medication of tramadol is not available in my dictionary. Okay, and then the event. So yeah, for now I'll just enter psychosis because that is what it was mentioned, the tramadol induced psychosis. And then I'll do my search. Search means I'm searching for the duplicate cases. If I have any duplicate case, then I'll get the results here. So for the duplicate cases also, we have some criteria that I'll again explain later after I complete the validity assessment. Okay, so after the duplicate search, okay, so there is some case actually. So this is also created, for the training purpose. So the same case. So here you can see this is how you will identify the case here. If there are any duplicates, then this is how you are going to identify it. Okay, to confirm if it is a duplicate or not, we can just right click case summary you have to see, so that you will identify the drug and the events that are there with this in this present case. Okay, so for the training purpose, I'm just going to continue to create the case again. Okay, for now, I'll just consider the case as a medically significant, and I'm going to click on book in. Okay, my case is created. I'm going to open the case. Okay, so once you open the case, you can see multiple tabs here: the general, patient, products, events, analysis, activities, additional information, and the regulatory authorities. So majorly, we do our case data entry from the general, patient, products, events, and analysis. Activities, additional info, and regulatory are kind of, we don't enter much information there. Only from the first 5 tabs is where we enter the information. The general tab includes what type of report it is, when we have received the report, who is the reporter. That's what, if it is a clinical trial, what is the study name and study details. If it is a literature, what is the journal name, author name, the title of the articles. That is what it includes in the general tab. And the patient tab includes all the patient details like the patient name. If it is the mother and baby case, we will have the parent tab as well, where we enter the mother details. And then here the patient age, gender, and height and weight. And then ethnicity, occupation, and if it is a pregnant lady, then the date of LMP, the breastfeeding information. If the patient dies, then the death details, other relevant history, that is all the medication data, and the patient lab values. That is what it includes in the parent tab. Then coming to the products tab, here is where we select and code all the drugs that have been taken by the patient. And then we have events tab. So this is where we enter what actually happened to the patient and the events along with the outcome, seriousness, everything we assess here. And then we have one more tab here, that is event assessment. It is where we do our causality and labeling. And then in analysis is where we write our narrative. Okay, so now let's process this case. So here it's already entered the report type and country of instance, everything is entered. And the reporter type, we need to mention the details about the physician, HCPSS. So what are the fields I'm entering? These are the mandatory data elements which has to be entered for the submission. Okay, and then coming to my patient tab. So here I have to enter about 59 years old. Ethnicity, height and weight is not mentioned. The female patient. Okay, and she's having underlying condition of, so I'll just select this current condition because the patient is still having the bipolar disorder type 1. So this is how our Medra dictionary will open, and we just need to select the term. Again, I'll explain in detail how we select the terminologies by using Medra. Okay, and the patient is being taken lithium for that, so I'll just add that in my notes section because that's what the patient has received. And then don't have any lab data, so it is just clinical picture, but they are not mentioned what it is. And then coming to a suspect medication, that is tramadol with acetaminophen. So usually that product will be there in the dictionary if you are marketing that. And this is where we will enter our indication for the drug. Again, it will select and code with the Medra. And then here we have the dosage regimen, the patient, when he started the medication, what dose, everything. So here they have not mentioned the dose, route of administration. So I'm just considering it as unknown because it is a mandatory data element. And then here the action taken, de-challenge and re-challenge we have to enter. And yeah, here also they have not taken, it was not reported, the action taken. So I'm just keeping it as unknown, all the information. And since the patient received the treatment, the de-challenge and re-challenge will become not applicable. And then coming to my events, one is obvious, persecutory, delusion, that coding option we have. So I'm just selecting it. And then we have tramadol-related psychosis, and then the recent treatment was received with risperidone, and outcome is recovered. They have mentioned the patient is in stable condition with the treatment. Okay, so now coming to the seriousness. So we have to consider a few points here because the reporter did not mention about the seriousness of the event. So I need to check my IMA list. It's my list. And then we also have some other checklist which we need to prepare. Again, I'll explain in detail when I take you through the seriousness assessment session. So for now, I'm just opening the Medra. Okay, this is my Medra dictionary. I'll just search the psychosis event if it is there. Acute psychosis is there. Okay. Epileptic psychosis, Parkinson. There are different types of psychotics. The psychotic symptoms are there. So yeah, this is there in our list. So I'm just going to consider as the serious event because any drug related psychosis will be the serious event, and I consider the prosecuted delusion as a symptom, and then I'll just establish the relationship here. I mean, I'm just tagging it as a diagnosis. So I'll again explain in detail how I have done that. And the causality assessment was mentioned as certain that is related. And the labeling assessment also I'll review by checking the different labeling documents, and then I'll save my case. Meanwhile, I'll start working on my narrative. Okay, this is initial report. We have some standard format to write the narrative. So again, so do not worry, like I'm just explaining you how we usually do the case processing so that you will understand all the steps. And again, in the later session, I'm going to explain in detail like how we write the narratives or how do we do this assessment, everything in detail. Okay, so this is an initial spontaneous report received from my physician. State. First aid. Next we enter the patient details. 59-year-old female patient who experienced related psychosis. Okay, so that's my starting statement, and then we just need to write the medical histories. What are the underlying conditions that the patient had. Okay, and then I'll start my concomitant medications. Anyway, we have covered the information that came there. So I'll start what are the suspect medication that the patient is taking. Here I'll start, the patient started receiving from an unknown date, since the date is not mentioned, but they have mentioned 1 month before hospitalization. at an unknown dose. Okay, and then we will talk about the events. The patient experienced let me just copy paste here because so that I will not change any meaning of the information. I mean, with this we have covered all the details that we have to mention about the patient, and then action taken will be unknown here. Action taken means what happened to the treatment medication. Action taken was not reported. And the seriousness and causality we have to mention. The seriousness of the events was not reported but considered as serious, important event, and causality was reported as related to the medication. Okay, so this is how I write my narrative. So again, like I said, I'll explain in detail, but the flow of narrative is we have to start with what type of report is it, and from whom we have received, when we have received, and who is the patient, and what is the patient underlying conditions, other medications that the patient is taking, what are the suspect medications, and then event details in a flow, and then action taken, outcome, seriousness, and causality. So this is how I complete my case processing. Okay, so these are all the steps. Whatever I have mentioned till now are the responsibilities of drug safety associate. So now coming to the medical reviewer or the physicians, what is their role in this case processing? So whatever the information that is entered by the case processor, they have to cross check. They have to check whether we have entered or we have selected all the events correctly, or the event coding is done correctly or not, and then the event seriousness is done correctly or not, and the causality assessment. So here you can see at a case processor level, we will not perform the causality, but it is a job of medical reviewers. They have to perform it. So based on their assessment, they have just considered it as possible. Then they have to provide the justification why they are considering the causality as separate. So that is the job role they have, and then they have to check the narrative as well, whether it is done correctly or not. And if they still need any other information, then this is where they can raise the follow-up request to the reporter for all the missing information in the action items. And then in additional information tab, the source documents, whatever you will be seeing the AE reports will be attached in this notes and attachment section, where we can just attach, browse the document, and then we can just open and then attach. So this is how we attach the documents in our cases with a date, and then the classification will be like source document or something. So here it is like again drop down option. We'll just have source document. So here it is not available, but usually source document is the one we select. And the last tab, regulatory reports tab. So this is where the information will be submitted to the regulatory authority. So this, we do not have to do anything. Argus will take care automatically about the submission of report to the regulatory authority. And if any manual submissions are required, like I said, the local case receipt unit will take care of submission of this information to the regulatory authority. Okay, so this is how we do our case processing. And like I said, we will have multiple conventions, and the multiple guidelines are there for each and every step that we have in our ICSR. Okay, so yeah, does anyone have any questions still here?
Yeah, hi Ramya. I have one question. Is there any mandatory or any rule like we need to enter that physician, reporter name and patient name in narrative?
No, name should not be entered. So it's always because we follow the data privacy rules. where each and every country has their own data privacy rules. So we should never mention the names of reporter or the patient in our narrative. And we have some
Okay.
checkboxes as well, where we check the protect confidentiality. That means no one will come to know about the reported detail or the patient details.
Okay. Okay, Ramya. Thank you.
Okay, so any other questions?
No,
No.
Ramya.
Okay, so then first let's see. We were discussing about these 4 validity criteria. Now let's see what are those validity criteria, and then so that tomorrow we can have the other examples or like some kind of workshop on the validity. So the 4 validity criteria that we check in any of the document are the identifiable patient, identifiable reporter, suspect medication, adverse event, or the fatal outcome. So if the case has all these 4 information, then only we will consider the case as valid, and then only we create the case in our database. So if only these 4 details are there, we can just create the case. So the first one, identifiable patient. So which cases we consider as identifiable patient and which cases we will not consider. So at least if we have one single identifiable patient criteria like patient initials or the patient number or ID, date of birth, patient age or age group or the patient address, if any of the information, at least the patient ethnicity or any details if we have, we can consider the patient as identifiable. In case, so now we don't have any of the patient details, but still we can consider the report as valid if the report clearly defines that there is a patient who experienced event with the suspect medication. That means we do get multiple reports where we get like the physician will report, I have a patient who experienced some event after taking some medication. In that situation, we clearly know there is actually a patient exists who has some adverse event after taking the medication. Though we don't have all these characters, we can still consider that case as valid for identifiable patient. And then coming to the criteria, which we consider it as non-identifiable patient. So if any report that refers to a definite number of patients should not be regarded as a case until and unless we have the minimum 4 criteria for the case reporting. For example, if a report states like 2 patients are experienced or like few patients experienced and they are not mentioned the details, any details of the patient, like we need to have at least patient gender or ethnicity or any information should be there to consider the cases as valid. Now, if they mention we have 2 patients, one is female or one is male, then yeah, we create 2 separate cases for each and every patient. And if we receive any cases with the medical inquiries about AEs with no patient criteria, still we consider it as non-valid case under non-identifiable patient criteria. All the medical inquiries, like some patients before taking the medication only they will call and they will ask the labeling document state these events can happen after taking the medication, like whether can I take or not. Like this, some medical inquiry cases also we do get. So in that situation, that will be non-valid because the patient had not yet taken the medication. And the next one is the patient identifiable reporter. So under this criteria, we can consider the cases as valid if we have any information about the reporter, like any information like the qualification, either physician, pharmacist, dentist, or any patient name, initials, or address, or the consumer, or it can be any patient relatives or the friend or any patient spouse or any family members. That is also fine. We can consider it as a criteria. So whenever possible, we need to always get the patient contact details so that we can contact them back for any missing information. And whoever has provided the case information will be considered as identifiable, but not only the reporter, but one important thing that we have to consider here is the reporter must have the first-hand knowledge on the event experience. That means, so for example, I am reporting for a friend or for my friend's mother who I have never met, but I have got to know about adverse event experience from my friend. Then that report will not become valid because I do not know the patient. The reporter must know the patient, then only we consider the report as valid under this identifiable reporter criteria. And then come on to the suspect medication, suspect product. So this whenever we join any company, we will be trying on our products, like what are the products we are marketing. If you're not aware of it, then again we will be provided with some list of medications that we are marketing in the different markets. So whenever we get any cases, the first thing we have to check the marketing authorization status of the product of the reported suspect product in that particular country. If we are marketing, then only we consider the case as valid. If not, we will not consider the case as valid. And then when we are checking the product registration status, we should also check the marketing status, licensing partner agreements, or any co-promoted or means like I said, in few countries there may be some other companies who will be marketing our medications under some agreements. That information also we have to check. And if only generic name is mentioned without the manufacturer name, then if you are marketing that product in the country of incidence, then we have to consider it as a valid case. And also for the clinical trial cases, if the patient is enrolled in the trial and receiving the study medication or the blinded product, then also we will consider it as a valid case. And when we consider the cases as not valid for suspect medication, if any generic formulation is reported or where the manufacturer name is mentioned, and if there is no event reported to the company medication, then we'll consider the cases as non-valid. Because sometimes there will be many situations wherein we get some reports where the patients will be taking multiple medications and they clearly mention the event has happened with some medication. If you are not manufacturing it, then that means we'll make the case as invalid. Okay, so I'll stop here and then the next criteria we can discuss tomorrow about the adverse event, and then we can also discuss few more examples as well in the tomorrow session. Okay, so does anyone have any questions still here in today's topics at whatever we have covered?
No.
Okay, so now after this case receipt and triaging, so we have come to the case processing. So case processing is like a team where we work with all the independent cases or the individual cases that we are receiving from the patients from the multiple sources. So what are the activities that we do in the case processing? So in the case processing, we have two roles. One role is for the safety specialist or the safety scientist, and another one is for the physician. So these are the activities that are done by the safety specialist or the scientist. So the activities include, so if you remember the case processing that we have discussed in our earlier sessions, where we add the correct report type. So when we are getting any reports, we need to understand from where this information is received. So for that we have multiple options, whether we have it is a clinical trial case or the post marketing report or the literature or the licensing partner or the other company report, or it is a PMS that we have to identify. How we will identify that? We can look from the source document, we can observe, and then we will know what type of report it is. And that information has to be added because after the case processing, the case will directly go for submission. So we need to make sure whatever the information we are entering is submitting correctly to the regulatory authorities. And then the MRD, CRD and country of incidence, we have to confirm. So this information will be added if we are receiving the case from local safety unit. But if you are creating directly in our Argus at the global PV level, then we have to check whether we have added correct MRD, CRD or the country of incidence. Because this MRD defines the regulatory submission timelines and country of incidence defines like the report where it is going and getting submitted. For example, if the country of incidence is US, then it has to be submitted to US FDA. along with all the other markets depending on the seriousness of the event. But if the country of incidence is like Pakistan or any other Arab countries, they only need the domestic cases to be submitted them. They do not want any other cases apart from the reports that are happening in their country. Like this, the the submission of the cases will it will impact the country of incidence has major impact on the submission of the report. So we need to choose the country of incidence correctly. So for that, we have to check the report thoroughly. So we need to understand where actually the event happened. Usually, we will directly get where the event happened, but there will be one situation. If the patient is traveling and, for example, the patient is from US, but event happened when he went for a vacation in Canada or maybe when he went to vacation in UK, then country of incidence will be UK, but the reporter's country will be US. So that is how in that situations only where we may get confused, or else usually country of incidence will be like we can easily understand from the document. And for the clinical trial cases, we have to enter correct study ID details. And then if it is the follow-up case, we have to assess whether the follow-up is significant or non-significant. So that information, how we will define. So that is completely on the judgment base. If any information that we are receiving in follow-up is having, because of the follow-up information, if you are updating any E2B field in our Argus. So we have E2B data elements that I'll again explain in detail when I show you the case processing, what are the E2B data elements. If any information is adding in the E2B data element, then that case we consider as the significant follow-up. If not, the follow-up will become non-significant. So how and why we have to consider the follow-up as significant or non-significant? Because it defines the submission of the report. If we consider follow-up as significant, then only the report will be submitted, or else non-significant follow-ups will not be submitted to the regulatory authorities. And then we have multiple data entry fields. In the general tab, report information or the patient details, lab data, medical histories, and suspect medications we have to code by using drug dictionary, and concomitant medications we have to code by using WHO drug dictionary, and we have to perform the seriousness assessment, labeling assessment, causality assessment, and then the case process has to write the narrative. So these are the activities that we have at the case processing level. And then we have the physician role. The physician, whatever the information that is entered by the case processor has to be confirmed by the physician. That means technically they do not have much work in the case processing role. In the ICSR processing, they just need to confirm whatever the data that is entered by the processor is correct or not. That means whether we have selected events correctly or not, event coding is correct or not as per the Medra convention, seriousness assessment is correct or not, labeling, causality, de-challenge and re-challenge. These are the factors that will be assessed. So at the case processing level, the processor no need to check the causality. He just need to copy paste whatever the information that is reported in the source document, especially for the causality assessment. But it is the physician responsibility to assess the causality by considering all the points. That again, I'll explain in detail what are the points we have to consider when we are doing this causality assessment. Okay, so these are the activities that we do with all the reports that we are receiving through our ICSR reports. Okay, so any questions in the roles or the activities that we are doing?
Ramya, I have one question. So
Yes.
all these activities comes under like case
Drug safety
processing is
associate
nothing
role.
but drug safety associate role, right?
Correct.
So drug safety associate and medical
Reviewer.
services associate.
Same, medical service specialist, medical service.
Okay, same.
Physician. Yeah, we have physician, drug safety physician and drug safety specialist will be there. And drug safety analyst also we can call, or the drug safety scientist. So like this, we have multiple names for the job description that I have mentioned in the earlier slide.
Okay, Ramya.
Okay, so all the the coding of the drugs and all, or the data entry, that anyway you will understand with the practice, with the argus, like how we enter the information. Okay, and the major conventions is what I'm going to discuss. Rest all data entry fields, we can see when I show you the multiple case examples, then you can understand how we enter the information. So now I want to majorly concentrate on the core areas of the or the key processes that we have in our case processing. So in that first one is Medra, that is event selection and code. To select events correctly at the ICSR level, because it defines the submission of report, and also it defines the signal management, and also aggregate report preparation, and all the risk and benefit profile of the medication is completely depending on how we are selecting the events, and then how we are submitting or the coding the events by using the Medra dictionary. So that is what we are going to learn in this Medra session. So let me just give you a sample scenario how it will impact, or the how the companies will have to follow this Medra guidelines. So there was a situation maybe in 2012, between 2012 to 2015, I guess, with the company called Roche. So what they did is, there were like so many reports have been reported for their products. For example, it was reported as aplastic anemia, but the company has coded the event as anemia, not as event. They have coded it as unspecified anemia. So this, in this example, aplastic anemia is a kind of life-threatening condition, and even one occurrence of aplastic anemia, the drug will be removed from the market. That much significant is that event is. So to avoid that, the companies have coded it as unspecified anemia, and they have considered the event as non-serious. With that, that report never submitted to the regulatory authority. So no one came to know like this event is happening with this product. Okay, and also there will be some other situations. So there are like wantedly the companies do the cases, do the assessment like this, and there are a few cases, a few situation where it was reported as suicide. Okay, so usually if it is a suicide, then we have to perform the follow up to understand whether it is a completed suicide or attempted suicide. Completed suicide will fall in the category of fatal, that is the death as a seriousness factor there. And if it is attempted suicide, then it is a kind of non-serious event, or it is normal medically significant event. So if we don't do the follow-up, and if we just consider the suicide as attempted suicide, then entire risk and benefit profile of the medication will change. That's how important the Medra, understanding the Medra concepts is. And majorly, if you are applying for the drug safety physician role, then it is best to have the certified Medra coder certificate. Okay, so it's a test that is done by the Medra itself, the Medra organization, which is a part of ICH team. So maybe if you're interested, you can enroll into that course, and it's just a, for exam only you need to pay around $200 or something. That's what, when I have given the test, I have paid around that much only. So, and they give us some scenarios where we have to select the events, and then we have to provide the event coding options for those events. That is how the test is. So for the drug safety physician role, or maybe if you want to grow further in the company under as a safety physician, it's a best to have the certification with the Medra coding. So that's advice I can provide, but it's upon your interest whether to take the test or not. But in the test, what are the questions that they will ask, how the test will be, how situation they will discuss, that's all anyway I'll be discussing in our session. But if you want to have some certificate from the ICH team, then you can attend that test offline.
So is there a
Okay.
website where we can apply for the examination?
Yes, CMC link I'll share you in the chat window by the end of the session.
Okay.
Yeah.
Yeah, that will be good. Thank you, Ramya.
Okay. So now let's start with the Medra. So what is this Medra is, and how the events are categorized. We know there will be like thousands of medical conditions are there. How this Medra is designed, how we select the events from the Medra. Let's see how it is. Okay. So this was developed under the auspices of ICH team, that is International Council for Harmonization of Technical Requirements for Pharmaceuticals for the Human Use. They have developed this Medra dictionary. Why did they develop? Because to harmonize the event coding irrespective of the different regulatory authorities. So for example, let me just give a simple example of nausea. Nausea can be reported as like felt like throwing up, feeling nauseous, or the felt like vomiting, or like the stomach is churning and I'm feeling like, so like this, there may be different type of verbatims one can report for the nausea. So to harmonize or like to make all these terminologies into one standard term, they have prepared this Medra browser. Okay, so that we know if the so though it can be reported in a different ways by the reporter, but when we are submitting the information to the regulatory authorities or whenever we are preparing our analysis reports, we can directly retrieve the cases with the term called nausea. So all the synonym terms or the events can pull in that analysis report. That's how important Medra is, because we are harmonizing the event coding irrespective of the different verbatims that we have. And also we can have the multiple languages. Sometimes the regulatory authorities need the reports to be submitted in their own language, and for us to code the event separately again in their own language, it's kind of difficult. So with this Medra, we are converting the terminologies into some code, and when through the electronic exchange, if you are submitting information to the regulatory authority, it will automatically translate into their own language with that particular code. Okay, so that's how important the Medra is. And almost who maintains this Medra? So it is maintained by it is actually developed by the ICH, but it is maintained by the MSSO, which is Medra Maintenance and Supporting Services Organization, that is MSSO. And it includes majorly the three areas of ICH, that is US, European Union, and the Japan. These three regions provide the guidelines on how to maintain and which terminologies to be added. But we have some observatory committees as well, like we have UK MHRA, Health Canada, and WHO, and also we have International Pharmaceutical Association that is also a part of this as an observer, where if they have any comments on the Medra, that they can also provide the guidance. And almost 5,200 organizations are using this Medra in almost 115 countries. So you can see anywhere, whatever the country you join, this is the dictionary you are going to use for the event selection and event coding. So this is the full form of Medra, that is Medical Dictionary for Regulatory Activities. And the main purpose, like I said, it facilitates the clinical exchange of information to the standardization. So we are standardizing all the different terminologies by using the Medra, and it is important tool for the product evaluation, monitoring, communication, and electronic record exchange, and also on the oversight. So here, like I said, whenever we are pulling the reports, we pull it by depending on the events, and then we will understand what is the risk associated or what is the benefits associated, what are the events associated with the medication. This all information we can identify only through the Medra coding only. And it supports coding, retrieval, and analysis of clinical information about human medicinal products, including pharmaceuticals, biologics, vaccines, and other drug device combination products. Any medical conditions can be coded by using this Medra. And this is the definition of Medra, which is a clinically validated international medical terminology. So it's a validated one. It is used by the regulatory authorities and the regulated biopharma industry. So all the regulatory authorities are using the Medra dictionary for the event selection and the coding. And the terminology is used through the entire regulatory process, from the pre-marketing to the post-marketing, for the data entry, retrieval, evaluation, and the presentation. So this is not only used in our normal ICSR processing, but even in the pre-marketing, whenever any side effects or any event that is being identified in the animal models, that is where also we use this Medra dictionary. And this is where we use the Medra right from the preclinical to the post-marketing. And it is used in all the regulatory authority and industry database. That means in our Argus, we have the Medra dictionary enabled. If you can remember our yesterday's class or the day before yesterday's session on the case creation, where I have coded all the events by using the Medra dictionary, that means Medra is already enabled in our in our safety database. And there is some offline Medra dictionary as well. So for that, you need to have license to have the offline version. But I have some free version available. So for your practice, you can use either this free website, or maybe you can use the Medra that is available in the Argus. And for all the ICSR reports and to write the safety summaries, we have to use this Medra dictionary. and to write the clinical study reports. That is one of the annual report or the end of the study report we publish for any clinical trials. And we have investigator brochures. This is a kind of protocol for the clinical trials. And the labeling documents like core safety information or SMPC or USPA or any patient packaging insert. These all documents will have the adverse event terms from the Medra dictionary only. And when the company is submitting any marketing applications or advertising publication or any prescribing information, so in all wherever we are talking about safety of the medication, then where we have to use only Medra terminologies for the discussion part. And then coming to the scope of Medra. So scope is, it is majorly used to code the medical conditions, okay, whatever adverse events that happen to the patient after receiving the medication. Indications means for which the patient is taking medication. For example, if the patient is having hypertension, then he can take Diavon. So this hypertension also has to be coded by using Medra dictionary. And then we have all the investigations, like all the lab data we can code, and surgeries and the medical procedures, medical, social, and family histories. That means medical history. If the patient is having some underlying condition of any chest cardiac arrest or any hypercholesterolemia, any condition can be coded by using Medra. And social history, like tobacco user or any alcohol use, these conditions also we can code. Family history, that means family history of cancer or the family history of diabetes. So any of the family member have any history, that coding also we can code by using Medra. All the medication errors, product quality issues, device related issues, product use issues, pharmacogenetic and toxicological and standardized queries, all these terminologies we can code by using Medra. That you can in detail you can see when I show you the Medra, you can understand what are the different terminologies that we have. And coming to the out of scope, that means which things we will not code by using Medra. So it is not a frequency qualifier. That means by just looking at the Medra, we cannot understand anything. It is just a random the event name that is about Medra is. And the numerical value, so with the Medra, once we select and code, it will get converted into one number. That number does not define anything. So you might get confused with the medical coding and Medra coding. So these two are completely different. Medical coding, with the medical code, we can understand what actually the patient has experienced, because the medical codes are designed like that. But Medra codes are not like that. They are just random numbers which are assigned sequentially. So we cannot understand anything with these Medra codes. And it can also not describe the severity, and it is not an equipment but device or the diagnostic product dictionary. That means we cannot code any products by using Medra. And the clinical style study design terms, the patient demographics also we cannot code by using Medra. For example, in our ICSR, if we have received any case, then all this highlighted information we select and code by using Medra. Now let's see the sample case. So it is about a 75-year-old male patient receiving some drug for rheumatoid arthritis, that is indication, developed symptomatic aortic valve stenosis, that is the event, and medical history included colon cancer and cigarette smoking, and he underwent some surgery, that is aortic valve replacement, and then he developed some sternal wound infection 3 days post surgery. So in this narrative, the indication of the drug, the event, medical history, social history, surgery, and the event. So these all factors we have to code by using Medra. Okay, so whenever we receive any case, everyone will have their own style of working on the cases. But my way of working is always I will highlight like which will go into which tab of Argus, so that I cannot miss any information. So in that time only I will just highlight what are the terminologies that I have to code by using Medra, so that I cannot miss any I cannot miss any events that were not selected by using Medra. And this is the Medra structure. So we have 5 hierarchy levels in the Medra. So first one is lowest level term, and then we have PT, HLT, HLGT, and SOC, that is preferred term, high level group term, and the system organ class. So the lowest level term include almost 90,000 plus terminologies are there. Okay, So let me just, in the meanwhile, let me just open my Argus while I explain the case. Okay. Okay, so the lowest level term include whatever the terminology that is reported by the reporter. Like I said in the same example, the patient may report nausea as feeling nauseous or felt like throwing up or anything. So these are all kind of synonym terms or the similar terms will be available in this lowest level term. So whenever we are coding in our database, we have to choose the LLT term that is matching to the reported event term. For example, if the patient has mentioned, I am not feeling well and I'm having some I'm feeling like nausea. So this is what mentioned. Then what we have to search in our Medra is in the LLT term we have to mention the patient is feeling unwell. So we have to search for the feeling unwell and then feeling nausea. So these are the kind of terminologies we search in the LLT. And if there is any direct coding option, we can directly code it. And once we select this LLT term, so all these LLT terms, what are the synonyms we have, these all grouped into one standard medical term. Like I said in the example, under nausea, we can see the multiple examples of we can see the multiple LLT terms for this nausea in the LLT term. So now all these PT terms are categorized into all these PT terms are further grouped into one single medical concept. That is what the HLT includes, the high level term, and all the high level terms are further grouped based on the anatomical specification and etiological specification. They are grouped into some HLGT terms, and all these HLGT terms are categorized into 27 system organ classes. System organ classes are the respiratory system or the nervous system or the cardiac disorder. So like this, depending on the anatomical origin or the pathophysiology or the physiological origin, all these 90,000 LLT terms are grouped into these 27 system organ classes. Okay, so that is how our Medra is designed. So let me just show you this sample. So here you can see this is the LLT term, that is bruise. And for the bruise, which is categorized into single medical concept of contusion, and it is going into skin injuries. But coming to the HLGT, it is categorized as epidermal and dermal condition, and the body site and etiology, it will go into skin and subcutaneous tissue disorder. So like this, there will be SOCs assigned for each and every LLT. Okay, now let me just see another example from our Argus. Okay, so this is where we code our Medra. So now let me just open the Medra dictionary. So this is how our safety database will include our Medra dictionary. So now here you can see the multiple levels LLT, PT, HLT, and HLGT and SOC. So whatever the event that we are searching, we will search in the LLT term. So but now let's just see what are the different LLT options that we have for the nausea. So like I said, it has like stomach churning, feeling queasy, nausea, nausea aggravated, nausea post chemotherapy, nauseated, nauseous, postprandial nausea, or like queasy. So like this, the reporter can report in any way. So if the reporter has mentioned I'm feeling queasy, then we have to select the LLT of feeling queasy. Okay, and then if we just select, then this is how we will do our event coding, where we have selected the LLT term.
Ramya, have you shared the screen for Argus? Because I cannot see it.
Okay.
Okay. Thank you.
Yeah. Okay, so here you can see, so once you select the feeling queasy, then yeah, you can see the PT term of nausea. And if I just search in the PT term of nausea, so this is how we get the different LLT terms. Okay, so whenever we are seeing any, if we see any case, so for example, in this example only, let me just present you. So here we have seen, so this is the verbatim where the patient is taking this drug for rheumatoid arthritis and developed symptomatic aortic valve stenosis. We have to search this verbatim under this LLT section. Okay, if it is not available, we will do some aortic valve stenosis. Okay, and then the rest all PT, HLT, HLGT, SOC will be automatically aligned because they are already assigned in the Medra dictionary. Our job is to select the correct LLT term which is matching as equal to the reported verbatim. Okay, and this is how the Medra is designed. So this aortic valve stenosis will go into the SOC of cardiac disorder. So we mainly focus on the PT and the LLT and SOC. HLGT and HLT we rarely use, especially not in the, I mean, we never use them in our ICSR processing, but we may use them in the preparation of some tables or analyzing some conditions in the signal analysis or maybe in the aggregate report preparation, but not in the ICSR process. Okay, so it is how the Medra works, and this is the structure of the Medra. So you can see another examples as well. And
So Ramya, I have one question.
yes,
How does this classification like actually help, like the lowest level term or the preferred term and the other higher level groups? Like when we select only the lowest level or we put in the preferred term, right? So
yes,
the other three are automatically generated. So in
correct.
the case report, do all the 5 classifications are submitted, or is it only the lowest or the preferred term that we are selecting?
Yeah, it is always when we are submitting the report, only PT term will get submitted to the regulatory authorities. So how
Okay.
and why this classification is required? Because at the end, whenever we are preparing the summary of the reports, that is
Okay.
when this classification helps us. Okay,
Okay.
because we cannot include all, I mean, whenever we are categorizing any events, we have to categorize them by using this SOC terms only. Let me just show you the sample because anyway you have asked the question so that you will be clear like how this information will represent in our reports.
Okay.
Let me just give me a moment. I'll open the document. Okay. So here you can see whenever we are preparing a reports, this is how we have to prepare the reports, where we have to include what is the SOC, what is the PT term, how many patients have experienced, and what the patient is on which therapy. That is how we prepare the summary reports in our PSUR preparation or in the DSR preparation. Or for
Okay.
any report generation, this is how we include the information. Sometimes we do create the reports for the HLGT and HLT terms as well because
Okay.
it gives us the more details like where and which system is getting more affected because of the medication.
Okay.
Okay, that is
So
the
here they are
importance of
mentioning
categorizing.
the number of patient experience, like each category, each classification.
So
Okay.
that we will
Okay.
get
So
to
that
know at
makes.
the end, yes, which system the patient is having major impact because of the medication.
Got it. So, like to understand it, um, uh, you gave an example about aplastic anemia, that the company had not mentioned that it was aplastic anemia and they just wrote anemia. So, in that scenario, had Medra dictionary been there, then they would have this categorization and they would have to mention which number of patient had like that particular aplastic anemia. Is that how it would help in that scenario?
No, Medra training will help, not the Medra dictionary itself. Because
Yeah.
with the Medra dictionary, it is always the person whoever is selecting the LLT term that is what it defines. Okay, so I mean the example that I gave you about plastic anemia, they have wantedly selected as unspecified anemia because unspecified anemia is a non-serious condition because
Okay.
it does not even submit to the case because if they submit a plastic anemia, like I said, even one
They would have
experience
to
of a
pull that
plastic anemia
back.
from the product
Yeah.
from the market. That's what
Yeah. So
maybe
they did
that's like
not
frequently
select
will
it. And,
happen. Yes, it's like frequently, I mean, not with the terminology, but with for
okay.
some events, they do ask because in the ICSR, though we do the assessment and we select the correct events, but once it go to the safety management team, sometimes to avoid these situations, they do ask us to
Oh,
downgrade the event selection. Like if it
oh,
is reported as some
got it.
nephritis or like a kidney injury or something, then they will ask us to
Uh-huh.
put it as kidney disorder. Kidney disorder is like it can be any kind of thing,
Anything. Yeah.
and yeah, it will be some kind of non-serious event as well. But kidney injury or like any nephritis will be the serious
Right.
conditions. So that
Right.
is how it
Right.
works. So sometimes it is
Okay.
because of our unawareness of the type of event options that are available in the LLT or in the Medra. Sometimes it may be because of wantedly we are downgrading the reports. There are two type of
Okay.
errors that can happen in the industry.
So it
That is
still
why
can happen, right?
yes,
If
with the
someone
Medra
chooses to.
training. If
Yeah.
they choose to downgrade, then we cannot avoid the situation. But if you by
Right.
mistakenly do, then it will come
Mm-hmm.
back very strong on us because by mistakenly we should never do. But if you're
Never
doing on
do.
wantedly, then there may be some justification can be provided by the person whoever has done that,
Mm-hmm.
depending on the product conventions. But by mistakenly, if you do it, we have to do the corrections. We have to provide the root cause for not doing it correctly. We have to provide how corrective action and preventive action. It's a kind of big headache thing if we do any
Right.
incorrect event codings.
Okay. So that's where Medra training comes into play, right?
Yeah, I mean, in
If
our
we,
organizations, they do provide the Medra training, but they provide
okay.
only the basic training on the Medra, like how to use and all, but they will not explain with the multiple case scenarios on how and exactly how we have to select the event codings.
Okay. Okay.
Because even this training I have designed for my employees, so I usually
Mm.
give trainings to the new joiners. So
Okay.
whenever they come here, I'll just majorly concentrate on these areas because this is where they do the errors.
Their mistakes. Yeah. Right.
Yes. Okay.
Okay. Thank you.
Ramya,
Yes. Mhm.
um, if you don't mind, can you please show the Medra coding once again? Tab
Yeah,
in order
sure.
safety.
Yep, I'll show anyway. We have so many examples as well, even in the next sessions. Yeah. So in
No,
the LLT term,
what my doubt is,
hmm
for example, now you coded as aortic valve stenosis, right?
hmm yes,
So each and every, I mean, each and every drug and each and every event, so we need to code right under LLT only.
correct. In LLT only we have to select the event coding. Each, like I said, now in LLT has almost 90,000 plus terminologies. I mean, I don't think more than 90,000 terminologies are there in, I mean, not more than that medical conditions are there. If there are any new conditions, like for example, all the COVID terminologies are like new, right? So the
Mm-hmm.
Medra in the version, they have updated all the COVID terminologies as well. Since it is the older version, maybe you will not see in the training version, whatever we are using Medra, that is the older version. But in the current Medra version, you can see those terminologies. Let me just show you as well the offline
But
version. Yes,
Ramya, but how can we come to know that we selected term is correct or not?
that's what we have to check. So for now, in this example only, let's see aortic valve stenosis or maybe some infection. I'll select. Okay, so now you can see this chest infection can go into the respiratory or the infection or the infestation. So by looking into the SOC, HLGT and HLT, we will know whether we have selected the correct coding or not. And for that,
Okay.
first, if you're not aware about the disease, I mean, we will not remember all those different medical terminologies. I will, if whenever I see any condition, but not like fever or the nausea, normal conditions. But if there is any some names like Steven Johnson syndrome or like some Wickenberg type AV block, like this, if these kind of terminologies comes to me, first what I'll do is I will just understand the basic understanding of the diagnosis. Okay, what is this diagnosis is? First, I'll try to understand and I'll see like which system it is going to affect. If I know that basic knowledge, then when I select my event coding, then I'll go and see my HLT, HLGT, and SOC. Then I'll see, okay, so now my event is coded correctly because this is where this infection or this particular event is can happen as per my search that I have done in the medical websites.
Okay. Okay. And suppose in the same example, if it is not the correct one, it's not under infection, the yellow one. If it is the other one, so can we
Yeah, we
change
can
that?
choose. Yeah, that is what it is. We call it as secondary SOCs. If we have some criteria that we have to follow as well, that again I'll explain how
Okay.
we select the SOC terms.
Okay.
We just need to highlight. So yellow ones are the primary SOCs. What are the other one is the secondary SOC. That I'll select. I'll tell you how we select the secondary SOCs.
Okay.
Ramya, for example,
Okay.
if you enter like pyrexia, that is different word of fever, right? In LLT, so it will pop up as in SOC, HLT, and HLD like fever or only
Let
pyrexia.
me show. No, it will go into general disorder if my guess is correct. Okay, under pyrexia, we have different type of fever is there. Pyrexia is the PT term, that
Okay.
is a standard term for the different type of the fever that we have. We have high temperature, intermittent
Okay.
fever, periodic fever, febrile reaction, or the fibricular, the drug fever, PU. I don't know what is this PU is. Okay, and then we have pyrexia of unknown origin, slight fever, slight temperature, spiking temperature. So for all these LLT terms, the standard code is the pyrexia, which will go into the body temperature conditions, and it is a general disorder. So any general
Okay.
disorders, if it, I mean, like fever, it's a general disorder. We cannot particularly assign to the some origin or
Organ
the some
system.
organ. Yeah, so that is
Yeah.
why it will go into general disorder. Okay, so now I'll move back to the slide so that we can, once we have examples, because this is, like I said, it's a bigger session. We have almost 3 sessions only for the Medra. Okay, so now at the end of the session, I have planned almost like 30 to 40 examples I have placed so that you will have more understanding on how we select and code the events. Okay, so yeah, first we'll understand the basic concepts and then we'll go into the more example scenarios. So now this is how the event is getting converted into some code. Even in the example you can see. So here you can see the terminology coding. So like I said, it's just a random number. If any next event is added, then it will become 5 and 2, 5 and 3. So that is how the event, the codes are defined in the Medra. So irrespective of the different languages, whenever we're submitting, it will submit by using this standard code.
Okay.
And these are the different system organ classes we have. So we have the blood and lymphatic system disorder. Let me just show you in the meanwhile. Okay, now I'll show under blood and lymphatic system disorder what can be the different. Okay, so this is the SOC. In the SOC, you can see we have anemias, and then we have some coagulation issues. We have hematological, and then homeopathic and hemoglobinopathies, thalassemia disorder, thalassemia. So like this, we have multiple terminologies under blood and lymphatic system disorder. We have so many cardiac disorders are there, and then congenital familial and genetic disorder for any baby who is exposed to the medication during pregnancy, and if they have any condition, then we have to select them by using the congenital terms. So now
Yeah.
let's look into how these congenital terms are. Let me just open. Okay, so here you can see in the cardiac lymphasis or some. So like this, we will have some congenital terminologies. In case any abnormalities or congenital disorders. So in the congenital disorders, you can see the terminologies like congenital anomaly, congenital absence, or the congenital anomalies, or we have congenital cyst, and then congenital or the conjoined twin. So like this, you will find so many terminologies with this congenital, and whenever anything that happened to the baby because of drug exposure in utero, it has to go to the SOC of congenital. That is the one thing that we have to keep in mind whenever we are selecting the LLT term for the baby cases. And then we have for ear and labyrinth and endocrine disorders, eye disorders, gastrointestinal and general disorders. For any events which cannot be categorized into any of the system, they all will be there in the general disorder and administration site condition, like any drug administration, injection site pain, or any injection site rash, or any injection site reactions, or any, if it is a patch, then topical patch reactions, or any subcutaneous or the skin reactions. These all fall under this administration site reactions. And we have hepatobiliary, immune system, infections and infestations. We have all the infections are categorized into one SOC of infections, injury, poisoning, and procedural complications, any injuries that are there, and all the investigations, all the lab data will be there in the investigation. Investigation include the normal lab value, high value, and abnormal value. Let me just show you that is for example, let's just see in the blood pressure. In the blood pressure, you can see blood pressure, the only blood pressure is there, and then we have blood pressure abnormal is there, and then blood pressure decreased is there, blood pressure diastolic abnormal or the blood pressure high is there, and then blood pressure increased is there. So like this, we will have the multiple LLT terms which goes into the SOC. So whenever for example, if a case has mentioned the patient has BP of 180 by 150 and they have not provided the diagnosis of hypertension, if the diagnosis is provided, then we will not select the lab data as an event. If the diagnosis is not provided, we have to consider the lab data as event, where we consider the event verbatim as blood pressure increased, not as hypertension. So that means any abnormal lab data are there without the diagnosis, it has to go to the SOC of investigation. That is one rule that we have to remember. If the diagnosis is mentioned, it can go to any of the system organ class where the depending on the site where that event can happen. And we have all the metabolism disorder, musculoskeletal, all the cancers are in one SOC, nervous systems, and all the pregnancy conditions are there in one group. And any product related issues like product quality issues, the tablet is breaking, or the capsule is like the capsule is not done, or like the granules in the capsule are like leaking, or the some needle issues, or the some foreign particles in the injection. So any kind of product issues, you can find them in this product issue SOC. On the psychiatric disorder, renal and urinary system, reproductive system disorder, or the respiratory, skin and subcutaneous, social circumstances. I will explain that later. And then we have surgical and medical procedure and the vascular disorders as well. So like this, we have so many SOC terms are there. So coming to the social circumstances. Now let's understand what can be there in the social circumstances. So this is the offline Medra version I'm using. Okay, so under the social circumstances, you will find multiple HLGCs and HLT, which are social circumstances are not the medical conditions, but there can be any underlying conditions which can affect the patient's mental and the physical health. In that, we have some age-related factors, economic factors, and environmental issues, family issues, gender-related, legal issues, and the lifestyle issues. In the lifestyle issues, we can see any alcohol use or the dietary or issues, disability issues, tobacco use. And the legal issues, we have any diverse issue or the crime victims, or in the or is in abusing or homicide, or is in the jail or imprisonment. So like this, we have so many conditions are there in the social circumstances. Usually, selecting an event under social circumstances, it's like very rare to happen. But whenever if you are handling any psychiatric drugs, then these events are more frequently you will see these terminologies. Okay, so these are the different SOC terms we have. So now let's see what is this multi-axial terminology. That means in the examples where you have asked, we have some primary SOC and secondary SOC. How and why these multi-axial terminologies are there in the Medra. So here it is a representation of the medical concept in the multiple SOCs is what we call it as the multi-axial terminologies. Because of this multi-axial terminology, it allows grouping by the different classification, and it will help us to retrieval and presentation by the different data sets. Because majorly all the PTs are assigned to the primary SOC, and which PT it has to, which SOC it has to go, it depending on the Medra is already assigned the primary SOC. But if we want to change, usually we should not change the predefined allocations. But if we want to change, we have to follow some we have to follow some hierarchy level whenever we are changing it. Okay, so now let's see how which hierarchy level we have to follow. So first we have to give priority to the congenital, familial, and genetic disorder, or all the neoplasm conditions or the infections. If these three are there, then the first priority should go to the congenital, and if congenital is not there, then neoplasm, or if it is not there, then it will go for the infection and infestation. Now let's see the example. Okay, so yeah. So these social circumstances, medical procedure, and investigation, for these three, there will not be secondary SOC, but for all the other SOC, for all the other medical conditions, you will find the secondary SOCs, but not for these three SOCs. Okay, so here you can see the example. For the congenital HIV infection, can have multiple SOC terms, and as per our hierarchy level, first we need to give our priority to the congenital terminologies, and if it is not there, then we have to give priority to the neoplasm. If the neoplasm is also not there, then we have to give the priority to the infection and infestation. But since the primary SOC is already allocated as a congenital, we no need to change it. Okay, so whenever we are considering the secondary SOC, I mean, it is very rare we choose the secondary SOCs, but sometimes if we are in that situation, then we have to take opinion from the physician, and then we have to take the confirmation from the particular product lead for changing the SOC, and then we have to update the case according to it. Okay, is this clear about multi-axial terminology allocation? Okay, so the coming to the Medra maintenance. So now if there is any terminology that we have received, if the terminology is not there in the Medra, then how the Medra will change, or how the companies will go ahead for updating the Medra. So usually this MSSO team will accept the change request. Any organization can send 100 change requests per month. For simple changes at the PT and LLT level, they can change within 7 to 10 working days. But for the complex changes, that is, if they have to update HLDT, HLT, and SOC, then they are done for the every year twice. So first in the March and another one in the September. So currently we are at 24.1 version of the Medra that is released in the last September. So that is how the Medra is maintained. If there is any new terminologies, every year they do keep on adding these terminologies. I remember when I joined the PV, I like we have around 50 to 60,000 of Medra terms, but currently, like I said, we are almost at 90,000 Medra terminologies. So that is how they do keep adding, and they do delete some terminologies as well. They combine those terminologies with some other terminologies. So they do a lot of changes every year. Okay, so now let's see what is this auto encoding is. So whenever we are selecting any event, sometimes Argus will automatically select, or our database will automatically select depending on the information that is available in the LLT term. But it is always incorrect. So whatever, if it is the direct hit, then only that event coding will be correct, or else we have to double check whether the event is coded correctly or not. Because allergic to CAT scan can be auto encoded as allergic to the cats, which is incorrect. And myocardial infarction in the fall of 2000 can be selected as myocardial infarction and fall. So here fall is not an event, it is just a season. So that factors also we have to consider. When the coding is done, we have to double check whether the event is coded correctly or not. Okay, and these are the important coding errors where the Medra has stressed, like this has to be mentioned or given training to the all the person whoever is handling the patient information. One is missed concept and another one is soft coding. Missed concept means missing to select the serious term that is reported in the case. For example, whatever the event that happened to the patient after receiving the medication, we have to select and code them. So here, if you see example, these are like kind of live examples or the situations that are happened where these examples are provided to us by the Medra team. They mentioned one manufacturer has not selected all the events. The information received is the patient took some drug X and developed alopecia, increased LFT and pancreatitis. But they have selected only alopecia and increased LFTs, which are non-serious events, but pancreatitis is completely missed to code. That means it is a serious event. Okay, they have completely missed to code this event. And another example where the patient took some drug and developed interstitial nephritis, which later deteriorated into renal failure, but they have only coded interstitial nephritis and completely missed the concept of renal failure. So this is about the missed concept. Coming to the soft coding means selecting the term which is less specific and less severe than the reported term. That is what we call it as soft coding. The example, aplastic anemia coded as unspecified anemia, or liver failure coded as hepatotoxicity or increased LFTs, rash, which is diagnosed as Stephen-Johnson syndrome, but they have only selected rash as event, but the diagnosis is not coded. So aplastic anemia, SJS. So SJS is also the Stephen-Johnson syndrome, also a kind of situation where even one case of SJS, the product will be off the market, or maybe there has to be so many measures have to be taken while dispensing or while using the medication. Okay, so that is about introduction to the Medra. So maybe in the next session, we will discuss about how do we code with the Medra and how do we select the events. Okay, so does anyone have any questions in today's topics that we have discussed?
No, not for now.
Okay, yeah, anyway, we still have so many examples. Maybe you can, if you have any questions in the example, then we can have the discussion later.
Yes, sure.
Okay, so we will start with the coding of the Medra. So we have so many conventions which we need to remember when we are coding with the Medra. So these are, we call it as PTC points, which is term selection points. And these are in the Medra guidance, they have provided guidance for each and every scenario, which I'll explain in brief. So the first one is diagnosis and provisional diagnosis with or without sign and symptom. So for this, what we have to follow. So what is definitive diagnosis? Definitive diagnosis means we have the symptoms which are associated with the diagnosis. And in the report, they have clearly mentioned these symptoms are diagnosed as this condition. That term we call it as definitive diagnosis. Provisional diagnosis means if our report mentions it as a possible diagnosis, or it can be, or it might be such event where the reporter has not yet confirmed the diagnosis, then those reactions we call it as provisional diagnosis event. So in case only diagnosis is mentioned without any signs or the symptoms, then yes, we have only one possible option to select the diagnosis in our Medra coding. That means if it is reported as myocardial infarction, then we have to select and code it as myocardial infarction. And if it is reported as possible myocardial infarction, then also we have to select the myocardial infarction term, which is a definitive diagnosis term, because in our Medra, we don't have the provisional diagnosis terms available. Okay, now if there is any signs and symptoms are reported, okay, then we have to, like for example, anaphylaxis with rash and dyspnea, hypotension and laryngospasm is reported, then we can select only diagnosis, or we can also select the symptoms as well in the case, and then we tag them to the diagnosis. Okay, in the previous example, if you remember where I have tagged the symptom into the diagnosis, again I will explain how we do this tagging in our database. And in case of provisional diagnosis, though signs and symptoms are mentioned, all the events have to be selected as diagnosis only, because the was not confirmed in this example. So for example, if it is reported as possible myocardial infarction with chest pain, dyspnea and diaphoresis, then we have to select the terminology separately, and then we have to code and tag all of them as diagnosis. So in our database, in the events tab, we have two options, so the two radio buttons, one for the diagnosis and one for the symptoms. So we need to choose these radio buttons whenever we are selecting the symptoms for the definitive diagnosis, and if you are having the symptoms for the provisional diagnosis, then we have to choose the diagnosis radio button. Okay, so this is the convention we have to follow for the diagnosis and provisional diagnosis. And the next one is death and other patient outcomes. So meanwhile, let me just open my database as well so that I can show the different terminologies that are under these term selection points. Okay, so now if we see death and other death and other patient outcomes, so let's see. So we will have, with the death there will be so many events, like death of the relative will be there, and the normal death from the natural cause, or like unintended death or the sudden death. Like this you will see the lot of LLT terms. So let me just see in the Argus only. Okay, so now you can see with the death, we can see multiple death LLTs, like accidental death, we have apparent death or agonal death struggle, or we have some cell death, cord death, the crypt death. So like this, we have multiple LLT terms are there. So not only death, you can find hospitalization term, like only hospitalization is
I
available. Sorry, you're
okay.
not able to see
I
the
could not
screen, I
see
guess.
the screen. Yes, yes.
Yes. And then we have the disability. The disability also, we have so many LLT terms like this. For each and every seriousness outcome, we have the LLT term option available. Okay, so this is how we have the multiple outcomes are there in our LLT terms. So now let's see the next term selection point about suicide and self-harm. So here we need to follow a few conventions. If it is only reported as suicide, then we have to choose the term of only suicide. And if they clearly mention it is a completed suicide or attempted suicide, or like it is a para suicide or the suicide attempt or the suicidal ideation. So we have suicidal ideation as also one of the event term. So however it is reported, that is how we have to choose the LLT terms, because depending on the LLT that we are selecting, our seriousness will be changing. So we need to choose correctly when we are choosing the LLT term for the suicide. So here you can see there are multiple LLT terms options are there. And then we have some self-harm events as well. So if you see the self-harm, so like we have thoughts of self-harm is there, and then we have some events of deliberate self-injury, and then impaired self-care or intentional self-injury, or like self-hatred. And like this, we have multiple terminology options are there under this self-injury and the self-harm and some self-hatred. Like this, we have the multiple terms are there. And the next one is when we have conflicting, ambiguous, or vague information, then how do we go ahead? So let me just show you when we have conflicting or ambiguous information, how we select the terminologies. So first, whenever we have conflicting or ambiguous information, we need to follow up with the reporter and we have to get the confirmation. And if you're not able to wait, or the case has to be submitted on priority, then we have to take some actions. For example, if it is reported as hyperkalemia with serum potassium of 1.6 milliequivalent per liter, so here the diagnosis is hyperkalemia, but the event is actually the lab value is lower than the normal, that is serum potassium is low. So here we have some conflicting information. In this situation, we can select the terminology as serum potassium abnormal, and then we can query to understand what actually happened to the patient, whether it is increased or decreased term. And then next, if it is mentioned as GU pain, we don't know whether it is genitourinary pain or the gastric ulcer pain. So, but here we know it's actually pain. So we can select the LLD term of pain, and then we can ask what is the abbreviation for this GU in the present example. Okay, that is how we have to follow if we have the conflicting or ambiguous information. That is same for vague information as well. For example, if it is reported as turned green, So we don't know what turned green is, that whether it is a patient nail or the body part turned green, or the patient tablet capsule or the injection color has changed. We don't know what actually it is. This also, since it is a vague terminology, we can consider it as unevaluable event, and then we have to query back to the reporter to understand what actually they are trying to imply. And if they mention the patient had medical problem of unclear type, for these type of terminology, we use the standard LLT term of ill-defined disorder, because we don't know. We know that the patient had some problem, but we don't know what problem it is. So in this situation, we select the LLT of ill-defined disorder. Okay, so this is how we follow up with the vague informations. Now let's go back to the term selection points. Okay, so now coming to the combination term. So when we have the combination terms, what are the rules we have to follow? Let's look into the examples first for the combination terms. So in the combination term, when one condition is more specific than the other condition, then we have to choose the most specific term. For example, if it is reported as arrhythmia due to atrial fibrillation, we can select it as atrial fibrillation, and yes, arrhythmia can be selected as a symptom for atrial fibrillation. And hepatic function disorder, and they are clearly specified that disorder, hepatic function disorder is a kind of vague term. It can be any disorder, but here they are clearly mentioned it is acute hepatitis. So we have to choose the LLT term of acute hepatitis. And if the combination term is available, then we have to always choose the combination term. That, for example, retinopathy due to diabetes can be diabetic retinopathy. Rash with itching, we have the LLT option of itchy rash. Like this, we can select the combination term. In case if we don't have, if our situation does not fall in any of this scenario, then we have to see whether we can split the terminologies. Like diarrhea and vomiting can be split into two terms, and wrist fracture due to fall can be split into wrist fracture and fall as the two separate LLTs. So these 3 points we have to consider whenever we have the combination term. First, we should always search whether we have combination term option is available or not, and then we have to see the specificity of the event, and we have to check whether we can split the events and code the events separately without changing the meaning of the case scenario. Okay, so that is how we follow for the when we have the combination terms.
So
Okay, and
Ramya,
then
in
next.
case of, yeah, I have one question. In case of combination term, is there an option available in the Argus or the Medra that we can select more than one term? Because in LLT, in all those, yeah, when you're selecting up, I see you're selecting only one term. You're writing just one term.
Yeah, we will
So
select only one term, but if you put the
one at
percentage
a time.
search, yeah, you can get all the terminology that is available with the retinopathy. So the same, we have HIV
So,
retinopathy, diabetic retinopathy is there, hypertension retinopathy is there. And so for each and every condition, there is retinopathy associated. So this is how we get the combination term. Yes, we do have the
okay.
combination term options available
So
in the
like if
LLT.
there is vomiting and diarrhea, so how do we do both of them?
Yeah,
So
for
we just
that,
write vomiting and diarrhea?
yes. So first we'll search with the vomiting. If we can get any, we can either search with the vomiting or we can search with the diarrhea with the percentage search. Then we can see,
Percentage.
okay, the percentage search we put, and then we will get all the reports, all the results from this particular vomiting. So we have different vomiting. So yeah. Okay, so we have we have to check whether we have diarrhea and vomiting in
Diarrhea
one.
also.
Yeah.
Okay.
If it is not there, then we'll split the terminology.
Then how do we do that?
So yeah, in the same example, if it is reported as diarrhea and vomiting, then we select one LLT as diarrhea, and then we will just select the term, and then we select one more new event, and then we select it as a vomiting. Okay, like this, we split the term, and then because it doesn't change the meaning, right? Because we are coding
Right.
two terms, so it's like actually there is no one common terminology for this diarrhea and vomiting. If we are
Okay.
changing the meaning of the event, that is when we have to change or we have to look for the more specific term or the combination term? Usually in our normal situations, we can easily understand. I mean, we usually get, if the combination term, we usually get the coding option for the combination term. Okay.
Okay, you will have a coding option for that.
Yes.
So otherwise, if we cannot find vomiting and diarrhea under vomiting, then you again check for diarrhea, percentage diarrhea. If you cannot find under that also, then we
we'll
will
split the
create
term.
like split it, a
Yes.
new option, right? And then we
Yes,
will
correct.
select the other one. Okay.
Correct.
Thank you.
Okay. And then we have age versus event specificity. So now we can see in our Medra, let me. Okay, so if we put like event with elderly, or maybe if you put with adult, then you will find so many events with the adult maltreatment syndrome, or if it is dermatocytosis is reported. Okay, and if the patient age group is like in falling under adult, then we can choose this adult dermatomyositis rather than only choosing dermatomyositis as an event, because we have age specific LLD term options are available. Okay, adult rickets, RDS, like this. Yes, we do get so many event codings. I mean, we have so many event codings with the age group, with the age option is available. And then we have body site versus event specificity as well. So now, if you see like a hand, okay, so you will have all the hand rash or the hand numbness, and then you will find osteoarthritis or maybe edema of hands, malformation of hand, or like this. We have body site specific event terminologies are also there. For example, if a report clearly states the patient is having rash in upper limbs, then we have to select the LLT which has that option, like, okay, rash in upper limbs. We will see. Okay, we can just search. We have like edema, upper limb is there. So yeah, you will get the codings like this only. We just need to search for the terms, or maybe if it is not available, yeah, we have like rash hand or hand rash is there, I guess. Yeah, we have hand rash. So like this, we need to choose the body site specificity as well whenever we are selecting the LLT terms. It will be there irrespective of the body site. Okay, and then location specific versus microorganism specific information. So now if you just enter like any yeast, then you will find like we have vaginal yeast, or like urinary tract yeast infection, or like or the yeast infection of the skin, mouth, like this. We have location specific location specific and microorganism specific infection options are also there in the LLD. So we need to choose the terms as reported by the reporter. And the next one is modification of pre-existing condition. So what does this means? Modification of pre-existing condition is, so now let's see, the patient is having hypertension as a medical history. Okay, under this hypertension, after taking the medication, if it has worsened or decreased, so we have that options as well. So now let's see, not with hypertension. Okay, you can see hypertension aggravated. Okay, and then you can find hypertension exacerbated, and you will also find hypertension worsened, and like this you will find the modification of pre-existing conditions as well. Because why I am saying this is, sometimes we do get many reports wherein the patient's underlying condition or the pre-existing condition got aggravated, so we need to choose that LLT term along with the condition whenever we are coding such cases. For example, the patient is having like hypertension even before taking the medication, and after taking medication his hypertension has become he's worsened and it's unable to control. If that is what reported, then we have to come to our Medra and search for the aggravated condition, like that is hypertension aggravated. Okay, so
Okay.
this aggravated option is not available for all the events. In that situation, how do we select the event terminologies? In such situation, if the aggravation term is not available, one event that we select is the hypertension, and then with the same verbatim, okay, let me just show you with an example. So now, for example, if it is mentioned as hypertension aggravated after taking the medication. Okay, so now since the one option is directly there, so I'm just going to keep this option. If my aggravated is not there in my case, then what I'll do is I will select one term as hypertension. I'll just copy the same event with the hypertension aggravated, and other event term that I'll be selecting is the disease progression or something. So we have disease aggravation. So this option is what I will consider. Okay.
Okay. Okay. So in that disease or aggravation, we'll have to select just the disease aggravation. So it will show the terms for hypertension.
No, we just need to select the option of disease aggravation as an LLT. If
Okay.
because we
So
don't have the direct coding option,
first one, okay,
will
first
be
one you
only
did
hypertension,
hypertension,
and the
and the
second one
second
will be
you
the
did
condition aggravated.
condition aggravated. Okay.
Yes.
So what are
So
the HLT and SOC for all of for
that
disease
will be general
aggravation?
disorder. Yeah, it will be the
General.
general
Okay.
disorder. Yes.
Okay.
So now, I mean, in the current Medra version, we have an event coding option of pre-existing condition. Okay, we have like progression of pre-existing condition as well, or like, so that option is what we are selecting it in the current Medra version, because the one that I'm showing does not have the pre-existing condition. So but yeah, we have the option of pre-existing condition aggravated, so that option progression of pre-existing condition, that option also we select if that option is available, or else condition aggravated we can select. So this is
Okay.
this situation is for both for pre-existing condition and also for the product indication. For example, the patient is taking medication for hypertension, and after taking, if the patient condition was worsened, that in that situation also we follow the same scenario.
Okay.
Okay, and the next one is exposure during pregnancy and breastfeeding. So yeah, for that, let's see what are the different terminologies are there for the exposure during the pregnancy. Okay, pregnancy.
So do we write this percentage sign all the time before the term?
I mean, I always use, because so that in one time only I can get all the results that are there with this pregnancy term or any other term. So you can see the pregnancy, we have abdominal pregnancy, and then we have aborted pregnancy or abortion or ectopic pregnancy or immunization, anemia and pregnancy, and the normal any condition that is in the pregnancy. Yes, we have all those options. Diabetes complicating pregnancy or the diabetes in pregnancy. We have the false pregnancy as well. Somatoform disorder. Okay. Or that we have fear of pregnancy as well. Okay, and you can see the first trimester pregnancy. Okay, and then you do find some coding options like drug exposure during pregnancy. That's the searching for, yeah, drug exposure before pregnancy, during pregnancy. We have these two options. So depending on the reported information, we choose these options in the pregnancy cases. Okay, and then yes, we have the breast feeding as well. Okay, so you can see transmission of drug via breast milk, or the vaccine exposure via breast milk, or the drug exposure via breast milk. So this coding option is also available. This one we use majorly for the baby cases. Okay, and then we have all the congenital terms. So like I have shown in the yesterday's session, we have so many congenital terms are available. So what are the problem that the baby can have? All those problems are there in these congenital terms. Okay, and all the neoplasms. So the neoplasms we have in one SOC, all the cancer conditions. So you can see the multiple cancers, all the terminologies are available. So whenever any neoplasm is reported, it must go to the SOC of neoplasm, but not into the infection. That's what we have to keep in mind. And the next one is medical and surgical procedures. So if you just search in the, maybe in HLT, I'll search SOC. The surgical and medical procedure, you can see the different surgeries the patients can have. So all this information is there. So different, all the surgeries are there in our under this SOC of surgical and
SSC.
medical procedures. And investigation. So yeah, like I shown you in yesterday, we have the multiple investigations. So for each and every SOC, we have all the different type of investigations. So whatever the LLT or whatever the lab data that the patient have, we will have the coding option for all the lab datas. And then coming to the medication errors, accidental exposure and occupational exposures. Let me just show you in the medication errors what options can be there. I have just selected the HLTT term in my offline Medra, and then you can see the multiple options. So in the medication error, you can see circumstances or information capable of leading to the medication error, contraindicated device use or the device difficult, even if the drug like product administration error, product use complaint, or like wrong device or the wrong drug or the wrong patient has taken wrong route of administration. Like this, we have different terminologies are there for the medication errors. Okay, so yeah, I mean, anyway, I have special session which is planned for my for the special situations, the medication errors. There you will have some understanding because the most difficult part of the post-marketing reports is selecting and coding the medication error or the special situations. So though we regularly have processed these cases or irrespective of years of experience, still we do mistakes in selecting these medication error cases. I try to select few examples for our practice. Maybe you will get some understanding on how we receive those medication error cases. But meanwhile, you can look into it. What are the different type of medication error coding options we have? So in the overdose, we have different overdose. We have accidental overdose, intentional overdose, multiple drug overdose accidental, and multiple drug overdose intentional, and radiation overdose and the normal overdose. So how medication error cases will be reported, like for example, there is a patient who has taken 100 tablets of Tylenol, 100 tablets of acetaminophen, 100 tablets of other ibuprofen. So like this, the patient has taken different tablets to commit a suicide. In this situation, this term of taking the medication, the multiple medications in a overdose, we have to choose a term of multiple drug overdose intentional rather than the normal overdose or the other multiple drug overdose. Okay, so we have to understand the reported term and then we have to select the LLT terms according to it for the medication errors. Okay, irrespective of associated event or not, we need to always select these medication error terminologies. If there is an event associated, then we can choose it according to it. So let me just show you an example. This we have reported. Okay, this I can explain. Okay. Yep. So these are the examples of medication error. Here, if you see, the patient was administered wrong drug and experienced hypotension. So here, the LLT option we have to select for the patient was administered wrong drug is wrong drug administration and the event that is associated as hypotension. Okay. And the next example, where insulin was given using the wrong syringe, resulting in the administration of the overdose and the patient developed hypoglycemia. So here we have two medication errors. One is using wrong syringe, that is drug administration with the wrong device. And then we have accidental overdose because of this wrong device. And then hypoglycemia, which is the event because of this medication error. And also in case of medication was given intravenously instead of intramuscularly, yes, again we have LLT option of intramuscular formulation administered by the other route. Okay, and so like this we have so many events, and there is one specific situation here where if the pharmacist has noticed that the name of two drugs are similar and is concerned that this may result in a medication error. So here actually no one has taken the medication, and the error was the pharmacist was able to prevent this error to happen. In that situation also we have one LT option we select, that is circumstances or information capable of leading to the medication error. Okay, this LLT term we select in case of any medication error which was able to prevented by the pharmacist or anyone, any HCPs. Okay, so and then we'll go back to the term selection point. Still we have, yeah, I wanted to discuss this. Lab value. So in case of any ambiguous lab data is there, then we have to code it as abnormal, and if we clearly know the lab value is low or high, then we can select the LLT term according to it. So how do you know whether the lab value is within the normal range or not, because we cannot identify each and everything. So all the reference information will be provided in the source document. If it is not provided by the reporter, then we will have the information provided by the company in the form of Merck lab data values. The Merck document is the one that is used by all the companies as a standard for the lab value, upper limit and the lower limit. And based on that, you will identify whether it is an increased event or the decreased event, decreased lab value. Yes, and then yeah, we have all the overdose, drug toxicity, and drug poisoning terms, and then we have all the device related terms, drug interactions like drug-drug interaction, drug-food interactions, and we have the normal terms like the no adverse effects. So let's just see, no adverse drug effect. So that event are also there, and we also have the normal terms of adverse drug reaction. So like this, we have this normal terminologies are also there under general disorders, and unexpected therapeutic effect, that coding option is available. So for example, if the patient is taking a medication for example, we take the example of minoxidil. So minoxidil is actually a drug for the vascular disorder, but during the clinical trials, they have identified some abnormal hair growth in the clinical trial participants. So what they have done is they have developed this topical formulation of minoxidil. So now this topical formulation is used for the hair growth or the alopecia. So like this, if any beneficial effect is being identified, then this is the option that we select for that particular beneficial effect, which is unexpected therapeutic effect. Okay, and then we have modification of effect, that is aggravated, decreased, lowered. So that is the term which we have already discussed in the earlier slide. And the social circumstances, yeah, I have shown you about the multiple examples under the social circumstances. So if you see, we have age-related, economic, environmental, family issues, gender-related, legal issues like the crime victims or the criminal activities. So like this, we do get some reports as well, like lifestyle issues of alcohol use, okay, and the dietary issues. So all this information will be there. All the coding options are there in our Medra for social circumstances. And the medical and social history. So if you see some family history of cancer, CV disease, diabetes, like this, we have these coding options are also available. And then you can see the social like antisocial behavior and asocial reaction. Yeah, like this, these coding options are also available. Social alcohol drinker, social anxiety disorder, social degeneration, like these conditions are also available in our Medra. And all the indications for the product use, like irrespective of whatever the condition that the patient is using the medication, all the coding options are there in our Medra. And off-label use and the product quality issues are also there. Let me just show you what kind of product quality issues. So any product contamination issues, any product labeling issues like confusion, expiration date issue, or the label confusion, counterfeit issue, any packaging issues, any physical issues like color change, deposits, coating issues, physical issues, order, or the gel information, shape, size, solubility, taste, all these coding options are there in our Medra. Okay, so whatever the product quality issues reported, that we can use. And then yes, so we have multiple off-label use terms as well. Let me search with the PT in the off-label. You can see the multiple off-label use term. So yeah, depending on the reported term, we choose the correct LLT. It goes to the PT of off-label use. Okay, so these are the different term selection points are there in our Medra. So by using all these terms, we can select the event terms according to it, and then we have to choose the right LLT option. So that you need to aware that these different kind of options are there in our Medra. Okay, so the last before we go into the examples, I just need to clarify few definitions. So we have drug misuse, drug abuse, drug addiction, medication error, and off-label use. In which situation, which terminology we use? So the first one is drug misuse. So drug misuse means if the patient or the consumer is intentionally not taking the medication as per prescribed information, then we consider that event as drug misuse. Abuse means the patient is intentionally misusing the drug not for the therapeutic purpose, but he is just abusing because he is dependent on the medication. Addiction is also same. If the patient does not take the medication, he will have some reactions. That is where the patient is continue to take some medication. So those kind of terminologies, we have to code it as drug addiction. And coming to the medication errors are not intentional. So they can happen from the patient or the consumer or maybe by the healthcare providers. Off-label use. Off-label use means if the healthcare provider is prescribing the medication not according to the labeling document, then we consider that terms as off-label use. So whenever you are selecting this off-label use terminology, please make sure that this was intentionally prescribed by the physician or by any HCP, not as per the labeling document. Then only we have to consider the event coding of off-label use. Okay, so yes, that is what I wanted to discuss today, and then tomorrow we can start with the different examples. I have like almost 40 scenarios are there in my examples. Maybe tomorrow we can discuss all these different scenarios and to check for understanding on the Medra concepts. Do you have any questions?
Okay, not so far. I think Medra will be more with practice, right?
Yes,
Getting
it is.
like
Yeah, the more you
what
have the
terminology,
practice.
where we have to check, what we have to do. So you said that there is something which we can use for practice, which is offline.
CMC. Yep, I'll give you the link in the chat window.
Yeah. Also, yesterday, we spoke about the certification exam which we can give.
Just a
Which one is
moment. I'll just give you that link as well. Yeah, I remember I wanted, but I just forgot once I joined the session. Okay, yes. Yep, I gave you the second one is a CMC test.
Okay.
Okay, there you can see all the sample questions, exam format as well. So.
Okay.
Okay, so you can just look into
Look
it.
into it. Yes.
Yeah.
Look into it.
Okay. So meanwhile, I open my Argus. You can look into this case scenario. So what you have to tell me is you have to select what are the events, and once you select the events, then we will go and check the how we code that events in the case. Okay, so in this example is about 79-year-old mother, I mean, that is a female patient, started taking move-free advanced and this medication and experienced diarrhea and uncontrollable shaking, and she would feel hot and then start shivering from cold, and she seemed very weak, which is uncharacteristic for her. Okay, so now let's see. Now let me know what are the events we will select in this example.
Diarrhea. She
First will
developed
be
diarrhea
diarrhea.
Mhm.
Diarrhea, then
and
uncontrollable
fever,
shaky,
uncontrollable shaking and fever and cold.
feeling hot. Yeah.
Not fever, it is feeling hot and then feeling
And
cold.
shivering from cold.
Yes. And
She seemed very
Very
weak.
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 yes. So very weak. So these are all the events. So now let's see how we code the events. I'll open some case that is there. Okay. So now, yeah, diarrhea, we will have the direct coding option. Okay, if the direct coding option is there, then this is how Argus will automatically select the event. Okay, so this is how the event will be selected automatically. And then the next event, we have uncontrollable shaking. So for that, let's search. Uncontrollable is not there. So I'll just do the searching with the percentage shaking. Okay, we have like shakiness, shaking, shaking inside and shaking of hands. So here we can just select it as shaking as an
Then
LLT term. This is how we have to paste the LLT term to the description to be coded so that anyone, if anyone touch the case later, so the coding will not change. If we just keep the coding as uncontrollable shaking, and if if they enter tab here, the enter coding option will change. Okay, and the next we have is the feeling hot
feeling
and then
hot.
start shivering from cold. So it's a, for me it is looking like the combination term. So now let's search if we have any coding option that is available for this combination term. So I'll just check with the hot. Oh, hot. I'll just search with the cold because with the hot I have got so many terms, so that I'm just narrowing my search. So yes, so the cold. These are all common cold. Yeah, feeling cold is there. Feeling cold. Yeah, here you can see the term that is feeling hot and cold.
Cold.
So this combination term is there. So I'm going to choose this combination term. Okay, and then the next we will see shivering is also there. Let's see if we have any event with the shivering. Yes, we have the direct coding option. Okay, so it goes to the PT of chills. And then the next we have very weak. Let's see our event coding option. Weakness is the option that is one we majorly use for this term. So yes, I'm choosing the term weakness for the very weak. Okay, so done. This is how we code the events in this example. Okay, now let's see. Let's go to another example. So this is about the product called Femera, and the patient is been on this product for almost 7 months, and her BP is slowly rising to horrible number. So she was put on correct 2 times a day, but still the BP is still high, and the patient have been on it for a week, and the patient is also having joint pain and the chills. Okay, so now let me know what are the events in this example.
BP is high,
BP rising,
joint
and
pain and
joint
chills.
pains and chills.
Yes, correct. So we have these three events here. So what I'll do is I'll just open one Medra browser so that we can just search. Okay, so the first one is the blood pressure. So now blood pressure, they have mentioned it as blood pressure slowly rising. And like I said, for the convention of investigation and diagnosis, if the diagnosis of hypertension is mentioned, then only we have to choose the LLT term which is matching to the hypertension. Since in this example, the diagnosis is not provided, so we have to only select the LLT term which goes to the SOC of investigation. So now let's search. So we have BP abnormal is there, BP diastolic high, no, these are all BP diastolic. Okay, we have blood pressure high is there and then blood pressure increase. So we have two options here.
High.
If we select
Blood
blood
pressure
pressure
high.
high, it is going to the PT of hypertension and SOC of vascular disorder. And let's see about the blood pressure increased. So the blood pressure increased is going to the PT of BP increased and SOC of investigation. So here we are clear. The event that we have to select is the blood pressure increased but not the blood pressure high. Because if we select this term, it is going to the SOC of vascular disorder where the diagnosis is clearly mentioned. But in our current example, since the diagnosis is not clearly mentioned, we cannot choose this LLT term. And the next one, joint pain. Yeah, we have direct coding option for the joint pain, and even for the chills also we have the direct coding option. Okay, so now I'll move on to the next example. So it is about 28-year-old mother of two children developed jaundice and there are no obvious cause for hepatitis. And then investigation indicated some hepatitis but not of viral cause. And further case history found the patient is on the oral contraceptive and also the patient is taking short course of flucloxacillin 3 weeks prior to the hepatitis. Okay, so what will be the events in this case?
Jaundice.
Mhm.
Jaundice.
And
Hepatitis.
so we have, yes, hepatitis did indicate. So they have mentioned investigations did indicate hepatitis. So yeah, hepatitis is one event and jaundice is another event. So in this situation, we have to understand one thing first. We have to check whether the jaundice is the common symptom of hepatitis or not. So though we are aware of the common symptoms, but it's always best practice to check in the net to understand the common symptoms associated with certain medical conditions. So for that, we should always use the medical websites. Either can be Healthline or the Mayo Clinic or the Medline Place. So I preferably I use WebMD or the Mayo Clinic so that they have in detail details of the condition. So here you can see the common symptoms associated with the jaundice, so with the hepatitis are fatigue, nausea, abdominal discomfort, and low-grade fever, joint pain, and then intense itching. This is for hepatitis A. Okay, yes, and there is some yellowing jaundice as well. So yes, jaundice is also they have mentioned as a common symptom for this hepatitis. So when we are selecting the events, we have to follow some convention here. Now let me just so this fall under definitive diagnosis and the along with the symptoms. Now I'll show you how we tag the symptoms to the diagnosis. Okay, so now I have selected one event as hepatitis. Okay, and then we have to choose another event as jaundice. Okay, and here you can see some two radio buttons. One is for diagnosis and another for symptoms. So here jaundice we have identified as symptom. So I'm going to choose this radio button of symptom. Okay, and I'm going to establish the relationship. So in the right side corner here you can see the relationship button. Once you click, you will find all the unassociated symptoms. We just need to bring that to where we are going to tag. We can just use up and down arrows. So here if you want to add jaundice to the hepatitis, this is how you have to move the arrows. We have up and down arrow and then we just need to click OK. So now this symptom is tagged to this particular diagnosis of hepatitis. So this is how we follow or we process the cases, which includes diagnosis and the symptoms. Okay, is that clear?
Yes, Ramya.
Okay. I'll move on to the next example. So in this example, a patient aged 26 presented on this date with a history of fever, headache, body pain and sore throat of 3 days duration, and there is the temperature of 38.3 degrees, and inflamed oropharynx was identified, and the patient is been on this non-steroid injection since 2003 for her periods because of her irregular periods. And yes, that's what the information we have. So now let's read and let me know what are the events here.
Fever, headache, body pain, sore throat,
Mm-hmm.
inflamed oropharynx and rise in body temperature,
Mm-hmm.
and yeah, body temperature.
And yes, that's it. So here our suspect medication is non-steroid and the patient is taking suspect medication for the indication of irregular periods. And now our events are like fever, headache, body pain. So these all direct coding options we will get in our Medra. Let's see. Okay, fever, we can see the direct coding option. And then headache also we will get the direct coding option of headache. Okay, and then body pains also we have direct coding option. Okay, so here one thing we forgot. Okay, so here we will have coding of general body pain. Yeah, this is the option we code for the body pains. Okay, and so one thing that we missed here is the oropharynx inflammation. So let's check whether these can be the symptoms of oropharynx inflammation, which is pharyngitis in the medical terminology. Okay, so first let's understand the symptoms of this pharyngitis. So headache is there, body aches is there, fever are there, and okay, sore throat is there. So we have all these conditions. I mean, these are like commonly associated with the pharyngitis. So we will consider fever, headache, body pain, and sore throat as the symptoms for the diagnosis of inflamed oropharynx. For the so now let's see the coding of oropharynx inflammation. I first let us search in the Medra. We have only oropharyngeal something. So we don't have oropharyngeal inflammation as a separate coding. We only use the pharyngitis. So yes, oropharyngitis is there. So this is the option we select for this particular event of oropharyngeal inflammation. Okay, and which we called it as oropharyngitis, which is a diagnosis, and all these four will become the symptoms. Okay, and the temperature of 38 degrees, so where you will add this, because anyway fever is mentioned, so the high temperature, it will go in the patient tab. Now let me show you. So in the patient tab, so this is where we add the lab data. So here we will mention the temperature. We will have like this body temperature. Okay, and here you can see the values. We'll see if we have degree Celsius or something. Okay, so it's not there, and we just need to add data and right click, and then here we will add the results like 38, 0 degrees. Okay, so this is how we add the lab data if the diagnosis is mentioned. Okay, so shall I move on to the next example? Is this clear?
So in this pharyngitis will be the main disease, and then
Diagnosis.
in that, yeah, main diagnosis, and in that you will add fever, headache, body pain, and sore throat as the symptoms,
Correct.
right? Okay,
Yes.
thank you.
The next example is like about it is a literature case, but I have just taken the abstract. So let's read it. So the title also we have to consider for the literature cases. So it is a drug-induced liver injury due to the varenicline, which is a suspect medication. So you can read the case. It is about a 69-year-old Latino man who was referred to the hepatology clinic for urgent evaluation of new onset of jaundice, nausea, and fatigue associated with 40 times increase in his transaminase levels and the elevated INR and alkaline phosphatase. And now the patient had the prescription of varenicline for the smoking cessation therapy. And within 5 days of starting this varenicline, the patient developed these symptoms, and it was discontinued on day 5 by the patient. And some lab data evaluated revealed some acute viral hepatitis. I mean, there is no evidence of acute viral hepatitis, autoimmune metabolic, or alcohol related liver disorders, but the patient had the medical history of chronic hepatitis C, and then after discontinuing, the patient has become completely normal after 9 weeks. Okay, so in
So
this
here
case, what will be the events you will select?
the events nausea, jaundice,
Nausea, fatigue.
fatigue,
Then
and
the
then
lab will be um, investigations. Under investigations will be more than
like
40
transaminase levels,
transaminase, yeah,
and
transaminase
So,
then
what is the major
elevated.
diagnosis here? So, what is the major diagnosis and symptoms?
so major deep jaundice.
So, our primary diagnosis here is the
Hectic.
drug-induced liver injury.
Drug-induced liver injury. Okay.
Okay, so this will be our major diagnosis, and under this, all these jaundice, nausea, fatigue. Okay, let me
Okay.
just highlight them. Jaundice, nausea, fatigue, and increase in transaminase level will fall under as a lab data for the drug-induced liver injury. All the lab values, we will not consider them as events here because the diagnosis is mentioned. That is what I'm stressing out. Whenever the diagnosis is provided, all the lab data supporting to it, we will just add it in the patient tab, or else if the diagnosis is not mentioned, that is when we will add them as events. And we have nausea, one more, yeah, vomiting as one more event is there. So malaise and fatigue both are same, and then deep jaundice.
Deep
Okay.
jaundice.
Yeah, rest all we have some medical history of chronic hepatitis C as well. It's a medical history, not an event. I'll just unhighlight this. Okay, so now let's look into our Argus for the Event selection. So I guess in this older version, we don't have the drug-induced liver injury. Let's see. Okay, so only liver injury is there, but our case is specifically mentioned. It's a drug-induced liver injury. So now let's see in the current Medra dictionary, like how whether we have the direct coding option available or not. Kidney injuries, yes. So we have the coding option in the current Medra version with the drug-induced liver injury or the drug-induced hepatotoxicity. I guess we have hepatotoxicity with the drug-induced is there. It's a synonym anyway. Okay, it's not there in this older version, but yeah, if you see the current version where I have shown you, drug-induced liver injury direct coding option is there. So that is the one will be our diagnosis. Okay, and under which we add all this nausea, vomiting, jaundice, fatigue as the symptoms, and this 40-fold increase in transaminase levels, elevated INR, elevated alkaline phosphatase level will become our lab data. Now let's just let me show you how we add again in the lab data. So we just need to click on test. So here it is, transaminase. That's what they mentioned. They have not mentioned whether it is ALT or AST. So we can just mention it as transaminases. So option we have to select. If they mention it as alanine transferase or ALT or APT, then we can directly select the coding with those options. Since they are not clearly mentioned, we will only choose this option. And since they have just mentioned it as 40-fold increase, so the same we can mention. Okay, and it is the same with INR as well. INR direct coding option is there. So it's elevated. And then we have alkaline phosphatase. Okay, and then this one also they have mentioned it as elevated. Okay, so yeah, this is how we add our lab data in this case. And rest all yes will become a normal event. So now let's see for the smoking cessation, we will have any coding because it is the indication for the drug. So we need to check. I mean, I'm just showing you like in the products tab. This is where we add our indication. So let's just see if we have any coding option. Yes, we have the smoking cessation therapy option is there. So that is the one we choose for our product indication. Okay, so is this example clear?
Yeah.
Okay, then yes. Any
What
question?
about yes? What about the new symptoms, new onset? Like some of them are the same: nausea, vomiting. Then there is fatigue, malaise, deep jaundice. Okay. Since they are same, we are not coding again, right?
We have to, yeah, since they are same, we will not code it twice. I mean, one more convention that we have in our Argus or in any report is like same event should not be coded twice. If you code the same event twice, then it will impact the signal analysis. The signal
Okay.
analysis always work with the event number, not with the patient number or the case numbers. Okay,
Okay.
so that's why we should not use the same PT term should not be there in the case twice.
Okay. Got it.
Okay, so now I'll show a few examples of verbatims and then I'll give you some options. So you need to tell me like which option you will select in this situation. Okay, so the first verbatim is there is some clinical complication of IUD is what it is reported. Okay, Now let's see in our Medra what are the options we have. So we have like IUD complications, which is going to the PT of complication associated with the device, and we have intrauterine death, which is going to the PT of fetal death, and then we have third option of unevaluable event. So in this, which option you will choose for the coding?
First one, IUD complication. IUD complication.
So here, but the reporter has not clearly mentioned. So this fall under some ambiguous information category. Okay, it's
Okay.
because IUD can be intrauterine device or it can be intrauterine death. So since we don't
Okay, both.
so we have to choose the option 3, which is unevaluable event, because we
Okay.
cannot take any other. I mean, first we have to request the follow-up to understand what is this IUD is.
Mm-hmm.
Okay, and then we have to choose the options accordingly.
Okay.
Okay, so next example. So we have hypoglycemia with the blood glucose, and these are the options. So now let me know which options you will choose in this example.
Blood
Let me
glucose
know.
increased. Option 2.
But here you have to see it is reported as hypoglycemia.
Abnormal. Yeah, I think it
Yes.
will be
So
first
since it
one.
is a
Abnormal.
conflict information
Yeah.
we have, the diagnosis
Yeah,
is
because it
actually
is
reflecting as low blood glucose, but the actual lab value is
investigative
the
values
high
are more.
increase in value.
Yeah.
Okay,
Yeah.
so in this situation, we will go for option 1, which is
One.
blood glucose abnormal.
Abnormal.
Okay, then I'll move on to the next example. This is the combination term example. So we have like retinal disease from HIV with the near total blindness in the right and left eye. And these are the options we have. So which one we will choose?
Fourth one.
Yes, correct. So since
Yes.
we have the
HIV.
option of HIV retinopathy blindness in the both the eyes, that is the option we choose because it includes all the combination terms. And the next example, like some tests showed increase in serum creatinine and the BUN and also increase in the serum creatinine and the BUN ratio. And these are the options we have. So which one you will select?
Option 3.
So here for the lab data, we need to select all the available options. So in this example, we have to choose the 1, 2, and 3. These
All
three
three.
coding, yes, we have to select because they have clearly
Okay.
mentioned there is increase in serum protein, BUN is also increased, and along with the ratio. Since they have mentioned
Okay.
we should not categorize them as one event term, we also have to code these events separately.
Okay.
And the next one, so due to some prescribing error, the child was given with some drug X, which is labeled for use in adults only. This is an example of medication error situation, and these are the options we have. So which one we will select?
Drug prescribing error.
And
Medication error.
Medication error.
And I think
But
first one
first
also.
one, adult.
yes, first
Yeah.
and 1 and 3 are the options we have to choose here. So
Okay.
first one is the drug prescribing error, and the next one is adult product given to the child accidentally.
Okay.
Okay, the next example. The patient's renal function was measured every 6 months instead of on the monthly schedule recommended in the label for the drug. Okay, so these are the options we have. So which one you will choose here?
Option 3. Drug monitoring medication incorrectly
Correct. So that is the option we have to choose here. Okay. Yes. The next example, the patient unintentionally took more than maximum recommended dose due to the dispensing error. And these are the options we have.
And 2 and
1
3.
and 3
Correct. So 1 and 3 are the options we have. So that is because they have
accidentally taken more.
no. So now in this situation, what we have to do is,
Mhm.
so we have to understand the case first. So here the patient unintentionally took more than maximum recommended dose, but they are not okay. So sorry, I got confused. So it is more than maximum recommended dose means overdose. So there will be like two situations will be there in the in this overdose and incorrect dose or the administration. Okay, so in our labeling documents, we will have the maximum recommended dose on a daily basis. Okay, they will mention. I mean, we know the frequency, like for example, the paracetamol every 600 mg can be taken like every 6 hours. That means overall in a day we can take up to 2400 mg of paracetamol. Okay, that is the daily dose. Okay, and if the patient is taking extra dose, like if the patient is taking 3000 mg, then we can select it as extra dose administered because it may not be in the overdose section. So I will not confuse you more. So here the maximum or the daily dose, recommended daily dose for the paracetamol is 2400 mg, and
Mhm.
the maximum recommended dose is the 5000 mg. Okay, and if the patient
Okay.
is taking any dose between this 2400 mg and this 5000 mg,
5,000.
we call it as incorrect dose administration or the extra dose
Okay.
administration. Okay, and if the patient is
Okay.
taking more than 5000 mg, then only we
Then
have
it
to
will
select
be
the option of
overdose.
overdose.
Okay.
Okay, so
Got it.
that point we have to consider whenever we are choosing it. So here in this example, they have clearly mentioned it is more than maximum recommended dose. So we can
Mhm.
choose the option of accidental overdose. And anyway, the
Okay.
drug dispensing error. Okay, so these are the examples for the normal situations and the different cases. Okay, so now let's see with the special situations. Okay, so first we will understand what are the special situations are. So these special situations are non-standard medical conditions, okay, which provide valuable information about the medicinal product even when they don't occur in association with an adverse event or the medical condition. That means all these medication errors, all these overdose product quality errors, these all fall under these special situations. And why this information have to be collected? Because to comply with the regulatory authorities, it is a part of our guidelines to collect these special situation reports. And for the clinical trial cases, in the protocol also they clearly mention which reports have to be submitted. And for the continuous benefit and risk assessment of the medicines also, we have to collect this information. So now, what are these special situations? Like I said, the drug overdose, drug abuse, misuse, off-label use, occupational exposure, lack of therapeutic effect, all the medication errors. So that means the list is very exhaustive list. So yep, we will see some examples like how we handle the cases in the special situations. So the first thing is we need to understand what is that special situation is. Okay. And here for example purpose, I have just given you the clue as well. Okay. What can be the error? So now let's look into the first scenario where it is mentioned my husband missed a dose as the pharmacy was out of stock and the patient missed doses as the delivery got lost and reordering took several days. Okay, so yes, what
Okay.
will be the events you will select in this example?
Missed dose
Missed product, missed medication,
And
and
out of stock. Pharmacy was out of stock. So, mm,
okay, let us search. Okay, so first one is missed
Missed
dose.
dose.
Just a moment. Okay, sorry for that. Okay, so now let's see what will be the event coding. So I mean, since in the older version we will not get all the options, I'll just search in the updated version. So now let's see in the missed dose. Yeah, we have the direct coding option for the missed dose is there, which will go to the PT of product dose omission issue. Okay, and the next option, first let's understand the case. So here the patient is not able to take because the pharmacy was out of, out of stock, and reordering took some several days. So these kind of issues, we call them as supply chain errors. Okay, so now let's see if we have any coding with the supply chain or the restock issues. Yes, we have some drug supply chain interruption as one coding, and we have drug supply chain interruption due to pandemic is there. So two coding options we have, since they have not clearly mentioned why there is a mist in the supply chain. So we can choose the option of drug supply chain interruption as the another event code, and which goes to the PT of product supply issue. Okay, so these are the two options we have to select in the both the situations. Okay, then I'll move on to the next example. So here the patient missed doses as he wanted to go on vacation and he did not want to carry the home infusion kit in the luggage, and he was very well aware that he was not following the doctor's instruction. So how do you handle this case?
So missed doses and non-compliant patient.
Correct. So yes, whatever the things you have said are the correct. So one is clear, that is the drug missed dose. Now let's see what are the different type of the missed dose options we have. So we have intentional missed dose and missed dose in error is there, and normal missed dose. But our case example fall under intentional missed dose.
Intentional.
Yeah, so we will choose the verbatim of LLT of intentional missed dose in this scenario, and it will go to the PT of intentional dose omission. Okay, and then the another one where we are talking about non-complaints. Okay, so let's search with that term only, since you have mentioned if you find any event. So we have treatment non-complaints, diet non-complaints, and other non-complaints. So here it will fall under the LLT of treatment
Treatment.
non-complaint. So this is the LLT term we choose in this example. Okay, so the next one is patient could not receive the medication because of insurance issues.
Um,
Here, lack of
insurance.
medication.
Again,
No, no,
maybe
the same.
missed dose. Yeah, missed dose. But
Correct, correct.
another will be insurance issues. We can check that.
Yes.
What is there under that?
Yeah, here it is only missed dose, and another one is insurance issue direct coding option is available. I think so. Yes. So we have the insurance issue as a coding. So this is also one of the event term we can
Okay.
choose. Okay, and then the next example is about the next we are going to discuss about the off-label use cases. So whenever the off-label means the physician is or the is wantedly prescribing a medication not as per the labeling conventions, then we call it as off-label use indication. Okay, so in these situations, how do we handle the cases? Now let's see some examples. So here, the drug use indicated for use in adults and used off-labelly to treat a 6-year-old children. So what will be the medication error terms you will select in this example?
Off-label incorrect medication.
Yes, off-label error is correct. I mean, off-label error is one event coding, and there is another event is there here.
So is there like a term for drug used in children?
Yes, so the same term we can use. That's the same form. That is adult
Adult
product
use.
administered to the child.
Child. Okay.
This is also option we have. That option also we have to select. Okay, so now let's see another example. The patient received some drug X for the breast cancer and experienced stroke. And yes,
So
here the
stroke
drug,
is
Like
a
adverse effect,
event.
adverse event.
Adverse.
yes, correct. So here the drug
Yeah.
X for breast cancer is off-label indication. So yes, so if you know the clear-cut case, we can choose this off-label indication as well as one of the term. Okay, and the next is the patient received less than prescribed dose as the pump had some defect. Okay, so
Device
in the first
defect.
example, mm, not all will be the events we will select. Yes, go ahead.
Lack of dose.
And device defect.
I mean, first we'll start searching and then we will see. So first
Okay.
thing is patient received less than prescribed dose. Okay, so
Hmm.
less than prescribed dose, how do we search? So we have two options, overdose and we also have underdose option.
Underdose.
Okay, so
Underdose.
let's search with the underdose. So we have like radiation underdose, intentional, accidental, and the prescribed underdose is also there. Okay, so now what we will select the term because of the problem with the
Competitive
pump,
defect.
the patient not able to receive, so it will be an accidental underdose because it is not an intentional, and we clearly know this was happened because of the medication error. So the event coding we select in this situation is accidental underdose. Okay, and then we have another event. Okay, here
The
which
vaccine.
is the same thing. The patient received less than a prescribed dose because
Pump
of the
had a defect.
Hmm.
pump issues. Okay, so how we search here is we will directly search with the pump if we see any coding option, because you know we have everything in the
Mm-hmm.
Medra, so we can just search with the pump only. So we have
Mm-hmm.
pump over infusion is there, but our case is under infusion, so we will
Under.
check for under infusion. Yes, we have the option of pump under infusion. So that is the one of the event coding we can select in addition to the accidental underdose. Okay, the next statement, the vaccine was leaking through the barrel of the plunger. Okay, so what will be the event coding we will select here? There is only one
Like
event here.
device issue.
No, we'll directly search with what are the terms that is there in our case. I'll search with the leak. So if I
Leaking
get any option.
vaccine.
Okay, leak. I mean the barrel of the
Plunger.
plunger means plunger is the syringe where we administered. So plunger is a part of syringe. So maybe we can search with the syringe leak or something. That will be the best. So we need to understand what actually it is. So we have syringe leak option is there. So we can directly go ahead and select that terminology. So plunger is a part of syringe. So I'm just searching with the term, and leak also there. Okay, so the next one, nurse got solvent in the eye as she had difficulties in using the reconstitution kit. So it's an example of occupational exposure. So what will be the event terms you will select here?
Solvent.
Solvent in the eye. And
Yes, so that we call it as occupational exposure. So now let's search
okay.
in the occupational exposure only. What can be the options you will get? Occupational exposure in eye, maybe that option can be available. Inhalation therapy, exposure to the drug, the agent, occupational exposure to. Yes, we have the option occupational exposure to the product via eye. So that is the one event coding we will select, and then we have some difficulties when using some device.
Kit.
So we have to select the option of device difficult to handle or use. So we have device difficult to use option is there. That is the one we can select in addition to the occupational exposure. Okay, the next example where we have the multiple errors. Okay, so here there's a physician who always prescribes some drug A for me as I'm allergic to the drug B, and which is much cheaper. But in the holiday fill-in check-in, didn't check my files and wrote the drug B down, and which I and pharmacist also didn't recognize. Okay, and the patient nearly died from an anaphylactic shock when she took the medication. Okay, understand the case first. Okay, and then select what can be the events and let me know, and then we can search in our Medra together.
It is like prescribing medication error.
Yeah, prescription error and
It's not
accidental
a prescription error, it's a dispensing error actually.
Okay.
holiday filling checked my files and wrote drug B down.
Pharmacist didn't recognize it's actually dispensing error because whenever they are dispensing
Okay.
it, they have to check the allergic reaction. I mean, they have to check the profile of the patient profile to understand which medication they are allergic to.
And then anaphylactic shock.
Okay, so here we have one event of drug dispensing error. Okay, and then here the patient clearly mentioned the patient is allergic to the drug B. Okay, that means it is documented, and they also mentioned in the patient files it is documented. So let's see if you have any event coding option. I mean, this example may be somewhat tricky for you. Maybe you will not get that. Okay, so that is documented. Allergy to the administered drug. So this option is available. Okay, since the patient is administered to the drug which is already documented, in that situation we have to choose this LLT term. Okay, and then The patient is having anaphylactic reaction. So that is the one of the event we have. Okay, anaphylactic shock, we will have the direct coding option. Anaphylactic shock. Okay, direct coding option is there, which will go into the some serious problem. Okay, let's see what HLT. It's going to the circulatory collapse and the shock or the anaphylactic response. It's going into that SOC category. Okay, and there is one more event that is hidden here. Okay, for any medication to be whenever the patient is administering, in each and every product leaflet or the labeling document, they clearly mention if in the contraindication section, like if the patient is allergic to this medication, then they should not take this. And in any situation, if the patient is taking, then we have one event coding option available, that is contraindicated drug administration. Okay, so how we will select, like I said, in the labeling document they'll clearly mention. So contraindicated drug administration, contraindicated. Okay, it's not there in the older version, so let's just check in the current version. So we have contraindicated drug administered and the prescribed two options we have. So here we select the option of administered. Okay, so now I'll present you the last example. Maybe this one also might be tricky for you, but just try. Okay, we can look into the case. So this is about a patient who was accidentally prescribed drug X. Okay, and because the name of these drugs are similar, and the pharmacist thought there might have been error. So prior to dispensing, the pharmacist asked the physician, and the physician has confirmed the error which was intercepted, and the patient received the corrected drug. Okay, so this is the case. So what do you think are the events? Are there any events in this case, or like is it like are there any medication error is here or not? Because in this example we can clearly see there is actually patient is not involved. That means the patient has not yet taken the medication.
No medication error.
So he was prescribed drug X, but he has not taken it. He or she has not taken it. But the pharmacist already spoke to the physician and confirmed, and so the patient was given correct drug. So I think there was some error, but it was rectified.
But for that we have discussed something, right? In our yesterday's session, there is one event coding option we select whenever any error is prevented.
Okay.
Okay, so in our, Divya, maybe you might have been not aware, but yes, in our yesterday's session, we have discussed about this example. Okay, any error which was prevented before it happens, we have the one event coding option, that is circumstances or the information capable of leading to the medication error. But here, the reported term, the reporter has clearly mentioned it as intercepted. Okay, there is some prescription error, but which was intercepted. So now we will search in our Medra if we have any coding options available with intercepted. Yes, we have intercepted drug administration error, medication error, drug prescription error. So yes, we have the event coding option of intercepted drug prescribing error. So though the patient is not yet taken the medication, still this case will be valid for these events. And we have one more event here, which is the name of drug X and Y are similar. Okay, that we will select it as drug name confusion or something. Yeah, the drug name look alike, sound alike. So these coding options are available. So we can choose these LLT terms. Okay, so these are the examples I have chosen, and there may be some ambiguous verbatims as well, like difficulty with the needle insertion. So for this, if you have this multiple, which LLT you will choose? Like we have like needle issue, device difficult to use, wrong drink induction, infusion site induration, or poor venous access, or unavailable event. So here, if you see why the patient is
Difficulty with
having
the middle
difficulty, it can be
insertion.
because of middle issue or it can be
Middle
because of
issue,
poor
poor
venous
venous
access as well.
access.
Since we don't know, we have to choose the option of unavailable event.
Event.
Okay, so it is not always easy with the special situations, and whenever we
Mm-hmm.
have a spontaneous reports, especially, I can't even imagine like how much effort we have to keep. Though the cases are like single line cases. I mean, whenever if we receive single line cases, we'll be feeling like very happy. Okay, I have only single line case. But if that single line case is a special situation, then our entire day will go waste in searching the correct meta term.
Okay.
Okay, so yep, that is about the Medra. Okay, there are some ambiguous terms like whenever there is any ambiguous term, the one option that we choose is feeling abnormal. Okay, so that's our best to go coding option. If you see this example, let's see. The patient went to hell. So what the hell is this event? That's what we feel whenever we see such verbatim terms. Okay, the patient went to fail or recurrent fatal stroke. How can a fatal stroke can be recurrent? The patient is dying and then the patient is coming back to the life and then again he is having stroke or what? Okay, so like this we do get multiple situations. So we can mention the can't control patient during the menses. I mean, so like this, some vague terminologies are there. We know like there is something wrong with the patient, but we cannot search even in the Medra. To search also, we have to think 100 times like how to search in the Medra. So in all these situations, one of the best option we have for the coding is feeling abnormal.
Feeling abnormal.
And I will tell you one more trick that we use for the coding. So in our Argus, we can just search with the terminology. So until now, we have been searching with all the different options of the event coding. Okay, so now I'll just tell you how we can search the cases. Okay, so now we just enter the event verbatim. Okay, so now maybe for simple example, I'm just showing diarrhea. Then I'll get all the cases which is having the verbatim of diarrhea. Okay, so like this, if you have any like this kind of terminologies
Similar
like I've shown you
case.
in this multiple examples, if you have any of this, then you can just put that verbatim and then you can just see what it was coded by our earlier processors. That thing that we can use if you're not getting any coding options by yourself in the Medra, we can search in our database because the database has some thousands of the cases, so we can look into our previous processors like what they have done and then we can so that we
Mm-hmm.
can have some idea on how to search the events.
Okay.
Okay, so this is all about Medra. I hope you got some understanding on what is Medra and what do we do with the Medra.
Yeah, definitely. But I think Medra is more of a practice. Like the
Yes,
more
Medra
we will
is
practice, the more we will learn because each
yes
case will be
because
different.
correct, we have a lot of
Ramya,
things. Mm-hmm.
you told that you'll give that Medra website for
Oh
certification.
yeah, yesterday I have messaged you. Okay, I'll ping that again. No issues.
Okay.
Yeah.
Okay. Yes, I have pinged in the chat window, so that's the where you can see.
Okay,
They
Ramya,
have
I
some
got it.
sample test as well, so you can just review that website and then you can see where it was
Okay.
mentioned.
Okay, Ramya. Thank you.
Yeah. Okay, then if no further questions, that is it I have. Then we can conclude our session for this week. Okay, and then Monday we
Okay.
can have another discussion about the listedness and causality and the narrative. So by the end of next week, we'll be completing all the ICSR topics.
Okay. And
Okay,
what are
Ramya.
the ICH guidelines? Do we have like I see it everywhere mentioned that should be there in ICH guidelines. So is it like some guidelines that or
I'll just
is
show you. I mean,
every
anyway, yes, there are some guidelines, but whatever the point that we are discussing, they are from
Mhm.
the ICH guidelines only. But you know, the
Okay.
ICH guidelines does not talk in detail. So in that ICH, we have pharmacovigilance guidelines of like some 6 to 7 guidelines are there under efficacy. I mean, at the end of the session, I'll be showing those documents and then I'll explain like where I have taken the points from this document so that if you want to
Okay.
have some quick revision, that will be like kind of quick revision of these ICH guidelines.
Okay. Okay.
Okay, you don't need to worry. So I will be covering all these different, I will show you like the different guidelines as well, where we have taken the references.
Okay. Yeah, GCP and ICH are the two things I see.
GVP and ICH.
GVP. Okay.
Hmm.
GCP. Okay. Good clinical practice.
GCP of clinical trials. GVP is for pharmacovigilance.
Pharmacovigilance. Okay. 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 Thank you,
Okay.
Ramya.
So, like I said, the main objective for our expedited reporting are the seriousness assessment, expectedness, and the causality assessment. So, now we will understand in detail how and which case we have to prioritize first. So, as per the pharmacovigilance process requirement, the event which is having major impact on the public health, those cases are what we have to concentrate. So how do we know like which case is impacting? That we can understand it from our seriousness assessment. So now let's understand the definition of serious adverse event. So as per the ICH E2A, so they have defined a serious adverse event as any untoward medical occurrence or effect that at any dose which result in death, life threatening, inpatient hospitalization or prolongation of existing hospitalization. That means the patient has to be in hospital for more than 24 hours, and persistent or significant incapacity or substantial disruption of the ability to conduct the normal life function, that is disability, and any congenital anomaly or the birth defect in the babies, or if the event is to be if the none of the seriousness criteria were mentioned, but if we feel like this event is like kind of important medical event, and if we don't treat immediately, the patient may result in the death, life threatening or hospitalization. So, and here we use our medical judgment and we need to apply some knowledge in our we need to apply our medical knowledge as well here to understand whether the event is serious or non-serious. Okay, so now the first criteria. So the definition you must remember. So the definition of seriousness criteria. So the first point is the death. So one, and how we select the death as seriousness criteria. So the first and more important, the death must be reported by the reporter. Then only we can consider it as a reportable event. Okay, in case if the death is not mentioned, and if the reporter has just mentioned like the patient died while on or after receiving the treatment medication without any underlying cause, still we can select the event term as death, and then we can consider the case as valid. And if the reported event is completed suicide, so we have already discussed about the suicide situation, like suicide, attempted suicide, completed suicide. If it is completed suicide, then yes, our seriousness criteria will be death. But if it is attempted suicide, it will be a normal serious event. And if we don't know whether it is or what kind of suicide it is, then we have to follow up with the reporter so that we have complete information about what actually happened to the patient. And in some cases, a patient may die coincidentally because of some factor which are unrelated to the adverse drug reaction. So maybe the patient died because of some surgical complications or because of any underlying condition, but not because of adverse drug reaction. In this situation, the causality assessment we can rule out, but the seriousness should be death only we have to consider. Okay, so because once we select the seriousness category as death, and if you also select the causality factor as related or unrelated, that will impact our reporting regulations and also on the product marketing status. So we need to make sure we are selecting the correct seriousness criteria for the correct events. And like I said, if there is no underlying cause was mentioned for the death, then we can choose that event as death NOS or like not otherwise specified, or we can also use the sudden death as an event. So in these situations is where we use the death. Again, I have so many examples you can read through, and then you can understand how actually it will be reported in the practical scenarios. Okay, so the next seriousness criteria is life threatening. So life threatening, usually it has to be reported by the reporter, and if it is not reported, still we can take some call and we can consider the events as life threatening. So now we will understand in which situation we will consider it as a life threatening seriousness criteria. So the life threatening condition is, it is a reaction in which the patient was at risk of death at the time of reaction. Okay, that means when the event happened, that is when the patient is at risk of life threatening, and it does not refer to reaction that hypothetically might have caused death if the event is more severe. For example, we know like any cardiac arrest or any myocardial infarctions or the stroke are like fatal. So if they're not treated immediately, but still we cannot assume it until and unless there is some information is mentioned. So if the patient is gone to the patient was hospitalized because of cardiac arrest, then the seriousness will be hospitalization, but not the life-threatening condition. But if the patient is requiring coma, I mean the patient is in coma or requiring any ventilator support, then in that situations we can consider the seriousness as life threatening. And any treatment like any intensive care treatment for the any allergic bronchospasms or blood dyscrasia or the convulsions, these factors also we can consider it as life threatening conditions. So, and also, like I said, if the reporter clearly mentioned in the document, then we can consider the seriousness as life-threatening. Okay, so these are the points you need to remember for the life-threatening seriousness criteria. And the next one is hospitalization. And like I have said in the definition, hospitalization means the patient has to be in hospital for at least 24 hours. Okay, or it can be any prolongation of hospitalization as well, but it has to be more than 24 hours. Then only we can consider the seriousness as hospitalization. So usually we can easily understand about the seriousness criteria from the document, because they will mention like the patient was admitted in the hospital or the patient was discharged. Like this we get some information. So now let's see few sample scenarios where we cannot consider the seriousness as hospitalization. So the first one is like if the hospitalization is described in the protocol, that means for any drug administration or drug monitoring, if the or any lab data is required to be done, or the patient has to be in hospital at least for 24 hours to monitor after receiving the medication, in that situation we cannot consider it as a hospitalization. And if the patient is hospitalized for the social reasons like patients with some Alzheimer's disease, they will go to some rehab therapies. So these kind of treatments also we cannot consider as hospitalizations. And also any pre-planned surgeries and any emergency visit or accident visits where the patients were treated in outpatient and then released, that situation also we cannot consider them as hospitalization, because because we do get many scenarios wherein the patients will having some emergency room treatment, which the reporter will consider it as hospitalization, but as per the definition it is not a hospitalization, it is just a normal criteria. And like I said, any observation after 24 hours or any procedure done in an outpatient basis, and there are some treatment which are like are not associated with the hospital, but usually they can use for minor surgical procedures like laser eye treatments or the chiropody surgeries. These are all does not fall under this category of hospitalization. And the next one is disability criteria. So the disability can be considered as seriousness criteria if it is reported by the reporter, or any loss of body part because of event, that is any amputations, and any injuries that are requiring major intervention, that is any surgical intervention, or any burns that is requiring debridement of the skin grafts, or any loss or substantial limitation of bodily function lasting greater than 7 days. That means the body functions like breathing, dressing, undressing, or drinking, eating, or excretion, or getting into or out of the bed or the chair, or the hearing, seeing, sitting, sleeping, walking, any body functions are having some limitations for at least 7 days, then we can consider that particular event as disability. Okay, so this is when we consider the event as disability, and these are the points we have to remember if you are choosing the seriousness criteria as disability. And the next one, congenital anomaly. So yes, congenital anomalies are the birth defects or the congenital disorders, the congenital malformations or the abnormalities. These are the conditions of prenatal origin which are present at birth, potentially impacting on infant's health, developing, and also on the survival. So this criteria we need to use only for the pregnancy cases, especially if the baby is having any event because of the drug that is taken by the mother. Okay, so and if the baby had and the for the seriousness criteria, usually before all the congenital anomalies, I mean any event which is going to the SOC of congenital anomalies were considered as serious. But from the last March, we are being the IME list have been updated. I will show you what is this IME list as well. So in that list, it will include all the events which has to be considered as serious. So the list has updated and they are clearly given us the events which we need to consider as serious, which we should not consider as serious. Okay, and only those events is what we have to consider as the seriousness criteria where we have to check the congenital anomaly. And the next one and the most important one, that is seriousness criteria, which is important medical event. So to select this criteria, we need to use our medical judgment. Okay, so like for example, if the patient is requiring any medical or surgical intervention to prevent one of the outcome listed in the definition, that is death, life-threatening, or like hospitalization, for example, like any third degree burns or the second degree burns or the normal first degree burns, which are requiring some intervention, or maybe any type of allergic reactions which are requiring some steroid treatments. You know, the steroid treatments are given for intensive cares. So yeah, any such treatments, I mean, we have to consider the events as important medical event. And if the reported event is present in the IME list, which is prepared and maintained by the EMA, I'll show you that list as well. And also, if the event is which meets the criteria of grade 3 and above in the CTCAE list, that is common terminology criteria for the adverse event, it has to be considered as a serious events. Okay, so now I'll open some case. I mean, I'll open the documents for what is this IME list is. Okay, so yep, this is the IME list which is prepared by the Medra. So this is the last version, which is from the September, the current version which we are using currently. Okay, so now you can see all the PT terms. So that is why I said the event selection and coding is the major, because from the event coding only we are going to select the seriousness, causality, and the labeling assessment. So the PT names is what you can see. So this list has some around 700 or like some 1000 are there. Okay, there are so many events are there in this list. So what are the PT terms that are there in this IME list have to be considered as the event. Okay, yes, there are almost 7,000 terminologies are there, 7,000 PT terms are there. So all the events which are there in this IMA list must be considered as serious event irrespective of the reported. I mean, if the other seriousness criteria are reported, then we no need to look for this IMA list. If other seriousness criteria is not reported, that is when we have to come and look for this list of the events. Okay, and the other one which I said, which is a CTCA guidelines. So let's see what does this include. So the CTCA guidelines also, it has categorized the events into multiple grades. Grade 1 are the mild symptoms like normal symptomatic, or maybe mild symptoms like normal fever, nausea, or whatever the normal symptoms will fall under this grade 1. Grade 2 are the moderate, where some ADL is affected, that is activities of daily living, or any treatment is required. So in that events, they have categorized as grade 2. Grade 3 are severe or the medically significant, that is like hospitalization or any intervention or life threatening. Grade 4 the life threatening, and grade 5 are the death events. Okay, so now let's see how they have categorized. So you can see all the SOC terms again. Okay, for all the SOCs, they have selected few PT terms. So for example, in our case, it was reported as the patient is having hemoglobin level of like 3 millimoles per liter. So now you don't know, by just looking at the value, you cannot understand whether it is a serious event or non-serious event. That is when our CTCA guidelines will help. So here you can see under anemia, they are clearly mentioned if the hemoglobin value is less than 4.9 millimoles per liter, which is falling under grade 3, and our guidance states any event which is in the grade 3 and above has to be considered as serious. So now you know you have to upgrade that particular event. Like this, for a particular diagnosis, there will be some common symptoms associated. So they have categorized all those common symptoms in the different grades in the CTCA list. So we need to search for the particular event if that information is there. So now if you see, the patient is diagnosed with eosinophilia. So the normal eosinophilia it is a non-serious event, but they are clearly mentioned if the patient is receiving steroid treatment for the eosinophilia, then it is falling under grade 3. That means this event has to be considered as a serious event. Okay, so that is how we choose for each and event. We have to select and we have to understand what is there in the CTCA guidelines, and we have to document it accordingly. Okay, is this clear, the CTCA guidelines?
Yes.
Any questions?
Oh,
Okay.
Ramya.
Okay, so okay. And this is how we select our, and sometimes we do use our medical judgment as well. So for example, now the patient is having some fever. Okay, fever for 48 hours is like kind of non-serious only. I mean, fever, it will be there for the 48 hours. But now after 48 hours, if the fever is in the increasing end, not at the decreasing end, then that means the fever is at like 40 degree plus, or then we have done that will become a serious event. Okay, so in these situations, we have to consider we have to understand the particular disease first, and then we have to decide whether it has to be considered as serious or not, depending on our medical judgment. So now the fever will not be there in our EMA list or maybe in the CTCA list, but in the situation we have to understand and we have to analyze whether it will have any serious impact on the patient health or not, and then we have to select the seriousness criteria of IME. Okay, so now let's go through some examples so that you will understand how to select the seriousness criteria for these events. So the first example is, you can also read through it. So it's about a male patient who will receive the Gilenya drug for the multiple sclerosis, and the patient also treated with some other medications like metoprolol, beta blocker, amlodipine, calcium channel blocker, and the patient had completed 6 hours of monitoring after the first dose without any incident. That means after taking Gilenya, the patient has to be monitored for 6 hours because this drug can cause bradycardia or like AV block. That is why the patient has to be monitored, but died less than 24 hours after the first dose, and the exact cause of death has not been established. So now in this example, what will be the event you will select and what will be the seriousness criteria?
Actually, here patient stayed less than 24 hours.
Death will be the event. And
Yes.
so seriousness criteria was life-threatening death. So it will be considered as
Seriousness criteria as well. So the
death.
event
Yeah,
is
Better.
event that we select is the unknown cause of death. Seriousness is also death.
death.
Okay, that means it's a fatal case. So what are the timelines for fatal if it is a clinical trial case?
Less than 7 days.
Yes, 7 calendar days. And if it is any other than clinical trial report, we have 15 calendar days.
15. Yeah.
Okay, so the next example about 15-year-old Annabelle has been on some limicycline drug for a year and her acne has not improved. So she wanted to switch to some other medication. So she switched to Roaccutane drug before she got scaring. And after 9 months of taking this drug, this school girl took her own life. So what will be the event here and what is the seriousness criteria?
Event is suicide.
Okay, what suicide it is?
Completed. She
Yes,
already.
correct. Which was clearly mentioned, she took her own life. That means the patient died here. Yes,
Yeah.
the event selection is correct. That is completed suicide. The seriousness is
Seriousness is death.
okay. Yes, correct. So the seriousness is death with the completed suicide. The next example, about 13-year-old boy presented to his primary care provider with a 5-day history of abdominal pain and the 2-day history of diarrhea and vomiting after the use of this acetaminophen. And his pain is like sharp and originating in the epigastric region, radiating to the back, and it is exacerbated by the movement as well. And he's also having several episodes of non-bloody, non-bilious vomiting and watery diarrhea for about 10 times a day. And he admits to decrease in the oral intake throughout the duration of his symptoms. And medical history, he is having some ventricular septal defect, and then he had some surgery as well. And other than that, the patient did not have any other history. Okay, so what do you think about this case? What will be the events and the seriousness criteria?
So the event will be abdominal pain, diarrhea, vomiting,
Mm-hmm.
and um, there were there was in seriousness criteria there was death, there was life threatening, and what else was there? Hospitalization. I forgot.
We have seriousness, death, life-threatening, hospitalization, disability, congenital anomaly, and the IME, that is important medical event.
Okay. Okay.
Like disability. Can it fall under disability seriousness criteria?
For me, I'll just consider it as important medical
Medical
event.
significant.
Yes, some
Medically
medically
significant.
significant event because the
Okay.
patient is having like diarrhea 10 times a day and vomiting also like several times, and his oral intake is less. That means the patient is at risk of dehydration, and which can lead to the patient's other symptoms as well, the severe symptoms the patients can have. So that is why I'm going to consider this event as a serious event because of these symptoms. Okay, that is how we get so many situations, especially for the post-marketing reports. And if it's a clinical trial case, we don't need to worry much because it will be already reported in the document. But for the post-marketing reports, this is how we generally get the reports. So we need to understand, we need to analyze the situation, and then we have to
Right.
decide it. Okay, the next example, let me just zoom it if it is possible. I'm not able to, yeah. You can go through it. So, I mean, these are some live practical cases I have taken so that you can have some understanding on how we get the reports. Okay, so it is about 24-year-old female medical student. She was brought to the emergency room where she has ingested 20 Tylenol and 10 aspirin in a suicidal attempt. And then she was admitted to ICU for some rising liver enzymes. And she is medically cleared and admitted to the inpatient psychiatric service. And there is a diagnosis of major depression is made. And she is having history of 2 other episodes in before the suicidal attempt. And there is some family history as well, And then later she was discharged from the hospital with the sertraline medication, and also she received some psychiatric treatment for the cognitive behavior therapy. And then 2 weeks later, she mentioned she's improved with some crying spells and improved appetite, but she's still quite depressed and has some passive suicidal ideation. That means still that thing is there. And so the dose was increased further, and then the patient is now 65% improved, but not yet euthemic, and clearly not in remission. But and the patient also had some persistent problem with the sleep, so they have added some mirtazapine medication to the sertraline. Now the patient is responding well, and however she's having some weight gain problem. Since the patient is like kind of stabilized, she's treated with sertraline at 200 mg dose, and there is some thyroid hormone as well. But currently she is in some relationship, and she is concerned about her unacceptably low libido, and the reduction in the sertraline dose to the 100 mg is associated with the improvement in her libido without any return of depressive symptoms. So actually there are multiple cases here. So first we will talk about the Tylenol and aspirin case. Okay, so
Okay.
with the suspect medications of Tylenol and aspirin, what will be the events you will select?
Attempted suicide.
Correct? And
Um, liver enzymes are increased, right?
yes, increase in the liver enzymes. And that's it, because depression is actually medical history, it is not an event. So yes,
Yeah.
suicidal attempt, and also on the rising
Rising
enzymes.
liver
So
enzymes.
what will be your seriousness criteria? Hospitalization is there. Apart from hospitalization, I'm thinking, is there any other seriousness criteria? Because they are clearly mentioned the patient is admitted in hospital. Though they have not
Yeah,
mentioned the duration, but still we can
Like
see it's like he is discharged after 7 days in the hospital. So yes,
like
she's
prolongation of existing hospitalization.
that is same like hospitalization. Anyway, we are selecting it, right? So that will be the seriousness criteria. One of, I mean, we can select multiple seriousness criteria in case. So one is clear, like we clearly know the patient is hospitalized. So
hospitalization. Yeah.
is there any other serious event you want to select?
Um, she has attempted suicide, but she also has the history, right, of 2 attempts before.
I mean,
So it
not with the suicide, but it is with the
depression.
depression. She's
Yes.
diagnosed with major depressive disorder, MDD.
Okay. Okay. Then we will life-threatening.
Yes,
She's
because
attempting
they
suicide
mentioned the
after the.
patient the patient is receiving treatment in the intensive care treatment because her liver enzymes are like very high. That's what they have mentioned. So yes,
Mhm.
we can use the life-threatening condition as a seriousness factor here because the patient is in ICU treatment. Okay,
Yeah.
so that is about one case. So we have another non-serious case, that is with the sertraline. There is an event of low libido.
Low libido.
Okay, so that is one event. So that also we can create another case. So from the same case, this is how we can create the multiple separate cases
Okay.
for the same patient.
So the first will be a serious event, like serious case, and the second
Correct.
about low libido will be under non-serious criteria, right?
Correct. Okay, and okay, let's not. I mean, this case is like bit here and there. The information is missing, so we will not discuss this. Let's discuss the one now. Interesting case here. Okay. Okay, so this is about it's a pregnancy case. Now let's see what and how we old, 30 weeks of gestation. She was admitted for elective section
We
following an
cannot.
uncomplicated
Oh.
pregnancy. And she
Rami, I cannot see the
on the
case
third
you're.
month, she underwent some abdominal.
I cannot see the case you're reading on the screen.
Is it visible
So
now?
no, it's the
This
one I
one,
can
a
see.
46-year-old
Year-old
female case,
female.
right?
Yeah. Now I can see. January 8, 2019, right?
Now,
Is that the case?
Yeah.
29
Yes.
year old. Yeah.
Yes, that's the case.
Yes, now we can see.
Yes, 21-year-old, first pregnant lady. So she is having some on 38 weeks. She has admitted for a C-section, and she's having uncomplicated pregnancy. And from third month, she's having abdominal ultrasonography, and all are normal. And she had she had history of multiple sclerosis, and for this, she's been taking the interferon beta 1. And 2 months before the prior to the decision for the pregnancy, the drug was changed to the glatiramer acetate, 3 subcutaneous injections per week. That means the patient is taking glatiramer acetate throughout her pregnancy, and the father was 33-year-old, and there was no consanguinity, and there was no family history of birth defects or any antenatal infection or exposure to any other medications, alcohol, smoking, or X-ray. And the next day, she was operated, and there was a baby girl was born, and the ABGA score is also good, which is 8. And then, but after an hour after the birth, she became cyanotic and showed the signs of respiratory distress with the scaphoid abdomen and oscillation of bowel sounds in the left chest. So she was intubated, and she was transferred to the NICU, and a chest X-ray was showed, and it showed some air-filled intestine herniated into the left chest with the shift of mediastinal struggle to the right side with the right-sided pneumothorax, and was diagnosed with congenital diaphragmatic agenesis. And there was some tube was placed, and intubation was also done, but she was immediately placed on the ventilator as well. But in the way to the operating room, she was expired. Okay, so now, yeah, what is this case, and how do we select, and how many cases we create, and then how do we select the events here?
This is like here life-threatening and death.
Yeah.
Yes, we can choose one. So if we have both life-threatening and death, we can go
Death.
with the death. We don't need to select the life-threatening again, because anyway the patient died.
Okay, then death.
Yes. So what is the event? Event
Both,
selection. That's the main here.
both defect, congenital, both defects.
Congenital diaphragmatic agenesis, because they have
Agenesis.
clearly mentioned the diagnosis. Okay.
Yeah.
And one more event. I told you in the Medra session, we have to choose a terminology in the baby cases. One standard event coding, which is drug exposure in utero. Okay, so and because that is how the
Okay.
baby was exposed to the medication, right? So the
Right,
event
right.
here will be the drug exposure in utero with the diagnosis of congenital diaphragmatic agenesis. And yeah, we have some symptoms as well, like cyanotic and
Cyanotic,
the respiratory distress with the
distress.
scaphoid abdomen and oscillation of the bowel sounds. So these all will be the symptoms for this diagnosis of congenital diaphragmatic agenesis. Okay, and yes, that is about the baby case that we create, and the seriousness here is the death.
Death.
Okay, then yep, the next case is about 74. Are you able to see the next scenario?
Yes. 74-year-old
Yes.
woman.
Mhm. She was admitted with the unrelenting diarrhea and progressive malaise for over 2 weeks period, and her stools were watery, voluminous, and non-bloody and worsened after the oral intake. So she had associated some abdominal cramping, but denied fever, chills, rashes, oral lesions, ill contacts, and the recent antibiotic use, travel, or ingestion of raw or undercooked foods. These all she have declined, and the patient is taking like Prilosec, simvastatin, metoprolol, losartan, HC hydrochlorothiazide combination, and the aspirin medications. And her history showed there is some change in medication from atorvastatin to the simvastatin 2 days before the initial events. So now our suspect medication here is the simvastatin. So that is
Simvastatin.
only the medication, the patient was changed. And 4 hours later, after the simvastatin administration of the 20 mg dose, her symptoms were like reproduced with the multiple profuse watery bowel movements, and she has remained symptom free since the discontinuation. And protein losing enteropathy has been published in one patient, so it's about some explanations. But generally, the transient diarrhea induced by simvastatin is less than 3%, and this case represents a rare presentation of severe secretory diarrhea, which has not been previously reported. Okay, so What do you think the event that we are selecting here?
The event is diarrhea and malaise.
I
Severe,
want to check.
se-
I want to check this case in the Medra. Okay, let just give me a moment, because they
Mhm.
have mentioned it's a secretory diarrhea, and they have also given some name of transient diarrhea. So let's check if we have the event
Okay.
coding option, because we need to understand. I mean, we cannot directly code it with the we cannot directly code it as a diarrhea, because it's clearly mentioned it's a transient diarrhea, and they have mentioned the secretory diarrhea as well. So now let's see.
Okay.
And yes, we have other event as well, just progressive malaise. Okay, so this is also one event we have, and then unrelenting diarrhea. These two events are there.
Diarrhea.
So what about the seriousness criteria?
Medically significant.
Yes, correct. Because we have the patient, they have clearly mentioned it's a severe diarrhea. Okay, so transient diarrhea is not there.
Severe
Secretory diarrhea.
secretory diarrhea.
Yes, we have that secretory diarrhea. Yeah, it is going to the PDF diarrhea only, but still LLT, they have clearly mentioned it as secretory diarrhea. We have to choose that LLT option. And yes, so
Okay.
we will choose the seriousness criteria as serious, because they have mentioned it as severe. So they have given it as severe. So yeah, we can consider it as a serious. Okay, so that is about the seriousness assessment. So does anyone have any questions?
Not right now.
Okay, then yes, that is it. I have. If anyone have any questions, then we can discuss, or else we can conclude our session. And meanwhile, please do work on the cases in Argus so that you will have some hands-on experience. And if you have any questions, then we can have a discussion at the end of our sessions.
Okay.
Okay, then yeah. So we are going to discuss on the another important criteria for the expedited reporting, that is causality assessment. So once we are done with our event selection and coding and the seriousness assessment, we have other two steps, that is labeling assessment and causality assessment. So today we will understand what is causality assessment, and then what do we do in the causality assessment with the different conventions that we have. So meanwhile, if you are applying for any job roles for the pharmacovigilance, you can start applying so that you will have some understanding on what is their requirement and whether it is matching to whatever we are having in our course or not. Okay, so yes, we'll start with the causality assessment. So the causality assessment, we also call it as relatedness assessment. Here we determine whether there is a reasonable possibility that the product is causally related to the adverse event or not. That means here we are checking whether the adverse event is because of the medicinal product or not. When the causality is identified, then that adverse drug event will become the adverse drug reaction. Okay, and it is a common procedure in the pharmacovigilance, and this causality is done at different levels. So initially in our case processing, so the case processor does not have a role in the causality assessment. We just copy paste whatever it is reported by the reporter. But it is a requirement of the physician. So the drug safety physician who is there in the ICSR team has to perform the causality assessment, and also for the clinical trial cases, the investigators also will do the causality assessment. And also the professionals working in drug safety department of the pharma company, that is in the, I mean, in the ICSR processing and in the in our signal management, and the risk and benefit assessment. In all the phases, we have different levels of causality assessment do happen. And when we submit the information to the regulatory authority, even the regulatory authority will also they will also do their causality assessment. Okay, and it is a key factor for identification of any new signals, and it measures the strength of evidence, and it also helps in evaluating benefit and risk profile of the pharmaceutical product. So it is not only helpful in the, actually the causality is the core for any process that we have in our pharmacovigilance process. So it has to be done correctly. Based on that only, all the signals will be identified and the benefit and risk assessment of the product will be assessed. Now we will understand how the causality is done in at the signal detection or in the clinical trial phase, or whenever we are doing the RMP plan or the risk and benefit assessment, but not as the ICSR level. ICSR level I will again explain later. So how do they assess the causality assessment? So the first thing is causality assessment for the common adverse event. That means we have some events which are being repeatedly reported with the study medication, and that event is not reported with the comparative drug and also not even with the placebo, but the incident rate is higher with the test medication. In this situation, we clearly know this event is because of the drug, and we can just see the comparison of the cases, like how many of these cases have been reported with the test medication by comparing it to the comparative drug and the placebo treatment, and then we can easily establish the causality. Then what do we do for the uncommon essays? So uncommon essays means if any event is like which is happening in 1 in 1,000 or 1 in 10,000 population, then in those situations, how do we assess our causality assessment? So here we have to consider few points. First thing, whether the patient is when the event has happened, whether the event happened after receiving the medication or not. Okay, and the next one is whether the patient's clinical experience is meeting the criteria for the adverse event of interest. That means all the lab data or any other symptoms that are associated with the adverse event or not. And also we have to check whether there is any alternative explanation, like any other medical history or the concomitant medications available. And the next one, we have to check if the AE is a type which is commonly associated with the drug exposure. That means any hepatologic or hepatic, renal or dermatologic or proarrhythmatic events. Once if anyone has taken the drug, the ways how it passed, like absorption, distribution, and metabolism and excretion. So these four factors we consider, and if that event is related to if the one of the pharmacokinetic property, then we can consider it as event associated with the medication. And also if there is no other explanation, we have to check the comparison rate with the observed rate in the database. That means we have to check if the event is, how frequent this event is being reported with the current medication to all the other medications in the database. That there is some formula we use there that I will explain when I go through the signal detection process. Okay, so for example, aplastic anemia with a background rate of perhaps 1 per million persons year, finding even one case suggests a causal relationship. So, and like that, we have some around like 80 different type of events, which we call it as designated medical events. So, for designated medical events, even with one occurrence of the designated medical event, we will consider it as a drug related. Okay, that point also we have to consider. And also we have to check if the event is because of the class of medication. That means if you see with the statins, we have rhabdomyolysis as a common event, so we have to check that point as well. And also presence of other AEs in the database which are associated with the event of interest. That means, for example, in one case it is reported as transaminitis, and in another case it is reported as hepatic failure. So these two are like kind of similar events. We will consider this point also for our causality assessment. And then in our case, we have to check for the D-challenge and the re-challenge information. And if our case has both the D-challenge and re-challenge information, then we can clearly know like this event is because of medication. So for uncommon SEs, these are the multiple factors we consider at the case level and also at the class of drug level and also we have to check with the comparison rate with the background rates as well. Okay, so is this clear, the different points? Because this is the base for our causality assessment. Irrespective of the whatever the team you are working, whether you are working in the senior management team or in the aggregate reporting or in the risk and benefit or in the ICSR. So these are the points we consider for our causality assessment. Okay.
So, Ramya,
Mm-hmm. Yes.
here you told that like compelling alternative explanation for the event.
Mm-hmm.
Like alternative explanation means.
Okay, so for example, if the patient is having some underlying history of like if the patient had already one stroke or the cardiac arrest happened at like maybe 2 years before, and now also the
Okay.
cardiac arrest is being reported as a second event. So we know the patient is already having the history of cardiac arrest. That means our drug is not the actual cause of the underlying for the cardiac arrest. Because of the underlying issue, this event might happen. And also if the patient is having hypertension or the hypercholesterolemia, so these all factors will affect the any events that are related to the heart. So like this, we have to check whether there are any other underlying conditions are there which can cause this particular event. That factor also we have to consider.
Okay.
Okay, so whenever we are doing the causality assessment for this uncommon essays, we need to be very cautious about discussing this causality, because even when when we like when we assess the causality as unrelated, that means we are actually missing on the product safety information. So we have to make sure like we are doing the correct assessment. If we miss any such scenarios, and if the in future if those cases are being repeating and many patients are having some impact, then it will have some major impact on the company's reputation. That is why whenever we are doing the causality assessment, we have to make sure that we are doing it correctly. So now I'll give some examples where it was initially considered as not because of medication, but later there are like thousands of these cases have been reported. For example, like tendon rupture with quinoline antibiotics, and there is some heart valve lesions with fenfluramine, and there is some retroperitoneal fibrosis with Sansid, and pulmonary hypertension with aminorex, and suicidal ideation with interferon and Accutane, and interception with rotavirus vaccine, and pulmonary fibrosis with amiodarone. With the first occurrence or the initial few cases, they have assessed the event as unrelated, but later they have considered that these events are because of the medications only. Okay, so since if you don't identify it initially and if you don't give the risk minimization measures, then it will have some major impact on the population. That is why our causality is the key for whatever the risk evaluations that we are doing in the later processes that we have in the pharmacovigilance. So whenever we are considering the event as drug related, then we have to do some additional analysis. So in that, first we have to check explorations for the dose dependency. That means if the event is the symptoms are worsening with the dose increase, or if the symptoms are reducing with the dose decrease, this factor we have to check. And the time to onset. So now when the event happened, whether it is acute or like the lancet of event, like whether it is a acute event or the chronic event or the delayed reaction, we have to check. Acute means if the event is happening within 24 hours, or like we have if the duration is 2 to 4 days, or within the 7 days. So this time to onset we have to consider when the event is happened. So for example, we have three cases of some Steven Johnson syndrome is happening with the medication, and in all the cases within 1 hour the patients are experiencing rash. So this level of consistency also we check within the similar type of cases. We have to check the consistency as well, whether the time to onset is consistent in all the reports or not. And then adoption and the tolerance of the certain AEs like the non-serious events, and we have to check the drug demographics and disease and the drug interactions, and we have to analyze the events based on the severity. So these are the points we will consider for our additional analysis, and there are different ways to determine the causality. So we have the first one is the global introspection, that is the physician's assessment. So they do their assessment based on their judgment. And the next one, we have some standard algorithms as well. So based on this, we use the formal definitive mechanism or the decisive tree to come to a conclusion. Okay, so here we have like imputability for the France, And we have Rosal Bucla for Venulate or the Cash Lasagna or the WHO or the Naranjo. So usually WHO and the Naranjo scales are the common causality assessments. Okay, and like this, the clinical judgment. So the clinical judgment is used at the signal management level or the risk and benefit management level, because they have to review the multiple cases and they have to review the multiple literature articles and provide the information. But by using algorithms, it is just for the ICSR reports, we majorly use these algorithms because it gives us the individual case level questionnaires which we have to check, and there will be some scores will be there at the end. Based on that, we will assess whether it is related or not related. Again, I'll explain in detail what is this questionnaires and what questions will be there in these algorithms. And the next one is we have some statistical methods like probabilistic and Bayesian analysis and other statistic methods. But generally, these methods we are not using for our causality assessment. Though we get the results, still it has to be assessed further. And to use the statistic methods, we need the well detailed cases, and that is really impossible, especially for the post marketing cases. We will not get any well documented cases, we only get the partial information. So that is why we don't, though we get the results from the statistical methods, we won't consider it as a final result. We go for further assessment of the event. Okay, and these are the common questions when we are doing, we have to check whenever we are performing the individual case level causality assessment. That means we are only checking for a single patient. So first one, we have to check, are there any previous conclusion reports on this reaction? That means whether we have received any other reports for the same event, for the same product, and also we have to check when the event happened, whether it happened after the drug receival, that time to onset we have to check, and the D-challenge and the re-challenge information, that is after drug discontinuation, whether the ADR improved or the whether after reintroduction, whether the event reoccurred or the not reoccurred, that details also we have to check. And is there any alternative cause, that is any medical history, or if the event is happening with the placebo, not only with the study medication, and if there is any drug toxicity is being identified in the blood level, in the blood, or the dose increase and dose decrease. So what is the happening with the event with the dose increase and dose decrease, and are there any events that are being reported with the similar drug, or the patient is having any similar type of reaction before this medication. That means, for example, if the patient has taken simvastatin and had some event of muscle tissue disorder, but now she switched the medication to the atorvastatin, and even with atorvastatin she is having same event. So now when we are doing the causality assessment for the atorvastatin, we have to consider the previous history of simvastatin as well, because the patient is having similar event with the similar drug which he has taken before this. This point also we have to consider. So now what we will do by considering all these points. Okay, so first we will check if the event is being already reported with the suspect medication. Okay, so here what are the points we considered here are, so first we need to check if the event is, if the product is responsible or capable to cause this particular event. That means whether it is there in the, whether this event is already documented in some literature, or this event is already there in the labeling document or on the manufacturer files, then we consider it as a clear-cut related event. And if the event is known to be frequent with this product, and also we have to check whether this event is pharmacologically predictable, that is because of any pharmacological side effect, so that information we have to check. And are there any predisposing factors related to the product exposure, that is overdose or the route of administration or medication errors. This information also we have to consider. Route of administration, we know if the patient is experiencing any skin rash after taking topical formulation, then yes, we know because of this product only this event can happen. Like this, we have to think from the product point of view as well, not only from the event point of view. Okay, so that is what I'm trying to say here. Whenever you see any event, we just not just look it from the event mechanism of action or their events property, but we have to look from the medication side as well. And these are the points we have to consider, whether this is possible because of this medication. In that possibility, we have to check pharmacological, pharmacodynamic, and pharmacokinetic, all these properties, and the drug overdose information, medication error information also we have to check. And coming to the adverse event, So from the adverse event point of view, what are the points we have to consider? So first, we have to check whether this event is often because of the medication. That means we have to see that designated medical event. So like, there are like there will be some liver injury or the hepatotoxicity or any aplastic anemia or like any drug eruptions or the blood toxidermia or neuroleptic malignant syndrome or acute liver failure. So these are the type of events which can happen only because of medication. Okay, we clearly know these events are because of medication and there are no other factors can cause these events. And how you will know that information, that we have to search in the Google, we have to understand what is the common cause for this particular disease, and later we can decide whether it is related to the medication or not. And the next one is it specifically related to the suspect medication? That means the class of medication point of view we have to consider here. First, like I said, statins will cause rhabdomyolysis, and serotonin reuptake inhibitors can cause serotonergic syndrome. Anti-HIVs can cause lipodystrophy. Isotretinoins can cause the congenital anomalies. Amidarons can cause corneal Caspar deposits. So like this, there are some commonly associated events with the class of medication. So how you will know that information? That also again you have to search in the articles or in the web before you conclude the causality point. And also we have to check whether there are any clinical or the laboratory values are available which is suggesting the drug reaction, that is any drug induced hepatitis. That means the patient's liver values are being increased after the drug intake. That point we have to check. And also if the reaction is located in situ, that is site of application or the transit or the concentration or the excretion. So based on the pharmacokinetic property of the drug, if the event is happening in any of these site, then that point also we have to consider. And the next are on the patient history, like how the previous exposure or the prior information will impact our causality assessment. In that, first we have to consider the prior exposure which is specific to the ADR. So like I have mentioned, we call this as pre-challenge in the simvastatin or the atorvastatin example, where I have mentioned the muscle tissue disorder where the patient had with simvastatin and atorvastatin both. That information we have to consider here, that is a prior exposure. And if there is any that event is already there in the patient before, that point also we have to consider. And this we usually consider for allergic reactions as well. And the next one is pharmaceutical history, that the patient is taking any other medications apart from the suspect medication, and what is the if any interactions are being reported. So usually in our drug label, we can see the multiple interactions, the drug food interactions, drug interactions, everything will be documented. So that point also we have to consider. And the next one is about the medical history. So what are the other medical history the patient is having, and also other predisposing factors like age, gender, and weight, any allergic reactions, renal failure, or the hepatic failure. These all points also we have to consider before we conclude our causality assessment. Okay, so this level of causality is majorly required for both ICSR and also for the signal management. But usually in our ICSR, like I said, we have two roles in ICSR. So the drug safe case processor will not go through all these steps of identifying causality. What we do is, as a case processor, we just look into the source document and we just go and update whatever it is reported by the reporter. If the reporter is mentioned as related, we will consider it as related, and if we think it as unrelated, then we will consider it as unrelated, and if they don't report anything, we just update it as not reported or unknown. But it is the role of drug safety physician. So in our ICSR process, we have the drug safety physician as well. So he will be responsible to check all these points and give their final assessment on the causality. Okay, so here we also consider the time to onset, the de-challenge, re-challenge information, and if any information is missing, we have to double check with the reporter. Okay, so is that clear, like how we usually check the causality? The different points. maybe that's not be helpful to you in the ICSR processing, case processing, but if you are applying for the medical review role, then you must know all these points. And if you want to grow in the signal detection and in the aggregate reporting and the risk and benefit assessment team, then this information is needed, because this is the one we majorly use in the other roles. Okay, then yeah, I'll explain what are the different levels of causality assessment, which we also call it as scale for causality assessment. So here we have the different levels, like certain, possible, probable, unrelated, unlikely. Like this, we have the different levels of causality assessment. So the first one is certain. Certain means we have the event information available or the lab data. We have all the details available, and we clearly know that the event has happened after receiving the medication. Okay, and there is no other underlying factors or the concomitant medications. And also we have de-challenge and the re-challenge information as well, and all the pharmacological and phenomenological and the pharmacodynamic profile of the drug is matching with the event. Then we consider that as certain. That means 95% we are confident that this event is because of medication. Okay, and the next one is possible or probable or likely. That means here we know that the event has happened after the drug intake, and we know we don't have any other underlying factor or the concomitant medications, and we have the de-challenge information, but we don't know about the re-challenge. In that situation, we are somewhere between 50 to the 95% of confidence in our causality assessment. Then we choose this scale, which is probable. Next one is possible. Possible means we know event happened after receiving the medication, and apart from that, we don't know whatever happened to the medication. I mean, what are the other medications, or we don't know the de-challenge and the re-challenge information. That means we are like 10 to 50% confident, like this event is because of medication. That is why we choose the scale as possible. And then we have unlikely. Unlikely means we know this event. We know, like this event or the symptoms are already there before taking the medication, or the abnormal lab value is already there before taking the medication, but it was diagnosed later. That means time to onset is not matching, and we can clearly decline the causality here. So then we can use it as unlikely or unrelated. And then we have unclassified. That means we don't know what information it is, and we need the more data to assess our causality assessment. That is when we choose it as unclassified. And it is same, unaccessible or unclassifiable means we know like this event has happened, we don't have any dates. We just have the patient is taking medication and this is the event reported, and we are with the only available information, we are unable to confirm like what actually happened to the patient. In that situation, we can choose the option of unaccessible or unclassifiable. Okay, so these are the different levels of causality assessments we have as per the WHO. In simple, this is the table we can use for the different categories in that we have in the WHO. Okay, and the next one is the Naranjo algorithm. So here first we'll see what is this algorithm. The 10 questions, if you remember the common questions which were discussed in the earlier slides, those are the 10 questions we document here. And for each and every question, if it is yes, we have certain score, and if it is no, then we also have certain scores, and we have to calculate the total score, and then we have to see what is the result. And if the result is more than 9, then it is definitely related. If it is between 5 to 8, that means it is a probable. If it is between 1 to 4, that is a possible. And if it is less than 0, then we can directly consider it as unrelated or unlikely related. And this is another type of algorithm, that is Bradford Hill criteria. So this is the major one that is used at the signal management level. But if you see the questions, that they are the same 10 questions, and we just have yes and no. If the S is higher, that is related. If it is no is higher, then that is either we can classify it as unlikely related or unclassifiable. Okay, so yes, these are the different scales that we have. And coming to the clues to the causality. So as per SIOMS 3, it's like in detail guidelines. In this in detail in these guidelines they have mentioned in like very detailed data, like which points we have to consider for our causality assessment. But right now I'll just give you the overview of what actually it is there in the SIOM's guidance. So the first one is the positive de-challenge and re-challenge. So I mean, they have given a list where we can consider these points to consider the causality as related. So first one is the positive de-challenge and the re-challenge, and event is being happening with the the study medication for the clinical trial cases. And also there is some statistically significant differences in the clinical trials. That means event is being only reported with the study medication but not with the placebo and the other medication. And whether the event is because of overdose of the drug, and there is some pharmacokinetic evidence for interaction. Okay, and this event is already observed in the preclinical trials or in the animal studies, and the frequency with the study medication compared to the placebo. And this event is because of medication, which we call it as known mechanism, and it is recognized class effect. That means the class of medication effects. And what are the cases that we are receiving? We are having some consistent symptoms and also consistent time to onset. And the definitive cases where we have all the details, which we call them as definitive cases, where we have positive de-challenge, re-challenge, and all the other details about the event. And similar properties to the drugs with this AE, that means the similar class of medication. This AE is being reported commonly. And the time to onset is plausible. That means it is acute, subacute, or the late event. And it is identifiable subgroup at risk. That means the pregnant women, like if any congenital anomalies are being reported, and also if any pediatric events are being reported or any elderly events are being reported, or if this event is being reported only with the underlying condition of hepatic failure or the renal failure. These points also, if it is there, then we can consider it as related. And if you're getting high frequency of these events and it is biologically plausible, and if the event occur that is usually drug related, that is designated medical events, and there are no other lack of factors, lack of other confounding factors, and amount and duration of drug exposure is appropriate, and the drug is affecting in the same way in all the cases. And reporters feel that the AE is because of medication. That means if the reporter has considered it as related, then we must submit the information as related only. And low background incidents, that means without the medication, this event has never occurred to the patients in the current population, and only with the medication this event is being reported. And the cases have some clear cut, and we can easily evaluate the case. And if we need to have the objective data, and there is no other obvious alternative explanations, and there are no conmates, and if it is reported to occur only in the children or the high status capable reporters, although there is no other corroborative evidence, there is no contrary evidence as well. Even this point also we have to consider for our causality assessment. So for under this, in the SIOMS 3, they have provided multiple examples and explanations, but I think at this level we don't need to understand all this information. We just need to know, but these are the factors which are used at the high level causality assessment. Okay, so now let's see an example. Examples of some definitely related. So what kind of events can be considered as definitely related or possibly probably related or unrelated? Okay, so here there is some dizziness happen 3/4 hour after taking an oral antihypertensive drug with no concomitant drugs. That and event also stopped after the drug withdrawal, and also there is some positive information as well, that is positive re-challenge information. So we clearly know antihypertensives can lower the blood pressure. That is why the patient is having dizziness. And also the de-challenge and re-challenge is there, so it is clearly falling. The time to onset is there, we have pharmacodynamic profile is there, de-challenge and re-challenge. So yes, this case is definitely related. And also any injection site reaction which is happened 30 seconds after subcutaneous injection, and if the tablet got stuck in the pharynx while swallowing it and it has to be removed in the emergency room, or any anaphylactic reactions, we clearly know these are because of medications. And coming to the probably related, any event that happened, because so here you can see the pharmacodynamic property, that is thrombocytopenia after oncology drug, diarrhea with ampicillin, and vaginal candidiasis with antibiotic for bronchitis. And possibly we have any abnormal liver function test or headache, dyspepsia after a tablet or the capsule. Unlikely related means cancer of the colon, which is diagnosed after 3 doses of an antibiotic, and we clearly know that the cancer cannot occur within the 3 days after taking any medications. And also any myocardial infarctions like 3 weeks after taking a drug which has a terminal half-life of 10 minutes. So here you have to consider half-life of the product as well. So here the product has only 10-minute half-life, but the event happened after 10 weeks, sorry, 3 weeks. So we clearly know that is not because of medication. Okay, and then coming to the cascade or the secondary A's. So maybe we have so many documentations are there and we have so much guidelines are there for our causality assessment. But at the end of the day, there is no clear-cut sample on how we do the causality assessment, because each and every person's analysis will be different. Each and every case that we receive are different. So now let's see one of such example, which is cascade or secondary AEs. So cascade AE means the event is happening because of the event that the patient have. So here if you see the example, the patient is taking drug which has caused the anemia. Because of anemia, the patient experienced myocardial infarction. In this situation, how we will perform the causality? Okay, so it's like really difficult here. Okay, and like I said, there is no standard way of identifying causality for the medications, and especially for the secondary or the cascade AEs. In this situation, usually it's completely upon the physician, whoever is handling it. Few physicians may consider it as related, few can consider it as unrelated, and we cannot say anything. Either way, it will be correct only. And so if you see another example about the drug which has caused dizziness and causing the patient to fall and the break her leg. So in this situation, what we will consider here, we cannot consider it as related, or either we cannot consider it as unrelated as well. So that is why, like I said, there is no clear answer in all the cases. It may be appropriate to mention this in the IB or the product labeling for some ways, but it's not always. We cannot perform the causality in a clear-cut way. And though we have the different classifications, the different scale of causality assessment at our end, but when the report is getting submitted to regulatory authority, either it will submit as related or unrelated. All the other classifications we have will go as related, only unrelated will go as unrelated. Okay. So is this clear, or does anyone have any questions before we go into the examples?
No.
Okay, so now let's see the example. So you can let me know what do you consider the causality here. So it is about 65-year-old male, obese, smoker, hypertensive, type 2 diabetic with a hypercholesterolemia. He started with the new drug and has an inferior wall myocardial infarction 8 days after starting. Okay, so what do you think about this case? How do you consider whether it is related or unrelated?
I think it is unrelated because the patient already has hypercholesteremia. He's obese, he's hypertensive, all the risk factors for myocardial infarction are there before
Okay,
starting
yes.
the drug.
Yes, so that's your assessment is correct. But now we see how if we have the more information. I mean, we cannot clearly decline it because we can just either say it as like unlikely or remotely related, because we don't have clear-cut
Okay.
information of rechallenge all the information. Now let's see how underlying factor or other information will impact. So here, if you know, we have the patient medical history, the family history. The patient's parent are like both smoke and have elevated cholesterol, and they're 98-year-old. Okay, and he only took one dose and stopped it because it gave him headache, and the terminal half-life is 20 minutes. So with this information, you can clearly decline the causality. And then it is also mentioned his 20-year-old dog died the day before, and this was very stressful. And he also worked in the garden for 5 hours the day of the attack. Okay, so if we have all this information, we can clearly and confidently say yes,
It is not related. But in this case, like, are we provided only the first scenario, the case that you gave, only that much information, or all these information is also provided, or we have to find all these information to check whether it is related or not?
Yes, so that's what any well-documented case will be easy for us in the causality assessment. But if only, like you
Okay.
mentioned, only if we have the initial information, we cannot clearly
Mm-hmm.
decline the causality there. But here we can clearly decline the causality.
Okay, got it.
Okay, let's see another example. A pregnant woman takes a drug during the early in her pregnancy to prevent a miscarriage, and 14 years later, her daughter developed the cancer of vagina. So, what do you think about this?
Not related. You cannot say
Okay,
completely unrelated
and we cannot say related as well.
as well,
Okay.
yeah.
Yeah, actually, this
It may
case
or
was
may
a
not.
yeah, this situation was kind of sensation. It happened in some decades ago, but still we consider
Mm-hmm.
this as one of the example for our causality assessment. So the
Okay.
medication here is diethylstilbestrol, which is an estrogen. So it was prescribed to prevent the miscarriage, and as a result, almost a millions of pregnant women has taken this medication.
Mm-hmm.
And in 1970s, there was some study published stating like there is a woman. I mean, they have clearly mentioned this drug cannot prevent the miscarriage and the premature births because it's an estrogen, but not the progesterone. So, however, it was still continued until 1971. The patients are still using this medication. And later, in 1970s, they have initially identified some rare vaginal cancer in the girls and young women who had been exposed to the DES before the birth, that is in utero. So later, they have done so many analyses and there are so many studies are done. So clearly, they have identified like all the daughters whoever has exposed to this medication had this vaginal cancer.
Okay.
Okay, that's what I said. So there will be events which can happen after years of taking the medication, which we call it as long onset or
Yeah.
the delayed reactions. Okay, and yeah, can a drug can cause nausea and vomiting before exposure to it? Is it possible?
No,
No,
Okay.
no.
it is
Yeah,
only
but
psychological.
yes, that's what is happened with the cisplatinum anticipatory vomiting. So initially, for the first cycle, all these patients have experienced the nausea and vomiting, and then in the next cycle, psychologically, they had feeling like yes, this is because of medication only. I mean, this can happen to me. So before taking the chemotherapy, they have started having nausea and vomiting. Okay, and the next example about 75-year-old female, she has taken nitroglycerin for the chest pain and gets dizzy, and she fell and break her hip, has some surgery, recovered, and then developed pneumonia, but later she died 12 days later. So this is a kind of example of cascade or the secondary A, and it's like really critical in this example, especially to say because like I said, it's either we cannot
Mm.
decline or we
We
cannot
cannot
even
say yes or
consider it
no.
as related as well. And if we
Yeah.
con- if we consider it as related, where do we update in the labeling document? So for any event which we consider it as related, it has to go into the labeling document. So where do we
Okay.
update it? Whether in caution, precautions, or so we don't know where to put this information as well. That is why, for the safer side, I mean, when I worked with the different medical reviewers have the different opinions. So it's like I say, it's completely depending on them, on their mood as well. So if they want to keep it as related, they'll just keep it. If they want to keep it as unrelated, they just keep it as unrelated. So that is how we go ahead with the secondary and the cascade A's. And we don't have proper guidance as well on how do we handle these situations. Okay, that's I mean that I'm like saying if you're going for the medical review role and if you see any
Right.
secondary A, just take your opinion. If you want to write the detailed justification, you can go ahead and do whatever the assessment we can do for the cascade A's. So next one. So there is a drug for the weight loss which is heavily promoted to the teenagers, and there are almost 10,000 women between the ages of 11 and 21 has taken the drug in the first 3 months of marketing. Okay, and initially there is a case of mitral valve stenosis is reported from the Paris. Okay, so we have only this information initially. So if only this information, we don't know. I mean, we
Yeah.
cannot
It
assess the
can be
possibility here.
related. It cannot.
Yeah,
It may not
but
be. Yeah.
later, yeah, there is some famous person. She has mentioned that she took it and she lost the weight as well, but she had heart
Had
attack.
heart.
Okay, so now we have one more report. And after almost 73 cases of heart attacks and 14 mitral stenosis are reported in the next 2 weeks. Okay, so now since there is a high frequency of reports, we can consider it
Yeah,
as
Related.
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 Okay, so
Yeah.
so what are the examples or what are the guidance you have learned in the earlier slides? This is how it will be helpful. So if we have the high frequency, we can consider it as related, or else we cannot consider it as related event. Okay, so there is no conclusion for causality. So causality assessment is always tricky assessment that we have. And say like for the seriousness assessment or the labeling or the event selection and coding, we have the proper guidelines available.
Guidelines. Yeah.
Causality also, we have so many conventions and guidelines, but at the end of the day, it is always depending on the person, whoever is doing the causality assessment. But as per the
Right.
guidelines, we have to follow whatever the points that are there, but not uh, we don't, like I said, no, even though it's like if it is like I say, for seriousness, if it is reported as death, then it has to be death. It is not like that for
Right.
causality. So
Right.
that is why it's always tricky, the causality assessment is. And I hope you got some understanding on how we actually do the causality assessment. But if you go for the signal management, so in the signal team and the risk and benefit assessment team or the safety management team, so they will have some huge debates on whether to consider it as related or unrelated. One will say as related, one will say as unrelated, and they will have some heated discussions. Because I was a part of one of that discussion, I was like, why did I even go there? So that's how bad it will be, the discussions. They will talk about from the all the medical point of view, and from the there will be some product leads, because they are the research from the, we will have some people from research and development team as well. They will come and they will give their opinions from the product point of view, and the doctors will give their opinion from their event point of view and from the case
Okay.
point of view. So they will discuss a lot of things, because any event. So for example, if it is a normal serious events like any anaphylactic reactions or something, though, I mean, we don't need to have some such discussions, because we know we cannot this can happen because of the patient factor. But the events like any hepatotoxicity or aplastic anemia, or any designated medical events like hepatotoxicity or any kidney injuries, so these kind of things, to consider it as drug related, we need to have some solid evidence, and any of such reactions, it will impact the product marketing status. That is why they will have this kind of the various agreements, or sorry, various discussions to come to a conclusion whether it has to be considered as related or unrelated.
Okay.
Okay, so majorly causality is really important at the other levels that we have in the PV process. But yes, we do our initial assessment in the ICSR, because ICSR is the key for other process that we have in the pharmacovigilance. So, but the case processor, we don't do anything, but the medical reviewers, they do all this initial assessment by checking all the 10 questions that we have in the causality assessment.
Okay. So the individual, that is ICSR, it is only individual case, right? So like the
Yes.
case processor will bring all the similar cases together, and the medical reviewer will review them and do the assessment. Is that how it works?
No, case processor, he will just update whatever it is reported by the reporter. Okay, and even the medical reviewer, when he is doing the case level, he will only look at the case level causality assessment. He will only just look at the case.
Individual,
So
each individual case.
yeah.
Okay.
Like example, we
Okay.
have discussed what is the medical
Okay.
history, or are there any drug interactions, D-challenge and the re-challenge, time to onset. So only these are the few factors we consider for our individual case level assessment. And all the other
Okay.
points we use for our the next processes that we have in the pharmacovigilance.
Okay.
So Ramya, medical reviewer means what are all the job done by drug safety associate will be reviewed by MR, right?
Correct. Yes, they have to confirm it.
Okay.
Any mistakes that we do in the event coding or in the labeling or the causality, we can just directly put on the medical reviewers. We can say
Okay.
they have reviewed, but they have not said it's incorrect or something.
So the medical reviewer has to review each and every case, right?
Yes,
Done by
correct.
the. Okay.
Not all the cases, only expedited reporting cases. Medical
Okay.
review is not required for all the cases. Only for the reports which you are submitting, only those cases will need the medical review.
For serious
Okay.
cases only, right? Medical
Correct.
reviewer
Serious.
needs to review.
Yeah, usually
For
serious
non-serious,
cases.
for
Not
non-serious
required.
cases, we can directly submit.
Yeah, usually it is not required, but for some US companies, they also do the non-serious cases, so especially US cases to go to the medical review. Because FDA has a requirement of this PADR reports. In the PADR, we include all the non-serious reports as well. That's again, yeah, company specific convention it is. So you can, once you join any company, you will know which case will go for medical review. Usually serious cases is the one we will go for the medical review. And also in the serious cases, we won't send unrelated. If the reporter has clearly mentioned it as unrelated, that cases we will not send for the medical review. Because
Okay.
unrelated cases will not go for submission as well.
Okay.
Mhm. Okay, Ramya.
Okay, then yes, that's it I have. If any questions, we can have discussion, or else, yeah, we can conclude our session for today.
Okay.
Okay, Ramya.
No
Thank
questions
you.
for now. Thank you so much.
Okay, then yeah, bye bye.